Good morning. The Public Health Institute of Chile welcomes you and is thankful for your attendance to the 17th Scientific Conference. Today we have representatives from the academia, the industry, scientific societies, professionals associations, students, other health institutions, lecturers, and of course workers from the Public Health Institute. Welcome, everyone. This year, our goal is to be the promoters of science and regulation for public health. And specifically, today's topic, which is organized by the Department of the National Agency of Medical Devices, is called Regulation, a Pillar in Innovation and Development. A main uh, core function of the regulatory systems is promoting and protecting public health and supervising quality, safety, and effectiveness of medical devices. Chile is currently facing several gaps and challenges regarding medical devices regulation and devices for diagnosis in vitro. To do so, several strategies have been implemented in the legal and regulation framework, which is why on January 31st, the executive power presented the project to modernize the national system for health services in Congress, and it considers as a main goal to provide new powers to the Public Health Institute in terms of medical devices regulation in order to the regulation framework according proposed by the World Health Organization. A solid regulation that will promote innovation in health, competitiveness, and the development of the national productive industry. In our country, we have led several innovation endeavors, and not just that, there are several programs that are promoting this interaction. Before beginning with our conference, we invite you to please silence your phone. The first conference of the day is via streaming, and it's called Activities of Regulation Strengthening of the World Health Organization, Good Regulatory Practices and Good Reliance Practices. The presenter is Dr. Agnes Quijo from World Health Organization, OMS. Agnes is a pharmacist and holds a Master of Science degree in pharmaceutical technology. She has more than 23 years of experience in the regulation of drugs and medical devices. She is currently working as a technical officer at the World Health Organization headquarters in Geneva, Switzerland, and is responsible for the collaborative registration procedure for in vitro diagnostic medical devices and other activities related to facilitated regulatory pathways. Prior to joining WHO in March 2020, she worked for 19 years as a regulator in various technical, advisory, and leadership roles, enabling her to provide guidance and advice to harmonization initiatives globally. Now I give the floor to Dr. Agnes Quijo.
Hello, everybody. Can somebody confirm if you can see my screen well? Yes, so thank you. I guess I should proceed with my presentation. It is a great pleasure to be here today on this auspicious occasion to provide an overview of WHO's overview of WHO's collaboration with countries in strengthening regulatory systems. I would like to extend my sincere thanks or appreciation to the organizers for inviting WHO and for the continued collaborative spirit demonstrated by ISP Chile over the years. So this is the outline of my presentation. I understand my presentation is going to be for around 20 minutes. And I'm going to take you through the regulatory system strengthening activities that are undertaken by WHO, including the GBT plus medical devices, but also share with you some of the regulatory harmonization, convergence, and networking activities or initiatives that are spearheaded by WHO, and also good regulatory practices, good reliance practices, and then I would end up with one slide on conclusion. So before I dive into the specifics of regulatory strengthening activities, I'd like to briefly provide some context on why WHO is engaged in this work. Our mandate stems from the WHO World Health Assembly Resolution 67.20, adopted in May 2014. You may have heard of this resolution many times. It is in fact the cornerstone of our efforts. It underpins WHO's commitment to support member states in both achieving the sustaining effective regulatory oversight through the WHO Regulatory System Strengthening Program. The ultimate goal, as you can see also on this slide, the ultimate goal of this program is to promote access to quality assured medical products. To achieve this, we focus on two objectives, which are also highlighted on this slide. So one is building regulatory capacity in member states aligned with the good regulatory practices, which I will elaborate on it in the next few slides. And then fostering regulatory cooperation, harmonization and convergence of requirements and approaches. This includes enhancing transparency and promoting networking, essential elements for building trust and encouraging regulatory reliance. So this is what gives WHO the mandate and what are the focused objectives that we have as far as regulatory system strengthening is concerned. So to begin with, one of the important WHO regulatory strengthening activities, it is the WHO Global Benchmarking Tool. So WHO developed this innovative tool known as the Global Benchmarking Tool, initially introduced in 1997 for medicines and vaccines. It was later expanded, that you can also see on the slide. It was later expanded to include blood and blood products in 2019. And most recently, in 2024, a specific global benchmarking tool for medical devices was launched. So this is quite a big progress as far as the introduction of the GBT plus medical device, which we see it evolving and expanding to include also other medical products. So what is the GBT? Because some of you, it could be your first time to hear about the global benchmarking tool. So the global benchmarking tool is WHO's primary mechanism for objectively and consistently evaluating the performance of national regulatory systems as mandated by the World Health Assembly Resolution 67.20 that I mentioned on my previous slide. So using this tool, WHO conducts benchmarking assessments at the request of countries. These assessments identify institutional development plans, or in other words, you can call them gaps, which highlights regulatory gaps. So based on these findings, then WHO provides capacity building support through technical assistance, training, and networking opportunities in collaboration with other development partners. So the process is cyclical and includes continuous monitoring and documentation of outcomes and impact, ensuring that progress is tracked and that improvements are sustained over time. Because it is one thing to receive, for example, a maturity level 3 status, but I think it is also very important that the status is maintained throughout the life of the National Regulatory Authority. And so both ensuring that the standards are met, but also continuous monitoring of the progress, and also maintaining the impact based on the maturity assigned on the specific medical product. So this is just a slide that indicates the link where you can have access to the GBT plus medical devices version 2, which was published in December 2024. I would like to urge the NRA, but also the other stakeholders who are participating in this scientific conference, to find time to go through the specific sub-indicators for medical devices, because these are the sub-indicators that you will be assessed for in case, for example, ISP Chile decides to request for formal benchmarking using the GBT plus medical devices. But one of the things that I've always been advising national regulatory authorities is to also use the opportunity and take advantage of the availability of this published document to conduct self-assessment to address some of the gaps or IDPs that will be identified internally before requesting for formal benchmarking. So key message here is the link. I've provided the link on this particular slide, but also familiarize yourself with the sub-indicators. So whether you are within the NRA or outside, but most important is to conduct self-assessment and familiarize yourself also with some of the identified gaps, which can really be helpful when the time for formal benchmarking arrives. So this is the key message on this particular slide. So I want also to speak briefly on the second important objective of the WHO regulatory strengthening activities. So the second biggest activity is this support on global, regional, and regional harmonization initiatives. We do conduct a lot of meetings within WHO. I'll go back to the bullet points I've put on this slide, but I just wanted to mention that there are a number of meetings that WHO conducts with stakeholders in the year. For example, there is one big meeting that we do conduct with regulatory authorities. I'm not so sure if Chile attended the meeting last year, but it's called International Conference of Drug Regulatory Authorities. Although it reads drug, but it also covers medical devices. It is usually conducted every 2 years and gather all regulatory authorities and with full sponsorship of countries from low and middle income countries. So the last meeting was conducted in 2024 in India, a very successful ICTRA meeting where medical devices was given a special attention. And the next one will be conducted in 2026 in Saudi Arabia. There is also another meeting that is to happen this year. It's a global forum for medical devices, which is planned from 2nd of June to 4th of June remotely. And there is also another meeting on collaborative registration procedure, which I'm also going to speak about on my next slides. This is a model on reliance, but we usually have annual meetings every year and invite countries that are actively participating in the collaborative registration procedure. But I think the reason why I'm mentioning all these meetings is for you to see how we are investing in fostering regulatory cooperation, harmonization, networking, and also convergence of requirements and approaches. Because in this meeting, these are some of the things that are discussed between the national regulatory authorities to ensure that there is harmonization of of regulatory requirements, but also approaches which are introduced by individual regions or by individual countries. And in this particular slide, I've indicated some of the global and regional harmonization initiatives that WHO collaborates with or work with them to support what they do. So on the global level is the International Medical Devices Regulators Forum, in which I believe Chile also is a participant or is a member, but also in the regional harmonization initiatives, we support the Africa Medical Devices Forum. Recently, we have supported the group to establish a specific medical devices assessment technical committee to advance in regional joint activities, which may include assessment of dossiers, but also joint audit inspections, joint audits of the manufacturing facilities, and also identification of facilities for performance evaluation. And then we also work collaboratively, but this mostly is for medicines and vaccine with the Asian Pacific Economic Cooperation, but also very closely with the Global Harmonization Working Party and the Southeast Asia Regulatory Network. So during our interaction with all these global and regional harmonization initiatives, we collaborate in or contribute to the development of different guidelines, support, for example, in some strategies that individual regional harmonization initiative may wish to advance. For example, you might be aware that the IMDRF has recently called for comments on a playbook on reliance programs. So WHO is working very closely with the IMDRF to make sure that we also provide our feedback and comments in terms of how best the issue of reliance can be addressed through this playbook. So this is a way that WHO supports both global and regional harmonization initiatives, including also conducting or participating in specific joint activities, especially on assessment of technical files in some of the regional harmonization initiatives. So this is also one of the biggest area that we support. So I want to speak a little bit on the good regulatory practice. I'm not going to go, I know everybody might be aware of this important document that was published by WHO in 2021. This is a guidance document which was developed by WHO to support regulators in the oversight of medical products. I also mentioned that in the GBT for medicine and vaccines, blood and blood products, and for medical devices, we have made sure that all the elements of the good regulatory practices are also addressed in the GBT plus medical devices and other also versions for other medical products. So by the time when a formal benchmarking is conducted in a regulatory authority, we ensure that by addressing the sub-indicators in the GBT plus medical devices, the NRA will also be addressing elements on good regulatory practices. And so this is a document that was developed by WHO to ensure that the established systems are scientifically sound, affordable, and responsive to public health needs. And I think one of the points that I want to make is that the good regulatory practices is applicable to all national regulatory authorities, regardless of the maturity or regardless of the resources that they have. And this is why it is very important that regulators also adapt some of these elements that are proposed in the good regulatory practices. And the good regulatory practice framework is built around principles and enablers that apply to the three components of a regulatory system. So if you look at this particular slide, there is a place where I've indicated regulatory systems. So regulatory systems are made up of three important components. One component is the regulatory framework. The second important component is the regulatory institution. And then the third important element is the regulatory resources. So I will start with the regulatory framework as one of the main components of a regulatory system. So regulatory framework, this includes legislation, regulation, or decrees and guidelines. So when developing or reviewing a regulatory framework, it's essential that all the principles and enablers that I have indicated on the right-hand side of this particular slide are incorporated in order to have or to experience regulatory impact. So very important that when you're developing or when you're reviewing your regulatory framework, it is important that all the principles and all the GRP enablers are considered in these important documents. And then there is also another important component of the regulatory system, which is the regulatory institution, which is the National Regulatory Authority. So the organizational structure of the National Regulatory Authority involved in regulation must also be aligned with the good regulatory practices. We are talking about, for example, efficiency. We are talking about clarity. We are talking about independence. National regulatory authorities need to be independent. And so very important that this is demonstrated when establishing a national regulatory authority. But another important element are the enabler. Does the NRA, for example, has political and government support? Very, very important. Is there any effective communication and collaboration and coordination within the national regulatory authorities? Just to mention a few. Another important component that supports the regulatory system is the resources. So do you have, for example, adequate and sustainable human, technical, and financial resources? These are crucial to make sure that there is a stable regulatory system. So in short, for good regulatory practices to be effectively applied and implemented, these principles enablers must be established or strengthened across all levels of the system. And doing so helps ensure the desired regulatory outcomes and public health impact are achieved. So this is what is summarized basically in this particular slide. So few takeaway messages as far as good regulatory practice is concerned. So one is it is important to understand that the good regulatory practices serves as a benchmark. So when you're developing regulatory systems, when you're implementing regulatory system, or when you're reviewing any of the regulatory instruments, critical that GRP principles and enablers are addressed. And of course, this forms as a benchmark for all national regulatory authorities. It is applied in the whole regulatory system. I've mentioned the three important components of the regulatory system, but also it guides NRI in prioritizing their regulatory activities according to resources. For example, there is one principle that talks about flexibility. We all remember during the COVID pandemic how this particular principle became critical to allow availability of a quality assured diagnostics, but also other therapeutics. And so very important that when prioritizing, these principles are also considered. It is also important, I also mentioned this when I was talking about the GBT+, that these principles should be implemented consistently and effectively. Apart from that, then the regulatory systems will not be as stable. So very important that there is consistency in the regulatory systems, but also implemented effectively in order to really realize the regulatory impact, which is having in the market quality assured medical products. But the good regulatory practice also helps in setting up requirements and formulating decisions based on the principles, which will help in the manufacture, import, and of course, distribution of quality assured product. So I will jump quickly to the other important element that I wanted to talk about, which is the good reliance, practice. So in the good reliance practice, I know it's a busy slide, but I think key messages here is that regulatory work can be overwhelming, and this may result into lengthy regulatory approval for the needed medical products. And we all agree that patients are always waiting to receive the product. So timely access is important to make sure that the much needed quality assured products are available in the market. And therefore, reliance is used as a tool to facilitate and accelerate the regulatory decision and, of course, access to the quality assured products by leveraging decision or information or work that has been conducted by another regulatory authority, and in most cases, a more advanced or another trusted regulatory authority. I'm not going to read about the advantages, but I think one of the things that I wanted to emphasize is that there is no point of duplication. There is no point of repeating the work that has already been done by somebody else, and therefore timely access to quality assured products can only be realized when good reliance practices are adopted within the regulatory systems. I'm going to give an example of the WHO collaborative registration procedure. So this is one of the successful reliance model. So instead of going through the concept, I wanted to just give an example of the WHO collaborative registration procedure for in vitro diagnostics, which has really facilitated and reduced the timelines for assessing applications that we are going in some countries who are currently participating in the WHO collaborative registration procedure. So how does this work? Maybe some of you might have heard already about the collaborative registration procedure, but this is a collaboration between three parties. So it is WHO, the manufacturer, or we call them applicants, but also the national regulatory authorities. So what WHO does, it shares the report or the assessment outcome of WHO pre-qualified in vitro diagnostics. At the moment, the scope is only for some specific critical in vitro diagnostics, and we plan maybe in the near future, maybe next year, to also expand the scope to medical devices. But at the moment, it's just the in vitro diagnostics that I have put on the right-hand side in blue colors of the slide. So it requires manufacturer consent. So this information, as much as WHO is the one that has assessed this pre-qualified product, but the information is still manufacturers. And so we require the manufacturer to consent that we share these reports with the interested NRAs. But also the information sharing component is very important because we tend to share this information in a very highly confidential manner, considering the sensitivity of the information that we are sharing. And also maybe to clarify that the information that we are sharing is unredacted. So it is the same information as was assessed by WHO. We are not reducing any of the information. It is shared by the NRA. And the three reports that are shared is the dossier assessment report, site audit report based on ISO 13485, and also performance evaluation subjected to that assay. And then, so NRA used the WHO report or the manufacturer's report to expedite their review. And of course, the registration process with the timeline of 90 days. So 90 days is the maximum time. You can decide to maybe register within 10 days, 5 days, but 90 days is the maximum time. And the outcome is faster regulatory approval and access to quality assured IVDs. I also want just to show you briefly, and I think I have 2 more slides to go. This is just to give you a snapshot of the countries that are participating in the CRP for IVDs. Of course, I don't see Chile here, and I believe maybe after this conference we will also have Chile participating if they are interested to do so. This is a voluntary mechanism, so we don't force anybody to join. But you can also see that at the in AMRO or PAHO region, there is no NRA that is participating in this important mechanism, but we have 91% participation at the moment in Africa, and we have 35 agreements, and we have been able to register 38 products through the collaborative registration procedure in 2024 with a medium registration timeline of 81 working days. So this has proved to be a very important and very efficient way of introducing products rather than repeating the assessment that WHO has already conducted. I'm going to skip this particular slide because it further highlights some of the resources and the tools that we have, including also a very successful meeting that we conducted last year in Jakarta to discuss also implementation of the collaborative registration procedure for all medical products. So in concluding my presentation, and I hope I'm still on time, rollout, I'm going to start with the GBT because I started with the GBT at the beginning. So rollout of the GBT plus medical device, I believe, will transform the regulatory landscape for medical devices and IVDs based on the nicely crafted indicators that addresses some specific elements on the regulatory systems for medical devices. So once the NRA is able to address all the needs of the sub-indicators in the GBT plus medical devices, they will be able to advance to maturity level 3, and of course, higher maturities like maturity level 4, confirming that, the products that are translating that the products that are available in the market are of good quality because of the strong system that the NRA would demonstrate. But also this would also form basis for reliance, because one would want to rely from a national regulatory authority that has been systematically assessed and has been assigned a higher maturity level than the other. So as we are providing capacity or support to capacitate NRAs, but at the same time, we are also providing support to other NRA to select which countries to rely with. And also maybe to mention that this development of the GBT plus medical devices was developed based on the principles recommended in the WHO Global Model Regulatory Framework, which was also heavily revised in 2023. Another important element is on the regulatory cooperation, which I've also mentioned in some of my slides, a need to further cooperate, network, conducting work sharing and reliance mechanisms between the NRAs or between the regional harmonization initiatives. But this, for WHO, it remains to be a priority. We have seen a lot of positive aspects arising from these interactions. And of course, encouraging trust and confidence building between the national regulatory authorities, because it provides a platform for regulators to demonstrate or to showcase what they have in their national regulatory systems, and therefore giving opportunity for other regulators to either rely or to be able to know where to go when they need to have a specific or to address a specific gap. And then another important area that we are looking at really is the alignment and harmonization of regulatory harmonization initiatives. We've seen recently that there are a number of groups that are working towards regulatory harmonization. And so we want also to get to a point where through these regulatory harmonization initiatives, there is also alignment and there is also harmonization. And this is why WHO attends to the IMDRF, attends the meetings of the GHWP and any other regional harmonization initiatives of interest. We participate in making sure that the elements that are in the WHO Global Model Regulatory Framework and the GBT plus medical devices are also addressed. And then last but not the least now is the WHO listed authority. So at the moment, this policy is only for medicines, but it is expected that this policy will be expanded to also integrate medical devices, including IVDs. And so once the National Regulatory Authority has been listed by WHO, it also forms a strong basis for reliance. And so the WLA policy is building on the lessons from the use of the GBT plus medical devices. So once a national regulatory authority is assigned with maturity level 3, there is a performance evaluation which is conducted on top of the maturity level status to make sure that there is a comprehensive assessment. And once that has been demonstrated, then the NRA is listed as by WHO as WLA, and it forms as a basis for other NRIs to make reference as far as the requirements are concerned or regulatory systems are concerned. So in a nutshell, this is what I wanted to share with you this morning on your side, evening on my side. And I'm happy to take up any question. But once again, I would like to thank the organizers for providing this opportunity for WHO to present on the important work that we are prioritizing within WHO. So thank you very much. And I will hand it over back to the organizers. Thank you.
We are thankful for all of the organizations and good regulatory practices and good reliance practices. And we'd also like to comment all of the attendance that Chile and ISP has been participating as a member state in the harmonization group, GHWP, the ones that the doctor mentioned during her presentation. And since 2023, we have been part of the forum of IMDRF as an affiliate member. The participation of this medical devices team has allowed us to know all of the recommendations that are presented by Dr. Agnes, and all of these years we have been working with our teams, working on different initiatives to enable these regulatory activities that Chile needs for in vitro diagnosis devices. Dr. Agnes, once again, thank you very much for explaining very clearly the guidance, the guidelines of for ISP and for medical devices. And from our end, we can confirm that we are adopting all of these initiatives and recommendations from the World Health Organization and the Forum of Regulators of Medical Devices. We have a tight agenda, so thank you. Another round of applause for Dr. Agnes. And thank you so much for your willingness to support the activities of ISP. So thank you. Next conference, also via streaming, is called Introduction to the Program of Auditing of Medical Devices, MDSAP, and Dr. Marcos Barbosa is presenting from Brazil, from the National Agency of Health Surveillance from Brazil. He's a specialist in health authority and with ISO certificate. He has more than 18 years of experience in ANVISA, in registration, inspection, and medical device surveillance. He has more than 15 years of experience as an auditor in the certification of good practices of fabrication of medical devices by ANVISA, the agency, and 2 years of experience as the evaluator in the program of auditing of our medical devices. I give the floor now to Dr. Marcos Barbosa. And let's confirm that he is connected.
Bom dia a todos. Perfeito. Então, sou Marcos Jucá, Estou falando de Brasília, do Brasil. Gostaria de agradecer à organização por ter convidado a Anvisa para participar desse evento, 17ª Jornada Científica do Chile.
Escúchalo a ellos.
Dividir minha tela com vocês, que só um segundo.
No. ¿Tú sí? Yo no escucho. Bueno, no están hablando.
Consegue ver minha tela? Alguém pode me confirmar se a minha tela é visível?
Can you please confirm that you can see the screen?
Perfeito. Então, tenho alguns minutos para falar do programa MDSAP.
Perfect. I have a few minutes to talk about the program Medical Device Single Audit Program.
Segundo, eu estou apanhando do...
Please give us a
Não
second
estou conseguindo
because
passar
I'm working out the
o próximo
Zoom
slide.
and I cannot move to the next slide. All right.
Então, o que é o programa MDSAP?
So, what's the MDSAP program?
Ele falou sobre a harmonização.
After the presentation, we just heard of the World Health Organization presenter who talked about harmonization and to avoid duplicity of work that those organizations regulators have. And I will overview this during my presentation. I want to explain you about my program, which is an auditing program that allows us to have a high quality auditing for the regulatory requirements and support of different authorities to avoid duplicity and to accelerate their actions. This program is specifically for auditing of quality of the makers and not to authorize medical devices, but to approve this model so that we can have a unified.
Como que o sistema funciona? Então, as autoridades
And so how does it work? We have the regulating authorities participating Australia, Canada, Japan, and the United States. These authorities assess, authorize, and recognize auditing organizations. We currently have 15 of them, 15 organizations that are recognized to operate under this program. The auditing program has audited more than 5,000 makers in the world, and there are more than 7,000 participating in this program. And the auditing organizations have done this work. The reports are posted, but are confidential, and it can only be released by the auditing organizations, and it can be available for the new members with bidding processes. And with this data, the regulating agencies are making decisions on them, on the manufacturers of medical devices, in addition to medical equipment, materials for medical use, and devices for in vitro diagnosis. Some of the goals, the main goals here are to run a unified audit program that ensures reliable outcomes at auditing from manufacturers, to reduce duplicity. What's really important is that the goal of this program, and that's a really good advantage. The goal is to reduce the problems that exist between manufacturers. We know auditing creates a lot of anxiety and issues to manufacturers. So imagine a manufacturer receiving 5 auditings of different authorities to receive just one authorization. So the reduction of these processes makes things easier. Imagine every country having to audit manufacturers. So with this group, we have basically a group of authorities that has a sole auditing instead of having 8 audit processes in a year. This process seeks to accelerate this and avoid any issue uh, in the industry. It also wants to improve this auditing through collaboration and mutual acceptance, respecting each authority's autonomy, and to foster global alignment of regulatory approaches and technical standards following best practices. In this slide, you can see the timeline of MDSAP since 2012, started in Japan with participants such as Australia, Brazil, Canada, and United States. From 2014 to 2016, the pilot programs were created. In 2015, Japan became part of the fifth regulatory authority. In 2018, we did a proof of concept. We formalized these uh, proofs, the programs of the pilot, proofs of the pilot programs, and the program started working effectively. And in 2019, there was a significant milestone, which was the end of transition of the Canadian systems. And since 2019 onwards, Canada made the program mandatory. Currently, any kind of manufacturer of medical devices has to use this program to get to Canada. In 2020, the program of affiliate membership was created, and many countries started participating, other regulating authorities as well, from all over the world. And we have seen a lot of interest in knowing and participating of this program. As I said, we have the official members of the MDSAP program. We have the TGA, Australia Therapeutic Goods Administration. We have Brazil Health Surveillance National Agency, the Health Canada participating, and Food and Drug Administration. We also have other members as official observers, such as the World Health Organization, the European Union, the Medicines and Healthcare Regulatory Agency, and Singapore's Health Science Authority. Those are the 4 official observers of the program. We also have affiliate members that entered after we authorized that. We have the COFEPRIS of Mexico, the Federal Commission for Protection from Sanitary Risks. We also have Korea's Ministry of Food and Drug Safety. And we have the AMAT, which is the regulatory agency of Argentina, another affiliate member. We have Kenya, the FDA of Taiwan Food and Drug Administration, and from South Africa, Health Products Regulatory Authority. So how these auditing organizations are evaluated and recognized under MDSAP? How do these 5 member states do it, and how are these assessed? Well, we have some specific criterion to authorize the auditors, to recognize them, to receive the authorization. And we have several documents that you can see on screen, such as ISO 13485. We also have IMDRF, in addition to the MDCAA, MDSAP. We've made several other reports, and here we have the basic requirements for the assessment of the accreditation of these auditing organizations. This is done annually, every 4 years. This happens annually. We do this assessment. The regulating authorities, depending on the members, Brazil, Japan, change, and they go to these auditing authorities. This is a block assessment of all of the agents that guarantee the quality, so that they see how's the administration of the auditing criteria, the external resources, improvements, analysis, measurements. We also verify competence management, the auditing's being unbiased, and whether that's workers from the regulatory organizations or subcontractors to work on the regulations, the decisions, the process of auditing, as well as management of information and communications with the regulatory agencies, the communication of adverse events, all of those aspects from the auditing companies are assessed annually by the regulatory agencies. In addition to this, annually we have auditings that are observed where the regulatory authorities can go and visit the places to evaluate the performance of the auditing organizations in facilities where they sell medical devices or health devices. And they do monthly visits, and from those visits, they choose a manufacturer, and it depends on the need of each country. For example, in Brazil, they can make a decision of visiting a specific manufacturer if there is a complaint, if there is any adverse effect, and they go and visit the manufacturer so that the auditing mechanism can provide for a new opportunity for this manufacturer. So we have these audits for the manufacturers for any of these cases, and it will depend on the country. Every year, the auditing organizations have an auditing on site, and they are assessed on the auditing the manufacturers for medical devices for health, and the auditing authorities are there just observing. They don't intervene in the process. They do their work normally, and there is a report of the assessment formally. We provide the organization that to the organizations, the authorities, and there is an assessment with different observations and a working collaboration. This is a working collaboration so that later the authorities can decide whether they want to continue or not working with these authorities and regulatory organizations. Here we have a little bit more information about the assessment criteria, such as ISO 17021, that talks about the requirements for bodies providing audit and certification of management systems.
We have another document that is complementary regarding the requisites, requirements for the medical device auditing organizations, and the last document, which is a specific one for the competence and training requirements for auditing organizations that each auditing organization has to have, and they have to reapply with each of their auditors. So we have seen the criteria for the recognition of the auditing organisms and the criteria they have to fulfill so that the regulatory authorities can accept them as capable of carrying out this MDSAP audit. So, what are the requirements for a verified MDSAP audit? What do they have to fulfill or comply with it to be able to receive this MDSAP audit criteria and in the program? The requirements of an MDSAP audit are basically the ISO 13485 and also the specific requirements of each country: Australia, Brazil, Canada, Japan, and the United States. So these five authorities developed and prepared a document called MDSAP Audit Approach, which details all of the requirements of the ISO 13485 separated by task in a detailed way of what needs to be verified, and also giving explanation for each requirement and when the specific requirement of one country does not comply with the ISO 13485. For example, Brazil has specific devices for the purchase of raw material or components. These are not covered completely in the ISO 13485, so therefore it is specific to Brazil. Entonces, to be able to buy raw material, you have the requirements of the ISO plus the requirements of Brazil. When there is an audit to a manufacturer, and if it is from the Brazil, you have the requirements of Brazil as well in that specific occasion. But they have to, mostly they cover the five different jurisdictions, and then they can go into the five different markets. So this is in general. We have here MDSAP has the good practices of manufacturing in the countries and the specific requirements. We have the management of quality, the measurement, analysis, and improvement, which is in charge of all the complaints and anything happening internally. You also have design and development, production, and service provision. So these are authorizations of the product for the market, in particular, considering the good practices of manufacturing. So we already saw how the regulatory authorities recognize their auditing organisms. We also saw the criteria that is used by the auditing organisms for the medical devices, and they have to fulfill to be able to receive approval. And now I am going to mention the use of the functions of this for the manufacturers, which is very interesting because any manufacturer can hire an auditor to be able to do the unique audit for single audit program and to be able to have access to the markets of the five member countries with a single audit program. And this will be given, it'll meet the requirements of all participating regulatory authorities. And also, it can be used by the regulatory authorities. Each country defines how to use the MDSAP outcomes according to their local jurisdiction in accordance with their legislation. For example, in Canada, since 2019, it is the only way to be able to reach this market is through the MDSAP program. In Brazil, MDSAP is optional. The manufacturers of medical devices can decide whether if they want to go through through the traditional way of authorization, or they can go through MDSAP as long as they have products type 3 or 4. And to be able to start production in Brazil, the manufacturers have to register their products. All products of class 3 and 4 receive individual authorization for their commercialization in Brazil. And with this kind of product, you can regulate in Brazil only if the manufacturer is with the certification of good practices of manufacturing, which is given by Anvisa. Now, the manufacturers in Brazil can use the path that they have through the Land Visa or the MDSAP program. So there is protocol where they can be informed. They can inform Alamdisa where they can say that they have already been audited through MDSAP and they give a different code to Alamdisa where they have a recognition from the code indicating that they are a part of the MDSAP program and they have access to the repository of documents of MDSAP and they have access to all of the documents and all of the audits regarding the audits that the manufacturer received or the documents of conformity, considering the data and considering that Landisa decides if they certify or not without an extra audit or additional audit. So the members that are affiliate have this use of this audit. They have their regulatory organizations, but regarding the data and reports that they receive, they can make decisions based on the audits that have been carried out. Now, MDASC MDSAP promotes global regulatory harmonization, increasing efficiency and quality in managing medical devices while building mutual trust and easing the regulatory workload. Taking into consideration what the previous participant said, this is oriented towards medical device or and other devices as well. But in MDSAP, it is specific for the audit and quality management in manufacturing. And we have more than 7,000 manufacturers in the program, as I mentioned, and there is a strong reduction of workload in the national agencies, regulatory agencies. Andisa normally receives many requests to carry out audits throughout the world. It is a lot of work, and many times, considering the amount of manufacturers that there are in the world and that appear daily, this is almost an impossible task. But here we have mutual trust, and it is a path that regulatory authorities should follow to be able to ease the regulatory workload. That is what I had to present today. I apologize if I went too fast. I don't know if I have any time left, but I am available in case there might be any questions. We would like to thank the presentation. And now we have space for one or two questions, but very quick. If anybody has a question for Dr. Barbosa regarding this single audit program for medical devices. It seems that everything is very clear, Marcos, but we would like to thank you because what you are explaining as experience of Andisa demonstrates that the implementation of this regulatory reliance in this process somehow recognizes this certification of manufacturers between these different countries, somehow collaborates with efficiency of a regulatory agency as Andisa. So thank you very much for your presentation. And now that everything is clear, we give you another round of applause. Our next conference, which is also through streaming, is called Nightcrawler, a collaboration between Sysmetic and the public health system in Chile in the search for medical devices and medical substances that are illegal in online commerce by Nicolas Perez from Swissmedic. Dr. Perez works as a specialist and data scientist in the regulatory agency of medical substances and devices in Switzerland, Swissmedic. After obtaining his doctorate in the University of Johns Hopkins in the United States, he worked in the University Hospital of Zurich, developing multimodal models for radiology, algorithms for optimization of radiology and architectures based on transformers to be able to process images in rheumatology. His focus is in innovation and the development of applications to optimize processes in Swissmedic. Currently, his work is focused on optimization and monitoring of the market, identification of medications in images, search motors of information, and tools based on artificial intelligence that is generative. So now I will leave with you Dr. Nicolas Perez. Please go ahead.
Muchas gracias. ¿Me escuchan bien?
Can you hear me? Yes. Waiting for you to share your screen and presentation. We can see the screen, Nicolas. Go ahead. All right, wonderful. Yes, my name is Nicolas Loeffler Perez, ever since I got married, because in Switzerland you can have both last names. Let me share with you a project that started as an idea 4 years ago to collect illegal medical devices and substances online from the Department of Market Oversight in Swissmedic. And how this idea 4 years ago started to turn into a more solid project that led us to be where we are today, which is an area that exists today in Swissmedic. It's something that is used on a daily basis by our inspectors. This is a tool that is used and has become a standard, and how we have tried to share this with other agencies, and how on the pathway we have had this, and how this has become on a daily lives. And this is something that makes me feel very happy. Let me cover a few aspects of the presentation, and not explicitly, but I expect to communicate with you these 5 aspects. Who am I? What's my role within my team? What's Nightcrawler? What's this application, and what does it do? Where are we, and what's the state of this application in Swissmedic, and how we are collaborating with the Public Health Institute, and how we work on this collaboration. For those who don't know Swissmedic, you can see a photo to the photo to the right. This is where I work here in Bern, and this is a national authorization authority for surveillance of medication and medical products from Switzerland. It has a mandate with several tasks, but in the heart of it, Swissmedic guarantees medical products of high quality that are safe and efficient. That's the mandate of Swissmedic. Among these tasks, which are very explicit within the law, we have the authorization of medication, the surveillance of the market of medication and medical devices, clinical trials, and lab tests on the quality of medication. Just to name a few, Swissmedic has about 100 people. It's a very, let's say, company if you compare it at global level. So that is why we're always taking care of the quality of the work and always to improve it. How can we optimize resources? And within this context is that Nightcrawler came to be. The question is very simple. What happens when a user, a consumer in Switzerland finds or searches for an online product? They search for this online product, they believe the website is trustworthy, they buy the product, and then maybe they realize that the website isn't it isn't trustworthy, and the product that was purchased is slander, or it was false, it's fraud. And this is something that happens often. It's a conversation that always takes place in the UN, the WHO. They want to quantify how big the problem is, and it is a very significant problem. So what happens when that happens? When these types of situations occur? Uh, the person who searched for these products can let the our organization know privately with their name. They can communicate this to their main health provider, their doctors, and there are other pathways in which they can share this information with Swissmedic. But what Swissmedic wants to know is basically, where did you purchase this? Who uh, sold this to you? Swissmedic can take several actions on this. One of them is investigating. Hopefully, this would lead us to the person who made the sale, but specifically to understand what's the point sale. Nowadays, it's getting easier to have these point sales, which is why Swissmedic wants to eliminate these websites. Ideally, as an example, for example, on Monday we found a website where they are selling some kind of forged medication, and during the week we can already take actions to remove it, and by the end of that week leaving that product out of the market. It's not always available as such, and it's important that we can communicate with the websites that are providing the service for clients to buy directly from those websites. That is what we are trying to solve. And the main engine for someone like me who works as data sciences and Swissmedic is trust on the inspectors. The trust that the inspectors have. If they take this tool that we are applying, it started 4 years ago, it's a small prototype, but it has become and turned into a very solid tool. The main angel is for inspector of Swissmedic. Whenever they are using the tool, they can say, this is a tool that works well, creates trust, creates good results, I have the opportunity of reviewing very old results and compare them with current results. I have access to database. They look at these types of things, and everything is basically translated into trust. This website, as I mentioned, and we've had several development phases, four in total during 4 years, and that has allowed us to improve based on the feedback from the users. We have collected millions of products, and the last time I checked the database, we had about 3 million products. Thousands of those are reviewed by hand and removed from online presence. And last year in 2024, with the team that I work with, we were able and were very interested to collaborate with other agencies. So we started sharing the code with which we identify these online products. Among those is that we started a collaboration with the ISP. I have talked a lot about my team, and so far, and I haven't been working at one of the departments or specialized departments in a specific topic in Swissmedic. I work with a team that's called Swissmedic 4.0, and this is a team that has 7 people in it. 7 people who work in innovation, developing new technologies that can help Swissmedic not only doing the job right, but it, to think how can Swissmedic do the work in 10, 15 years in a better way, which is why our efforts are directed into machine learning. And for those who know the regulatory labor, the main resource that we have is knowledge, documents, historical documents, documents from companies, documents from consumers, complaints, reports. We have a huge amount of information, and a laboratory of information such as Swissmedic 4.0 can gather and provide all of these the data and provide insights and a better knowledge for the inspectors so that they can do their job in a better way and so that they are more efficient. Nightcrawler was born in that context. How can we think about the future? So, very specifically, what's Nightcrawler? What does it mean to review the online market? Nightcrawler is an application that works on a browser of the computers of the inspectors, and the goal is to find illegal products online. That's the main task. And what Nightcrawler does is assess if these products are suspicious of illegal activity, but it's finally the expert, the one that can determine if this is infringing the law. And as a disclaimer that included in the slide is that the decision is always on the hands of the inspectors. And as parentheses, because when we talk about AI and using it in regulatory tasks or in hospitals or in places that are a little bit more, or let's say where things have to be very clear or understand right how everything works, it's always a little complicated. And Nightcrawler has been a really good experience because it's allowed uh, the work of the surveillance inspectors to work closely to this um, platform, and that increases the capacity of these inspectors to work. It improves their work faster, um, more efficiently, but it does not replace them, and it doesn't replace the capacity that they have of seeing what they used to in the past. What we were doing when we were creating this, when we would do epi testing, as we call it in computer science. So what we would do is using the tool in one context, and we would do a very similar experiment without the tool. And with the tool, we realized that we were able to collect much more than what an inspector can collect on their own, and makes it more reproducible. And the idea of the tool in this case is never to replace the inspector. The inspector is the main driver making the decisions, and what the tool does is providing information about online market in a much more efficient way, in a more processed. This is how it looks like. You can see the slide on to the left of the slide. This is a screenshot of the app and how it looks. And to the right, you can see an example of how a product is sold in an online platform. And the idea for this specialist would be to see if that pharmacy is real and whether that pharmacy is a serious pharmacy. Is it approved by Swissmedic, or is this pharmacy a marketplace, just an online commerce that was created a few days ago by an actor with bad intentions. To the left, you can see that there are categories. When you do a search, such as compound or sildenafil, active substance, that substance will be used as a concept, and we are going to gather 500 links to different websites. All of those links are part of that first category that's called unprocessed. What an inspector at Swissmedic has to do is process each one of them and say, I am interested in this and I am going to mark it as bookmark. Or maybe I'm not interested in this product. And there is one category in particular that I really like, which is never relevant. Why do I like this category? Never relevant. I like this category because I can use it to improve my models that are making this decision. When an inspector marks a never relevant product, what I can obtain from that is that I can make an update to the model. The model can make better decisions. Our models today are trained with over 60,000 labels, all handcrafted. But hopefully, in the future, we can scale this a little bit more. And how to do this? We can do it with the labels, with this what the inspectors are marking. You can see individual products, and it is very easy to explain. There aren't very many possibilities of exploring the results, a lot of analytics options. What we're focusing on here is improve the process of those who are working in surveillance. What is possible to do with Nightcrawler, and how can this help the ISP? How did we reach this collaboration? First thing was, because it is an interesting proposal, the idea of having a database with data that would where we can obtain more information from websites that are probably selling something illegal, where we can collect 10 times more links that a person can review on their own time in their office. Is very attractive, and it means that we can do something much more automated. This isn't something working with words. This also works with photos. We do an inverse search, and with pictures, we can also find actors that are selling a product with that same picture, which makes it very attractive. The website that we have works specifically for inspectors. This was created specifically for government, and this means that it brings value, and it is differentiator agent. And what is it something that I would like to do for my team so that we can improve these relationships? And something that we did to Swissmedic, and hopefully this works for other countries. We work with others, the counterpart from Austria, and we work with them, and we wanted to do this work in a more generalized way so that it can be something that's useful for Swisscare, but also for ISP. And we can integrate also this with social media, among other things. And there were things that we decided to discard, such as the testing of the products, mysterious purchase, URS, automated elimination. We believe that's today outside of the scope and interest of the inspectors. So how can ISP access a technology such as this one? And how do we make this collaboration possible? So you have the users, we have users, each user in their own universe. And I say universe because every organization such as Swissmedic or the ISP has their own technologies. And Swissmedic created this application within the cloud, which means that it's somehow easy to integrate other users. And we were in charge of making a design that's direct so that this integration is as easy as it can be. That's point number one, right? The application in the cloud. The second point is related to the code. Swissmedic is owner of the code. We developed this within Swissmedic, and they are the owners of the code, which has an advantage. What do we do with the code? That's what we decide. And the third aspect, which is the most important one, is that the analysis of the processing AI, etc. We did all of this with an open source, which means that that part of the code is available on the internet. It's a repository in a platform called GitHub, and we made it accessible for decision making. So even if the ISP says, no, we don't want to collaborate with Swissmedic, the code is already out there. So any organization in any country could use the code and try to implement it. In collaboration, this is much easier because a developer like me or data scientist like me can help you understand this much easier. But what's important and the idea behind it is that we want to share. And from the government perspective, that is very important. So what we do as a challenge, or what our challenge today is adapting the code. And everything that we found from Nightcrawler, for example, the 1,000 suspicion websites, we can extract data of 900 URLs, and 500 URLs are relevant to the country, and maybe 400 URLs will be relevant according to our machine learning models. That's for Switzerland. Now, if we want to do a collaboration with other countries, we have to test. And we have done a lot of tests, and I included that CH because that's the name of Switzerland. But we have made a lot of tests in Switzerland and with other collaborators. And as I'm mentioning here to the right, for every one of these steps, you need to test, test, test. You do this with experts, experts from the surveillance teams. Each model requires adaptation to the local context. In short, so what we do is collecting all of these products that were labeled, and we train the model. We do this online on our computers. There's a complex way of doing it, but we're going to say that this is just we do it on the computer, and we check if that's relevant or not. We train that model. It usually takes a couple of hours, and now it's ready to make decisions on its own. You can see the typical process. So a product is fine. It's found online. We apply filters, the relevance for the country, we give it a score, and then we classify it through a AI system model that is later reviewed by an expert manually. And all of the data that is gathered is used to improve the model by having this virtuous cycle where the model can continue to learn for the future. So how are we going, and how do we measure the success? And how it works is collaboration between two agencies. And I've had the pleasure of working with a lot of experts from the ISP. We have learned that this is challenging, the collaboration between different parties. It's difficult, but it has been very beautiful to do so in a pragmatic manner. And today we already have a version where the heart of this application, the heart that's used in this AI model to make decisions, is set for the ISP.
We are in process of testing, and everybody who works in surveillance is going to have the final decision of saying if this improves their work and how Swiss Mega has done it. But for me, as data scientist, it has been very enriching to be able to experiment with colleagues and to see how the system is relatively flexible, that we can adapt it for other contexts. And in a very concrete way, what we have done is adapt one of the filters. This is an example. Basically, the products enter here. You have the processing, data processing. There are some scores that are given. There is a model that has specifications. And this model, and I don't want to go into too many detail because it's not necessary to go to the technical part, but that model is trained with labels that are handmade. They are used with the text that comes in as an input, and then you train the model, which is a very standard model in random forest model. And for that, we use technology that is free by Google, which is called BERT, and where the words within a website are transformed into a number representation. And that number representation, we then learn to see what are the suspicious ones. This adaptation, this model, is basically the most difficult part to adapt for the context of ISP in Chile. And it has been a challenge, but at the same time, it is very enriching to see how this works in different contexts for Swissmedic. How has the history of Nightcrawler in what is the future for Nightcrawler? And this is my last slide. We started 4 years ago with a prototype. We carried out something that was very basic and we liberated it to our colleagues. They gave us feedback. We decided to separate different models and different functions. We did another iteration. We developed the web page, the next iteration, and last year we started with Nightcrawler, which basically allows international collaboration. Today, the code is open source. It is open. Anybody can use it, download it, and make it work. I think that it is something that is, as someone that works in the government, I love being able to contribute and have the contribution of the work of others. And I hope that the implementation that we have carried out in this collaboration with ISP can have a future for long term. If you have any questions, if anybody wants to contact me, or if you have any ideas, please feel free to send me an email or contact me through LinkedIn. Thank you very much for your time and for listening to me. Thank you very much, Nicolas, for your excellent presentation. We are now going to give space in case there might be any question before we have the break coming up. Jacqueline, please. Good morning. Jacqueline Castro, Scientific Association of the Medical Devices. One question, this application, is it only for government level? And when you do find illegal products, do you communicate that to the legal manufacturers? And could a company have use of this application to search falsified products themselves? Thank you for your question. This is a very interesting question. Unfortunately, this is not within Swissmedic mandate. I would love for it to be, because I think the industry and the government, the better they coexist regarding how this technology works and how they can benefit it can be a benefit for the population in general. It would be something very good to be able to observe. It is not within Swissmedic's mandate, but so what we do for us is to develop this application so that it can give value to the workers of the surveillance department and to collaborators and others governments. We have also worked with people from the Austrian government as well as the public health department. So this is still within our mandate. We have not optimized it for private search. However, the code is open source. Therefore, if anybody was interested, they could have access to the code and adapt it to their own needs. We have another question. Thank you very much. First of all, I would like to congratulate you for this excellent initiative and for the possibility of collaboration with the ISP. I am from ProSalud Chile. And my question is, since many of these sites can be located outside of the country where the agency is, extraterritorial, basically, how do you tackle that issue? And I think cooperation with other countries must be key in this point. Precisely. Yes, thank you. This is a fantastic question. Precisely. That is one of the reasons why collaboration is so important for us. In Switzerland, we have the capability of establishing contact with vendors who have websites .ch or .ch, or those who are registered within the controller of the domains of Switzerland, which is separate. We have contact with them and we have an excellent relationship. But when one of these products is located online, the first step is to contact that website and ask them to remove that product. It's like a takedown notice. And most of them respond well. Since Nightcrawler has an automated process, we have discovered also that there are websites that have nothing to do with e-commerce, but that have been hacked, and they do have sales points within the website. It has been very beneficial. Therefore, our inspectors say they find many things that before they would not have found. But what happens when we get to a website in Germany, for example, or France or Austria? Swissmedic does not have the power to take those websites down. We cannot pull them down. It's unfortunate, but many of those websites can deliver in Switzerland. So it makes it very challenging. If we could collaborate and all use the same platform, we would definitely be able to have a effective way of making sure we can bring down more of these websites. Webcrawler has the motivation that what if we could harmonize all of this? What if we could use the same technology or the same platform even? Maybe we could start to help each other across jurisdictions. We can help our colleagues from Switzerland in surveillance in Germany, for example, and vice versa. And I love the question because precisely this is one of the things that has motivated us to be able to talk about how do we standardize this problem. Thank you very much, Nicolas. The truth is that personally, as a team of the Department of Medical Devices, we would like to thank you for your excellent availability to start off this development together with the Public Health Institute. And even when regulation between Switzerland and Chile at this time presents clear differences, there has always been great availability from your team to incorporate surveillance through this tool that Swissmedic presents to the medical devices. So thank you very much, Nicolas, and we hope that in your next visit to Chile, you can come and greet us here. To have professionals like you with this innovation that support our regulatory work of our colleagues within the organization is something that we require as well. So thank you very much, Nicolas. So to continue, we are going to carry out a short recess. We would like to invite you to visit the posters that are in the entrance of the tent and to share a coffee with us. We will be back here at 11. Continuing with our program, now we are going to continue with the Conference of Development and Innovation in Chile, Corfo Financing Programs, with Claudia Saavedra from the Production Development Corporation, Corfo. She is a commercial engineer and master's degree in innovation and technology management with more than 20 years of experiences with public programs related to the development and innovation. She has specialized leading public-private initiatives that want to install transformation processes oriented to the diversification and sophistication of our productive matrix. Currently, from Technological Capabilities Management of Corfo, she coordinates the program Transform Innovation in Health program that wants to push and accelerate the processes of innovation in the health sector through a collaborative work between the public, private sector, and the academia. So now I leave with you, Claudia Saavedra. Good morning. Thank you very much for the presentation and thank you for the invitation and for the invitation of Corfo to this. I am very impressed to see everything that is happening here and these 3 days full of activities. So I am going to share with you a little bit of what we do as Corfo and also with a focus more on health and what things we are financing and those things. Corfo is the national agency that supports productive development, innovation. We depend on the economy, Ministry of Economy, so everything that we do is related to supporting productive development in the country. We support in different ways to generate these highly innovative ecosystems and to promote different businesses through different areas of work. Entrepreneurship, we have more than 50 programs and instruments going around, which sometimes doesn't make it easy to understand, but I am going to focus on those areas in which we have received the highest amount of projects having to do with health. And you can see here that we have 5 large working areas. We have entrepreneurship, strengthening entrepreneurs, where we help the businesses. We also have investment that we do through the bank as a second floor platform. That is the way we work. And we also have an area to strengthen technological capabilities in the nation throughout different sectors, which is the area that I work in. And we also have support for the different regions, especially having to do with the development of territories. I'm going to go a little bit deeper in these 3 areas and share with you what we do. We have an area that has to do with the entrepreneurship where we support startups, researchers, or those who are developing ideas in their initial stages. In other words, before they become a company and there is an idea that they want to develop or they want a prototype, we have these financing lines that we call startup financing. And we support all the different phases of development, from the development of a pilot program or a prototype up to the point to where they can scale up in a business. In terms of budget for support of resources, we have this line of work that has the support between 15 to 75 million pesos with projects that can last between 1 to 2 years. This is a first line, and here also thinking that there is also a talent attraction. We have the program of Startup Chile. I'm sure many of you have at least heard of it. That is also on this line that wants to that looks for businesses with possibility to be able to scale at a global rate, but also has the same structure of support in its initial stages all the way till it can go to scale. We also support the ecosystem of startups in Chile through some external structures from Corfo, but that as Corfo we can help support so that they can work, which is business incubators. There is a mentor network for support as well. and national and international accelerators that we have agreements with as Corfo that you can also have access to if you are developing some kind of an idea. That is the area more having to do with startups, right? Initiatives that may not have started a full business yet, it's just beginning. Then we also have the area of innovation, where here what we offer is support for companies that have at least 18 months of being initiated. And we have a support program that is also scaled depending on what stage the solution is in. We start off with uh, Sumate a Innovar, which is a tool for the initial phases. And here we're thinking of innovations that have to do with borders or that are more disruptive. Then we have a work line for development. And there we have two different programs. One is Crea y Valida, and the other one is Innova Alta Tecnología. And then we have the Consolidate and Expand, which is in the different, so they can go to market and scale. We also have some international agreements, especially to be able to work on the search for some allies that can develop technological centers, for example, in Europe with the Eureka network. And we also have some specific to be able to find the connections. In resources, we go from 150 million for a project, especially Sumate a Innovar, to 1,000 to 1 billion that are more challenging projects and have more technological needs. And there we have high technology, and we have brought that up once again. They had been closed, but we opened them up again these last 2 years with high success rates. And there we have projects where we have up to $1 billion maximum. And to complement for the supporting companies, we have innovation with two instruments that are highly wanted. One of them is human capital. We support hiring of human capital that is highly specialized for 1 year in a company. You can also um, ask for these funds, apply for these funds. And then there's another one that is technological absorption, where we support groups of companies who have a technological challenge and they want to adopt. This is a program that can last up to 2 years, and this group of companies can search where this technology is. They can carry out an adoption of this technology, hire experts, etc., and which is also something that is highly requested by different industries within the country. And the last line of work is technological capabilities, and this is where what we do is facilitate themes like technological infrastructure, development of human capital that is more advanced, and also some knowledge in a collaborative way. And here we have everything that has to do with the packages of technological centers. We have many nationally that we support, and also the consortiums, technological consortiums and programs that are in this line of work. And we also work and support for goods in certain sectors. And as you can see, we cover most of them, the most important sectors of our country, where health is one of them.
Some figures focused on health, on data innovation, business support. We have financed more than 35 projects, more than 23 projects, with 14,000 millions in project, and a lot of projects on the health sector. And they are the projects that are applying for the research and development law. And we manage all of those. As you can see, the bars cover several areas of medicine, health, biotechnology, development of medical devices, biosafety, genome, advanced therapy. There's a broad diversity of the health sector, and these are part of the projects that we have financed. This is a summary of a few companies that we've worked with and the main tools that we use for this type of development, which is high technology, create and validate. And we also have two technological consortiums on the medicine. We have been supporting for more than 10 years already, and that has some really interesting results. And it will have a new 10-year period of work with us. And they are also these are also part of the ecosystem. And the good news is that not so long ago we did a collection of the projects that are financed with this high technology focus, and these are more at the border and from the different economic sectors. And we can see that the health sector accounts for a 32% of the projects, and the resources that we are allocating are health above mining. And that's really good news because in a way, this sector is a sector that works with trends of the newest technology. So it is also highly related to AI, as we just saw, some other aspects such as biotechnology and advanced manufacturing. And we'll see that with the presentation that we'll later see with Levita. That was supported by Corfo. Last year, in the framework of the context of what's happening in the health sector, and I think the fact that you're inviting us here in this very and highly specialized symposium is that we were invited to work in a project that's called Transforming and Innovation Health. It's a very strategic project, and we didn't have a project in the health sector that had a long-term view. We did a work of design to look with different stakeholders, pro health service providers, civil society, etc. And we looked at the productive development and how to create economic development in the country. And we looked at the health sector. We are a very classic country in terms of what we do. So we are focused on copper, on the export of fruits, the export of salmon, and this is something that's highly sophisticated that can contribute economically, and it's something that we're taking seriously, and which is why we have this 10-year agenda. And we are very happy about this because we see a lot of interest from many parties to make progress in this area. And we understand that in order to achieve and the development of a sector and to achieve the definition that we have, a definition from 2024, and we see that we have competitive advantages with the entire ecosystem that we have set up, and we can advance and be referenced in certain topics of health at the Latin American level, and to be better prepared to different scenarios such as what happened with the pandemic. So we need to be very prepared. These change health, change the models different, a new paradigm in health, and we need to be prepared, and we need to have this perspective on how innovation and technological development contributes. We identify gaps. This isn't easy because we know it's not just financing projects. Projects have to be used for innovation to happen, and that's a most complex part of it. And to contribute in the initial stages means investing a lot of resources. But how can we make these innovations a solution for the common citizens, and also from the economic point of view, for it to be something that is impactful? To do so, we cannot just look at it from the economy perspective, but also from the regulatory perspective. And the ISP has helped us. We have been working together to identify this vision and to understand that if we want to be a world-class country and be referenced, we need to work on the regulatory aspects. Innovation purchases. Today, we are installing a public purchase system that other countries have, and we are lacking a lot of that. And as you know, for this sector, public purchases are really important. It is the sector that's acquiring the technology, and we need to modernize. Innovation requires a new scale, and we need to take care of what we do And how are we taking charge of this as Corfo, as a system, the supply, demand, how to get it in the market, finding solutions, the lack of capacity of navigating the aspects of to be pilots, and we don't have that for the health sector to test the different kinds of technology. And also we need to advance into having a more articulated ecosystem. The countries that manage to take those leaps in innovation and development and that have a more sophisticated offer of production do it because they have collaboration networks that are quite tight, that are um, ongoing, and that's something that we can work on. It isn't spontaneous. So we defined the roadmap, and I have 10 seconds left. And we have 14 initiatives that we have prioritized, such as advancing having a brand as a country brand in the sector of health, and to work with a focus on also identifying the main challenges that we have in the health sector and trying to find innovation to have a match between supply and demand. And in that context, uh, new news. Something else that is included in this roadmap, we have a context of a new technological innovation. So technological project is how we call it in Corfu, but a model to work with the use of AI in the health industry and focus on the health industry. So we are doing dissemination of that information. If anybody is interested, our social media has that information also on the Corfu website. We have a search engine where you can find it, and please connect with our LinkedIn networks, with our social media, so that you know what's happening and so that we can continue to work together. Thank you. Thank you, Claudia, for your presentation. After the next presentations, we are going to ask you to be part of a roundtable where you can answer some questions. Now it's time for the lecture experience in innovation of medical devices presented by Dr. Alberto Rodriguez Navarro. He is executive director of Levita Magnetics, that has a which is located in Silicon Valley. Surgeon, minimally invasive member of the Association of Surgeons of the United States, former professor of the University of Chile, researcher with several international publications, and inventor with more than 65 patents. Levita developed a surgical platform that created a new category in robotic surgery, MARS, approved and authorized by the FDA of the United States, which offers several benefits: less incisions, less pain, and a faster recovery for patients, as well as a better visualization and precision for surgeons. Dr. Alberto Rodriguez Navarro, please come forward. Thank you very much for the invitation, being here. It is a privilege for me to be presenting in front of you. Thank you, Dr. C, for the invitation. All right. Let me tell you the story about Levita and what we have done. And this is the first time that I talk about this, addressing the regulatory aspects and how we worked with the FDA. I have some videos and things that I have never shared with anybody before. And the idea is for you to go home with three key concepts: innovation, of course, creativity, that's a core of what we do. But for the regulatory aspects, it's important because you need to know that you need to be creative whenever you're doing something new, and how to prove that technology is safe and that it works. So we need to be empathetic with regulatory entities, and we have to uh, put in their place, be in their place, and we have to work together. So I'll tell you how we did this with the FDA. We need to persevere. You need to have the energy to keep going until the end and work together with them, because these are slow process. So it's necessary to persevere. First, let me tell you what we do with our Mars robot. so that you know what we are doing. Then I'll tell you a little bit about the story, very brief. I speak fast, so please follow my presentation with the images I will be presenting. I will also show you a video later. This is a video that shows what we are doing and what does this Mars rover does. Valentina Munoz had an operation. I had a surgery 2 weeks ago. It was incredible. I never felt any pain. Came back home to do all of my activities. On Friday, I went to a wedding. Actually, I had no pain with the robot. It's just 3 incisions instead of laparoscopy that does 5 incisions. Incisions are very small. You can barely see them, and it's less painful, less invasive, and a lot of it's more aesthetic as well. This Levita platform, Bravo platform, allows us to optimize surgical times, optimization of surgical equipment, and that makes the work a lot more efficient. The implementation of this robot for our clinic and for Chile represents a huge progress for the surgical area. This robot allows to have better precision, um, shortens surgical times, therefore better recovery. We are the first private clinic that has this robot system. We have made more than 100 surgeries so far, specifically obesity, gastric sleeve, bypass, and this optimization of the work will allow us to do two things. It diminishes waiting lists for some surgeries that sometimes require 2 to 3 surgeons to perform. With the Mars platform of Levita, just one surgeon can do a surgery. I think this is very attractive for our patients as well as for our clinic, because today no other clinic can offer what we do. I felt like I was given this opportunity of accessing this technology. It was a privilege because this isn't something that they do at every clinic, robotic surgery. It's advanced technology. I would highly recommend this. Patients, surgeons sharing what they think about what this is. And basically, we reduce the amount of incisions during surgery. That's the clinical benefit. It's a robot with 2 arms. One controls the camera for you to see the surgery. The surgeon is on the side of the patient, and the magnetic aspect of it allows you to move the organ with less incisions. So you have a magnetic piece that is controlled with and it's a lot more efficient with less incisions, much more efficient. It uses a magnet. We have less incisions, less pain, less recovery time, less use of drugs, painkillers, less scarring. Everybody likes having less scarring. Surgeons are much more efficient. You don't have to depend on the assistant. It's a better tool to do the surgery. And for hospitals and clinics, it's a better experience for the patients and use of resources. So it provides benefits at all levels. This is an image for you to see our first surgeries where there's an assistant, and that's a picture of the surgical assistant that you need to operate. The idea is not them to rest, but they are looking at other patients and to use health resources, to use health personnel in the best possible way. This is a surgery. This is gastric sleeve surgery. The doctor's operating on his own, less time, patients bleed less. It takes less time to conclude a surgery, and it's a much more efficient process. Really good achievement for us is that this robot have been approved by the FDA. This robot is an accessory of our magnetic um, platform and robot, and I'll tell you how it went on how we received the approval from the FDA. And we were lucky enough to do the first surgeries on Cleveland Clinic, which is better than the Mayo Clinic in digestive surgery. We are working in the United States as well as in Chile. We are located in Dallas, Cleveland, also recently in Stanford. our robots was presented in the news, in the media in the United States. So patients from Chile and the United States are seeing the same benefits. This is something that was very exciting for me because we have the first Mars Center in the Luis Tisne Hospital. This was on December 2023. We've had more than 300 patients that have been operated in the Luis Tisne Hospital. We installed the second robot not so long ago, and we just had the experience of having two surgeons in the room, and now we have two robots working at the same time. We can double the surgical offer, which is the best way of working with the waiting list. This is Dr. Antonio Lassi. He came to Chile. He's a world reference. He invented the laparoscopy surgery. That's him. He's very excited. He has so much more experience than I have, and that's what we need. We need to always be looking for those new things and being love about this new technology. Regarding the development of Mars and the focus on the approval of the FDA, something that I never said before, first let me tell you what our roadmap was. We started by deciding this laparoscopy element or a tool that was manual, magnet and a claw, and the assistant would have to move the magnet. And what's important about this is that there's a change in paradigm. It's the first time that we use these magnets to move organs, and that was our first generation. With this product, we did about 6,000 surgeries, and with that, we were able to prove that it worked. After that, we moved on to the next stage where we included the robotic part of it, which is what we are currently doing, which is the second generation of what we are doing. And the third step is what's currently on development, and this is integrating robots, integrated AI, augmented reality, and sophisticating this process even more. And this is a process, so I want to tell you how it went. Now, after the great presentation that Claudia provided, we had this work done. We made this work, and as you can, we are a company from Corfo. Corfo is our strategic partner. That's how we consider it because they have supported us in every stage. And we started with this technology award, and we were able to develop the robot. After that, we received the support of Banco Estado, along with Corfo, and for the dev, which is for the commercial scaling, which is where we are at the moment. And in addition to that, we have the technology to continue to develop the next stage. I was born and raised in Santiago, and I've always liked to improve, and that's our engine at our company, and how we do things better, how we improve what we do, and to leave the place better than how we found it. I am a surgeon. I was formed with the J. Aguirre Hospital, and then I went to work at the Padre Hurtado Hospital. Some of you may know it. It's located in La Pintana. The concept is quite simple. Less incisions is better. One incision is better than two, than three, and then four. And the question is how to do a surgery with less incisions without committing the capacity of the surgeon to have a better and more efficient work. My dad is a mechanical engineer. Here's us drinking pisco sour, which is a trick to get new ideas. You just have to drink one though, because otherwise, bad luck. The idea is to be creative, but not really that much. And when we were thinking about how to do this with less scars, we remember that we had these water turtles when we were kids with my brother. And I'm not sure if you've seen these. You put the magnet and you clean the algae on the wall. It's the same concept with the abdominal wall, and you create movement in the cavity without affecting the wall. With that concept, we did the first prototype. It was very ugly, but it worked. It was functional. It helped us prove that it worked. We did this at the garage of my parents' house, and we did the first patent. Uh, and it's my dad and me. This is a Corfo program called to Go to Market, and they took me to Stanford Research Institute, which is like a cousin of Stanford, focused on innovation, and they trained people who weren't technical. I mean, people who had a technical background on innovation, or how to collect, gather resources, how doing they would do an assessment on you, and they tell me the feedback they give me is like, this looks good, but the probability of you doing this in Chile is minimum because you need a team of people that knows how to develop it and to have the ideas on how to turn that into in a medical product. And here you have two areas where you can do it, Silicon Valley, and that was the reason of why I went there. And Silicon Valley is because they not only develop software but also medical technology. And in that place you have the experts, the people who can develop it. And why Silicon Valley? Well, uh, those who are located there think about changing the world. They think big. There's a lot of networking. They are willing to introduce you to people that can help you, and meritocracy and teamwork. These are the first pictures of how the clamp started. We changed the product, the first renders. That was the first clinical clamp. After that, we received the first punch to our idea. In the beginning, we worked and talked to an FDA consultant. It's easy, you use these two things, and it wasn't that easy. The thing is, we need the novel pathway. This is for new technology, and even big companies are afraid of it because you have to convince the FDA that this is safe. How? You have to just do it. You have to convince them. And that was difficult for us because we didn't have the funds to do so, and we had to investigate, and everybody would say it's impossible because you're coming from Chile. How are you going to convince the FDA? It seems impossible. And that is why I talk about empathy, because when we said, okay, if we are from the FDA, how can we prove them that this is efficient and safe? And that's the first message I can share with you, to think you're from the FDA and think about what they want. How do you do a plan to prove that this is safe, and not just for the FDA, but to have a product that's safe for patients? And the first anecdote from FDA, they said, okay, what happens if this acts as a bullet that makes a hole on the wall of your abdomen? Because how can we convince them that this isn't like a wall, that this isn't going to damage your wall? What do we do? Do we send a magnet to them? It was very complex because if you wanted to go there, it would take like a year to get the authorization to go and show that to them. And this is what we came up uh, with. We have the clamp, and we did it from afar. Like that, we use the distance to show it to them, and you put it yourself in their place, and it makes sense that they have those questions and they were uncertain about that. So we did this video with a magnet in the hand, and we looked at it, and we showed that the clamp um, moves, but nothing happens with the hand. And this is a video that we showed the FDA, and we told them, hey, this is from the CEO. He's a surgeon, nothing's happening to him. And we convinced them that nothing would happen. And after that, we did the clinical trial in Chile with 50 patients. We designed the study to do it in Stanford, but we finally did it in the Salvador Hospital, Disney Hospital, and Padre Hurtado Hospital. And since these processes are long, very lengthy, it took us about 3 years, the first medical reviewer had some questions about the lack of strength of that traction and that you might not be able to see well. So we had to prove that this had the enough strength for you to see and do the surgery. They changed the medical reviewer. The problem, he said, is that too much strength that you might break the tissue, which is absolutely the opposite of what we had as a strategy. So we have to pivot there and convince the FDA. We had to change that and convince the FDA that there was this balance of strength. And we had several meetings with them, and we talked a lot with them, and they are willing to talk if you take time to do so and get their feedback.
So we had the FDA approval, which was a great accomplishment, and we have a new category, which is magnetic surgical instruments that exists now thanks to us. This is also published in the best surgical journal, which is Annals of Surgery. You can have technology and also have high impact. So we have publications in different parts of the United States. They showed us as the best technology of the year and the best, most innovative companies. And then we said, okay, we're ready. We passed the hardest part. We finished the problem, and that is where we have the commercial part. Because one thing is to get through FDA, and that is a good news, but the bad news is that now you have a product on the market and you have to prove and have people use it. And medical devices, you say that the dog it's dog food. We got to make sure it works. So Duke was our first customer, and all of the commercial area of finding the entrance into the market. We had the bariatric surgery started out, and so we started showing the benefits, and we started using this together with Da Vinci, which is the robot that exists commercially speaking. So we worked with Duke. We also did worked with Mayo Clinic, and then we started seeing the space that if we combined magnetic with robotic, it was something that did not exist, and it could bring benefit. So we started with ugly prototypes, as you can see on screen, but that they showed the concept. So then we did a first robot that was more stylish, that could move the magnet, and this was with high technology that we did this technology. So we had a robotic arm, and if we saw that if we could change the magnet, then we only have to hold the camera. And then we incorporated the other arm, and it controls the camera, and therefore we have a more advanced prototype from the robot that exists today. So what do we look at towards the future? We have many ideas. We are developing a camera, a magnetic camera. We have gone into the robotic part because it gives you multiple magnets in the different area that do not collide, and this allows you to have surgery in a less invasive way. We are developing robots to simulate the hand, the right hand better of the surgeon, and also we are carrying out development in augmented reality in the sense that the surgeon can interact with the instruments and they can have telesurgery and other things that we are working on. This is also a great, makes us very proud. We are the first ones to have augmented reality surgery. This also came out in the news in the United States, and this is the idea to be able to position Chile within the development of technology and medical technology around the world. I just wanted to show you a quick video of what we are carrying out, what we are doing, where you can see with a screen that is a virtual screen, you can see the surgery. You can see it much better. And that is the idea that in surgery, the visualization is everything. So if you can see well, then you can go quickly and you can be very secure. And that is, you can trust what you are doing, and it gives the surgeon more tools to do their work better. And with magnets, we hope that this can be less invasive and that the surgeon can see in a better way. And as we were saying, the best part of all of this is that everything that is software is being developed here with local developers. We are based in Silicon Valley, which is where we carry out the manufacture of the robot. However, half of the team is here in Chile, where we develop all of the software part. And with this, it also allows you to bring information in real time to the OR to be able to see all the information of the patient, and you can activate this with the sterile magnets on, which is the concept that we have to keep in mind in the OR. If it's a high risk patient, we have to be able to have all of their vital signs, and this develops technology so the surgeons can be better. We can also move the robot in a digital way, and if we are doing carrying out disruptive technology, and this is being carried out from Chile. So we're also doing artificial intelligence, but due to time, we're going to run through real quick, and to be able to finish the tips for innovation, we have to consider that startups are very fragile, and we have low resources, and we have to resolve sophisticated issues, so it's very important to be creative. And adaptation is the core of the game to see how you can quickly adapt. In this case, for example, to the FDA that was giving us different tests so that we had to be resolving, and we had to adapt quickly in such a way where we could be efficient. And we have to have clarity that there's quite a bit of uncertainty in the process, and you have to enjoy the uncertainty and know that that's the way it is. We know it's difficult, it's hard, and it is a part of the process. And you have to be very optimistic. You have to be very optimistic, the optimistic one of the team, because this is hard. People get tired, and as I said, this means that you have to be very persistent where the team is very important. I could not have done anything alone. I was very fortunate to be able to start off with my father and then set up a team that is very committed to be able to carry this out. And this is a picture from the Chile team. And here's Vivian Soto. Raise your hand. She is expert in clinical trials and the one that led the team here in Chile to be able to carry out everything that we have done. End to end, I don't know if anybody saw this movie. The movie was okay with Jean-Claude Van Damme, but when they would put the names that they wanted to the translations, that were very interesting. So the title is No Retreat, No Surrender, but the title is good as a that we never. The title is good that we have to persist and never give up. It's hard, but it's the only way that we can accomplish this. So to end, this is what we dream. What we dream is possible. And in Chile, there is a lot of talent and capabilities, and we have to organize a structure so that this talent can be expressed. So the three concepts, as I said, creativity, empathy, and with the regulatory organisms and perseverance that allows us to continue forward and make sure things get done. That is my email. There is my information if anybody wants to reach me. We would like to thank Dr. Rodriguez. He has another commitment at this time. So we are going to take advantage now. If you have any questions, he is not going to be able to accompany us later on, where we will have the rest of the speakers. So if anybody has any questions or comment, this is your opportunity after this excellent presentation. Innovation, Chilean innovation. So we would like to congratulate you, doctor, and then we will ask Dr. Janet Civias, head of the medical devices, to be able to give him an acknowledgement, and then we will give the word to Rosan, who has a question.
Rosana, por favor, brevemente la consulta.
Rosana, please, a brief question before we let him go.
Ahí le pueden pasar, por favor,
If you can pass him a microphone. Excellent presentation. How many surgeries have you carried out in the last years with the system? Good question. With the robot, we're close to 1,000 with the robot, and with the initial system, which was the manual way, we carried out around 6,000 surgeries. Now, I understand this product and something. Right now, they are operating somebody in Colonial Clinic as we are speaking. Oh, so this have a FONASA code, or is this still private? No, this is within what is paid for by the surgery. So that has been one of our challenges because many patients from FONASA with the pad bonus, one of the challenges for this technology, the idea is that this technology be accessible for the system so that it can be accessible to the highest amount of people. So here in Chile, we have only seven robots, and our idea is to make this robot accessible, and the commercial part is important, and that is our objective, that it can reach the highest amount of people, and considering that the cost be adequate, and that we bring high value to the people. Double congratulations. Thank you. Thank you very much. And now we will continue with the program. Now we have the Conference of Advance and Support Innovation with the professional Maria Graciela Rojas with medical devices of ISP. Maria Graciela, biochemist with many years of experience in regulation and medical devices, collaborated with the creation of this area in the ISP in 1997. And since then, Maria Graciela has been with different roles, like, for example, in charge of the trial of medical devices, evaluation and reviews of information that come with medical devices, and in vitro diagnostics. She also is in technovigilance or surveillance, and she has been in different courses of the ISP and contributed with a great part of the team that we have today. Currently, she is in charge of the area of establishment, medical devices, authorization of establishments. So, Gabriela Rojas. Maria Graciela Rojas. Hello, good afternoon. It is difficult to carry out this presentation after seeing so much technological progress and compare it with the regulation that we currently have of medical devices. But we are going to try to go forward in this sense and to help the innovators and manufacturers that are from here in Chile. This presentation has 3 sections. We will have general information of medical device. Then we are going to indicate the different support that we have for the national manufacturers, and then we have a proposal for now that is voluntary because we don't have a law that is supporting us to be able to go forward with the manufacturers and innovators nationally. So within some of the background information in general, as we have seen before, in the medical devices, the field is very large, diverse, competitive, and highly innovative, especially in the last few decades. The focus of the new technologies are placed in the nanotechnology and technology of information. Innovation in health grows very quickly, especially in the digital area. There is a lot of digital therapy and the adoption of artificial intelligence in sanitary or health attention. On the right, you can see there is a red circle, and we can see that innovation in medical technology is the second place of innovation after digital communication, and it is above other products of health, like, for example, medications, which is the green bar. I would like to share with you what a medical device is in general. They are products that are for health. They are instruments, equipment, in vitro reactive software, or articles that are destined by the manufacturer for prevention, diagnosis, treatment, rehabilitation, or and it does not use pharma, immune, um, metabolic means, or it is the risk that it is depending on the risk that it is for the patient or the health provider. We have 2 groups of medical devices, and we also have the reactive in vitro devices. The medical devices have 4 groups, being 1 the least risk, and 2, 3, and 4 the highest risk, and longer you use it. And then you have the reactive, being A the less risk, and C and D the highest risk, and usually risk is associated to the result of the test, the impact that the result has, whether it be on the individual or on the public health. And here we also have all of these reactives that are used in a blood bank, in scanning blood banks, and all of these other things. I would like to share with you what we are doing in our legislation at this time. The medical devices, the article of the health, the regular 825, number 98, in general, it establishes that the definition and risk, the requirement of a local certification when you want to incorporate a medical device to health control. We also have the faculty to authorize, and we have authorization of certifying bodies. We also can have technovigilance in the market. At this time is obligatory or mandatory for those who are under control. And also we can inspect, which is under the Seremi of Health. And this has basic requisites of all regulation of medical devices. For example, the sanitary registry of all of the medical devices and reactives that are commercialized within the country. We do not have the faculty to register companies or manufacturers or distributors of medical devices in ISP. We do not have the faculty to authorize manufacture facilities of importers or distributors. We do not have a nomenclature that is official as a country. And we do not have, which is something that is being used now, the recognition of decisions and evaluations of other health agencies to be considered in the approval of products in the country. Reliance.
It is necessary to update the legislation, and to do so we have two bills. First one is Fármacos 2, Drugs 2, which has been sitting in Congress since 2015 and has not made any progress. On January 31st, 2025, we presented the bill to modernize the national system of health services, which is an update to medical devices according to the World Health Organization recommendation. These recommendations include a gradual modernization of the regulation with a focus on risks and an acknowledgement of these international decisions in order to streamline our approvals. All of these requirements coming from the World WHO are in a book called World Model of Regulatory Framework of WHO, the second version, launched last year. It includes all of the basic, mid, and advanced requirements that ensure quality, performance, and safety of the products that are sold in a country. Since there are no basic requirements such as enrolling a factory voluntarily, we created an option which is called enrollment of manufacturing companies, importers, and distributors. The idea is for them to know who's manufacturing, distributing, and so know the medical devices that are being commercialized in the country. Today, we've had about 800 companies enrolled, and we have some national manufacturers, 101, and also importers and distributors, 696. The list of them is in the website of the Institute. And what are the national manufacturers manufacturing? In the beginning, they would say that they were manufacturing a lot of medical devices with very low risk, class 1, 2. But as time has gone by, we see we have a diversity of products. We even have different kinds of risks products. We have implants as well. We have different kinds of dressings, dialysis concentrates, immunomatologic, reactive, all of that that can be implemented. So as you can see, we have very simple devices as well as high-risk devices, such as diagnosis reactive and medical devices. Based on this information, we started to be concerned about how they are doing it, and also because they have requested technical meetings. Universities have requested it, entrepreneurs, as well as established manufacturers, and they ask, what are the guidelines that this health authority has regarding the options of manufacturing? Since we don't have that power, we said, okay, we need to start working on this. And last year, we had a day to support natural manufacturing and innovation. The goal, and 40 people attended, 40 companies attended, uh, companies that work on this area, on the design or the manufacturing. And the idea was to present the regulatory requirements that are requested internationally, because that's not applied yet here. In order to to approve a product, to approve the manufacturing and design of these medical devices. In that instance, several international reports were presented, such as the essential requirements for performance of that medical devices should have, the clinical research, risk management based on Regulation 971, and also a guide of good practices of fabrication, of manufacturing, that was based on the international norm ISO 13485, used globally by health agencies, and they approve these types of processes of design and creation. In addition to that event, we also create guides with international knowledge that has been harmonized, that we have homologated here. We have translated this as well, and has been discussed in the department, in the ISP, and then it has been posted. This is to support the manufacturers. They can use these guides. We have the essential principles for safety and performance, also for border products, and they don't know which regulation applies to that product, whether it's a pharmaceutical, cosmetic, or if it's a device. We also have the classification guide of risk. So what's the risk that the device manufacturing, what requirements, what's the evidence I have to prove that this is a safe product and it complies with the performance. We have a technical guide for devices at the borders. I already mentioned that, and then the guide based on the norm ISO that I just mentioned. So we are going to focus on the guide of the good practice for manufacturing. This is on the website, and this was also part of a public consultation. We received observation, and now we are at the stage of review and assessment of those observations. This is a guide that was made based on ISO 13455 485 from 2016, and we have the homologated norm, which is a national NSNCH ISO 13485 from 2017. This norm, this guide that addresses the entire life cycle of a medical device from the design and development, preclinical studies, manufacturing, regulatory approvals, and activities after it's presented in the market. All of this is based on the 1345485 of ISO, and same for the national one from 2017. This is a cycle that is repeated, which is it's also continuous. It goes from design, sales, post-market, surveillance, and if necessary to improve the project, they have to go back to design and development until it is a safe product that complies with this performance. Among the requirements of quality management systems, we have several aspects that are intersectional to any system to manage quality, such as the documentation requirements, responsibility of the directorate, management of resources, and we're going to focus on this occasion on the realization of the product. We're going to focus on design and development. This is something that innovators are interested in. And then we have also the production aspect of it, when it's ready to be commercialized. In design and development, there are several stages that need to be complied with and that are required internationally in order to have an approval of the product. You have the input of the design, results, review, verification, validation of the product. Once we have the product, we have the transfer to production, and we're going to focus on this. How do we start with a new product? First, we have a concept, an idea, a need in the market. And where are these ideas coming from? Academic institution, from health professionals, doctors, nurses, patients, or maybe from a manufacturer that does other kinds of devices and says, okay, I will work on this. So, and then the surveillance after commercialization. Engineers today are working on this, on technologies and on this area. Design and development is a key aspect, specifically for medical devices that are very innovative, and the 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 product that was presented before had to comply with all of these aspects in order to be approved by the FDA. So the input of design has three aspects that are really important. First, the requirement of performance, safety, and functioning. Then we have legal and regulation requirements. And then we have risk management plans. Among the first requirement related to performance, safety, and functioning, the manufacturer that is designing the product and that has the idea has to have a clear idea of what's the use, what are the specification of the performance. We need to specify all of the standards applicable to the medical device, foreseeable risks, clinical assessments, and if necessary to have clinical trials, pre-clinical trials, as long as that clinical assessment doesn't address those risks. Regarding the legal and regulation requirements, they need to know what are the requirements for the health um, registration. What are those requirements for the product that they will sell it and where they want it to be imported? They need to have a conformity assessment for the approval as well. It is also necessary for them to know if it's mandatory to notify adverse events at the national and international level, local labeling, as well as language that is going to be used in the instructions and labels. Another important aspect is the risk management plan that needs to be presented at this stage, where they need to identify the risks from the design and development, manufacturing, and use. Also, how to control or eliminate this risk, to determine residual risk, and also to communicate the risk. Once this is part of the development and design stage, they have a prototype of the product. With this prototype, the manufacturer need to have established procedure that all of these inputs were complied with. If not, they have to go back to them until they finally have a product the with all of the things that needs to have from that input of the design and development. It is necessary also after the results to know if it has an approval of the clinical trials and assessments of software, if that's the case. And this assessment needs to be completed before it's launched in the market. Lastly, there is a procedure of review of all of the final results to confirm that this is a safe product and it complies with all of the requirements. Once you have the product, the manufacturer needs to have a procedure to transfer the product and send it to commercial production. And of course, in any production, manufacturing, you need to have a control of the supplies, of the options of the manufacturing, identification, traceability. You also have some requirements for sterile medical devices, stability studies, the release of the product, the launching of this product, biological processes. Because, as an example, there are certain devices that are made with tissues, animal tissues, and it also needs to have the labeling and the packaging. Lastly, this is something that's very important for the manufacturer. They need to implement a program of surveillance in the market, an active surveillance as well as a passive surveillance. And it also depends on the medical devices that they are manufacturing and the risk, because the more risk, more surveillance from the manufacturer. Why? Because they need to manage the complaints I may have from the product. Second is that they need to be aware that there might be an event and how they notify that to the health authority, to do an audit in case there's non-conformity throughout the process, the non-conforming products that they're commercializing, and to withdraw the product from the market in that case, if necessary, and to also have a continuous improvement of the product. This is to highlight the regulations throughout the life cycle of a medical device. We have the norm of management, quality management, risk management throughout the life cycle of the device, and at the stage of design and development, where it's necessary to identify all of the regulations and standards that need to be applied when working on a medical device. And among them, we have the 9.3 norm of biological products or biocompatibility, clinical trials, labeling symbols, several international regulations that have been homologated to the Chilean norm. Production process includes some norms for the validation of the processes, and the health registry. It's important for the maker of the manufacturer to know where the product is going to be commercialized, what are the requirements, uh, what needs to comply with, if there is a conformity assessment necessary. And at the stage of feedback, it's important to gather all of the data from the product so that they can do a feedback of the risk management or also to have an improvement in the products. Lastly, and I think running out of time, we're going to present a proposal of uh, the offering of service, well, a voluntary offering of service of the manufacturers, and we are going to separate this in two different services, the same but separated depending on the stage they're in, whether it's design or development, and then we have manufacturing. And we have a stage that can be complied with in three stages. Mid, intermediate, and final stage. The first one for design and development includes all of the clinical trials, evidence that needs to be presented, all of the regulation requirements, design and development plan, foreseen use, performance. All of that evidence will be reviewed and we will check if that's complied with. And this is a proposal, not all of it is discussed here. And then we have the intermediate stage where we should have the management risk management report, performance assessment, and lastly, we have a historical file of development and design. This is an area that we're still discussing. It's a proposal, and for the ones manufacturing, this is what will happen at every stage, and it will be applied slowly. We have an initial, intermediate, and final stage that also ends with the technical archive. The results of this entire process and several different stages will include a certificate of compliance of the um, established requirements for every stage, and we will certify that they are complying with all of the requirements that are requested internationally, and that we are also going to apply nationally because we're going to modify the regulation of medical devices. And that's all I have for you today. I don't know if I had enough. I made it on time? Yes, I did. Thank you, Maria Graciela, for the presentation. Afterwards, we'll have space for questions and answers. Now we have the next program, radiodiagnosis and radiotherapy. Dr. Paola Caprile will present this from the Catholic University of Chile. Dr. Caprile is a physicist from the Catholic University of Chile and doctor in physics from the University of Heidelberg, Germany. She focused on medical physics. She is currently a teacher from the UC Physics and researcher from the Catholic University of Chile. Her current research is focused on dosimetry and the use of learning automated learning tools for the support of clinical decisions in diagnosis and radiotherapy. She specifically focused on personalized medicine, and she seeks to find predictive model to classify risk and response to therapy for cancer patients with the use of medical images. Dr. Paula Caprile, you have the floor.
Good afternoon, and thank you for the invitation. I'm going to speak very short and maybe different from what we have seen up till now, which are more concrete applications or guidelines. I would like to share with you the different applications that we have today of artificial intelligence in radiotherapy, especially, and also with diagnosis, and what implications this has in its clinical implementation. So, quickly, I'm going to start talking about the context, and then I will share what artificial intelligence is. We have heard in different conversations or lectures that we are going to use artificial intelligence, so we need to have clarity of what we are talking about when we talk about AI. And then I am going to talk about some applications that we have seen in radiotherapy where we use images that can be used in radio diagnostics as well. And we will finish talking about the perspectives of the challenges and opportunities that we can find. Today, we live in a world that is full of data. We consume data, we produce data, and artificial intelligence has allowed us to use this data to be able to resolve problems in concrete ways in different areas. We use it every day when we unblock our cell phone with facial recognition, or when we optimize the routes that we have to our jobs, to the place that we want to arrive. So the applications are very multiple and they are hidden and they're very practical. We can see it in our day to day. Beyond these applications that are practical applications, we also have some in the development of research. It is so important in research that last year, the two Nobel Prizes out of the six were given to artificial intelligence in physics due to the development of the foundations, and they presented the base for artificial intelligence that we use nowadays. And also, on the other hand, in chemistry, in the use of artificial intelligence to be able to predict the structure of the proteins. So it is something that is very relevant and relevant in health as well, where they also generate a lot of data, clinical data, patient data, and medical imaging. In particular, in cancer, it is evident, we can evidently see the artificial intelligence opportunities in therapy since we have in all of the diagnosis of cancer, I would dare say at some point we have to collect information of the patient as well as images. So it is a problem that today in Chile is the first cause of death, right? So the estimations say that this is a problem that is going to continue increasing. I center in radiotherapy because that is my research area, but also because it is the kind of therapy that is used around 50% of all treatments that are going to be received by the patient. So it has a great scope, a large scope, and a great potential. And to use artificial intelligence can help with treatment as well as diagnosis. So now we hear a lot about artificial intelligence, but what is it? What is artificial intelligence? What do we refer to when we talk about this? Well, there are multiple definitions. We can use this one that is the programs that have the ability to learn and reason like humans. Well, that is still a representation or a definition that is very broad. In reality, in my research area, what we use are algorithms that we learn machine learning or automated learning that composes all of these algorithms that are not explicitly programmed to find a solution, but they learn from the data. So we give them examples, the algorithms learn from this data, and then they give us a result. A subgroup of this automated we also have a deep learning, where it is a subset of machine learning in which there is different layers that we call neural networks. Maybe you have heard of these artificial neural networks. Those are the kinds of machine learning that we call deep learning. And it is probably the more complex way of learning because it has different implications and the restrictions of being able to implement this in the clinic setting. So we have different kinds of learning, of machine learning. Remember that machine learning is a subset of this artificial intelligence. And we don't have time to go into explain in detail each one of the types of machine learning. You have the supervised, unsupervised, or the reinforcement learning. But what we use usually is supervised learning. What does it mean that it is supervised? That means that to be able to train our models, to be able to feed our models, we need data that has a classification, or they have some kind of ground truth. So we can know the truth, and we can give the algorithm that truth that is already marked. So this is the kind of algorithm that we are going to use within this model, and this classification model, regression model, and different kinds of algorithms to be able to resolve each one of the problems. So to be able to understand a little bit better, what is this change? What is this paradigm? Because we have programmed all of our lives to be able to find solutions to the problems. But what is new? Before we would program in an explicit way, we would give the algorithm step by step to find a solution. As you can see, in the laser doesn't work, but on the top part, you can see that we have this traditional modeling where we have a model that is made specifically, and that through the computers we want to find that result based on certain data. In machine learning, we now are going to have examples. We have labeled examples, and the algorithm is going to learn from those labeled examples to be able to predict the result. And this is an iteration that we have to validate. And when we implement this kind of model, we have to have many considerations that go beyond the model itself, the results of the model itself. We also have to think about the explanation, thinking about the clinical use. Like, for example, the person who is going to use it is going to interpret that result, and they have to be able to trust in that result. So many times that is reversed to the complexity of the model. We have to consider an important point of how much data do we have to teach these algorithms so that they can learn from this data, because they are a limitation that is going to impact the performance the model can have, and that is one of the largest limitations that we have at this time, the amount of data and representation of the data that we have to be able to teach these models to be able to resolve the problems. So how do we apply this specifically in radiotherapy? As you can see here in these graphs, we can see that the last 10 years approximately have been explosive in the amount of publications that we have associated to AI in different areas of radiotherapy. You can see that there are other different search engines to be able to incorporate AI in radiotherapy for different applications, and useful for prognosis or for planning or for preparation of planning. So this has had a great impact, and it impacts the whole chain of radiotherapy from the moment of preparation, how we decide or plan the volume and the radiotherapy treatment, until we deliver it itself, when we have to adapt treatment during therapy, and also for evaluation of this. And I think that is the part that where we have presented many solutions, but we have not been able to generalize. However, there is a higher potential, I believe, to be able to go forward to personalized medicine, integrating multimodal data that can help us to predict what patients will benefit from the different types of therapy, and what patients are going to present, for example, toxicity, or not going to be able to benefit from this, so we can act in a timely manner. So here are some examples of these applications. We have the transformation of an image of an MRI into a CD image, just presenting examples to this algorithm of deep learning. And we can see on the right the reality, and in middle this image that was generated. Why do we want CT images from MRIs? Because the CT gives us information that is critical for the planning of radiotherapy treatment. In some modern devices of adaptive, we only have one MRI, so we would have to take them out of the MRI to take them to a CT scan. So this helps us plan with the patients. That has had quite a bit of success with the AI to be able to generate images of CT from MRI images. And it has also been very important in the segmentation of structures. As you can see on the bottom on the left, there is a structure. There is a tumor that was drawn by different experts. So as you can see, the variability of the different experts who segment a tumor on the same set of images is tremendous. So this is a step that is critical to be able to plan treatment, because we need to know where the incidence is going to be of radiation, and there is still variability that is pretty important between the different users, the different doctors who say where the treatment needs to take place. And this is a process that takes a lot of time, because we have to have an expert in each one of these slides. We have to have a tri-dimensional mark of where we want to treat. So in this stage AI, we can see on the right this ground truth and what artificial intelligence offers. So it allows us to not only select in a very efficient ways, because it is very efficient and automatic in structures like in normal autonomy, in normal anatomy, I'm sorry, and it also helps with the selection and this marking of the white volumes that need to be treated. Here it becomes very important to consider the amount of data that we have available to be able to teach these models, to know what, so they can know how to segment. I just wanted to present as well two applications. One of them has to do in the calculation of the distribution of the dose, where we can teach an algorithm with the image of the patient and with the energy flow, uh, what energy is going to be delivered. We can teach them to see the distribution of the dose in a patient in a very precise way. And somehow jumping over part, but the results are pretty impressive, and that helps us to accelerate this process. This last application, as I said, there's a lot of research that is being developed in this area, but it has been difficult to be able to generate models that can be traceable or generalized, because due in general to the lack of data. So this is just one example of integration of multimodal data. We have the image of the patient, the distribution of dose, the mask of the delivery of the dose, and all of these data are integrated in this particular case to be able to predict if this patient is going to present some kind of toxicity after therapy or not. And this is a treatment for head and neck. And this is an example as well as the radio genomes to be able to predict the success of a treatment and have multiple applications as well. And due to time, I just wanted to present a few so you can have a general idea, but that we can also discuss these challenges and the opportunities that there are for the clinical implementation of AI in radiotherapy, what the barriers are that I can visualize and also that appear, and also the opportunities that exist. Because as I was saying, many of these applications that we can see, many of them have been very successful and they have gone straight to clinical, but some of them are still in the research stage. It is important, and a big part of the symposium has to do with regulation issues. And of course, even though they are there to protect the patient, of course, they also generate certain barriers that are difficult to tackle, especially for this kind of solutions. One of them is that the applications have to protect privacy and of the patient, and these algorithms are fed a lot of data, so to be able to get that information, it becomes a little bit more difficult as time goes by to be able to generate an application that has a good performance. So one of the challenges is in that line, and another one is that the algorithms, in many cases the more complex algorithms of deep learning, are difficult to interpret. It's difficult to interpret the result, and that is a barrier as well, because it makes it difficult so that the clinical environment can adopt these new technologies if they do not understand or they cannot easily interpret how the algorithm has made that decision. So even though it is a support for decision making, it is difficult to be able to get past that barrier. The other thing is that these algorithms, even though they offer solutions, this is also connected to the previous one. The final responsibility is belongs to a human. That is why it is so important to be able to incorporate explanation tools to these algorithms. There are international standards that have already incorporated some of these elements in their requirements of certification, like ISO, IEC. In Chile, we are not staying behind to try to go forward in this area, approving the first national policy of artificial intelligence, which was updated in the year 2024 and already is in Congress. We also have a bill regarding this. So there are certain things that are being in progress and the intention to go forward in this as well. Within the technical and practical aspects, I am running out of time, so I'm going to try to be quick. The availability of data and bias, I could say that this is the great barrier that we have nowadays. So these algorithms need information, and to be able to obtain that quality information is very difficult. So you can see here an example of the representation of the fact that we have low representation of gender, for example, can make the models suffer in the population that is subrepresented. In this case, for example, it could be the women, because the algorithm did not know many cases of women, therefore, their interpretation could be worse in this case. That is something that we need to go forward with. And as I had mentioned previously, you have to be able to explain the explicant. There are many tools to be able to give explanations that we can use, but they have a lot of limitations. There is research being carried out, and we need more research because it will be required for the regulatory issues to be able to explain why that result. So this is something that we still have to go forward with. And of course, this is also connected to information, education, and the training of those people from the health area, for example, the doctors, and they have to be able to connect or the algorithms and have the links and to be able to get to know the limitations of these applications, especially using the data set that is being used to be able to train. So they have to report and know the limitations of these algorithms. So that is key, and we have to incorporate that in the future in the training programs of health professionals. And finally, there are some perspectives and some opportunities. AI gives us this opportunity to automate tasks in diagnosis and therapy. The potential is huge, and we also have the potential to be able to have a democratization of access to diagnosis. So places maybe where there is no specialist, that their data can be fed into a model that can finally be reviewed by a specialist in some cases to be able to use different processes. We also have to work, but an important part of the development of these explanations of AI and the prediction of the response, I think that is key, and it's going forward quite quickly. And I think that is going to be very important in the development of personalized medicine, and we need data for that from different sources, clinical data, images, genomes, and others. So we need to keep going forward in that. And this data, on the other hand, we have very limited data. It is difficult to have validations that are independent. It is important that we can have the knowledge and the information to identify the bias, gaps, and limitation that these algorithms have in such a way that we can know what the capabilities are of a generalization. And also, education is key. So with that, I can end, and I understand the questions will be afterwards. Thank you.
Thank you, Dr. Paula Caprile, for your presentation. Now it's time for the presentation, Clinical Research of Medical Devices, the Colombian Experience, presented by Dr. Mukul Romano Zapata from the National Surveillance Medication and Food Institute of Colombia. Dr. Romano studied electromedicine and after that, on 2001, biomedical engineering, focused on biomedical instrumentation and also rehabilitation engineering and technology management. He has a master's in administration and management of companies. He works as an engineer on technical services in hospitals, public and private hospitals, and equipment device companies, also teaching in universities on undergraduate and graduate programs. Dr. Mogoil, you have the floor. Good afternoon, everyone. What I will present today is related to the regulation aspect and how we see it in Colombia, the clinical trials. And from the point of view risk management, what is it that we need to guarantee? For those who don't know about this, in Colombia there are two entities at national level. We have the Ministry of Health, which is the organization establishes regulations and legislates, and the other agency is INVIMA, which is the health entity that uses the legislations and applies it and complies with it. Within INVIMA, in our structure, we have the Directorate of Medical Devices and Other Technologies. This directorate is focused on the surveillance before and after it has entered the market, and for reactives and other kinds of technologies, such as tissues, organs, and some other advanced therapies. In this cycle, we collect absolutely everything from the beginning, from the clinical trials to the aftermarket surveillance um, and the manufacturing, of course. In the research area, we have two parties on INVIMA that are in charge of doing assessments of the documentation, protocols, clinical research. They are the ones who review those files. And the goal is to determine safety and efficiency of the technologies. We have the specialized room of medical devices, which is advising committee of INVIMA, and it's made up of several professionals, which in the past would be there depending on their profiles, the industry, or the academia. Today, with meritocracy, this is opened up and to a public consultation where we request for a specific profile, and whoever feels is suitable for it can apply and be part of it. And it will depend on the experience, education, and based on the cases that are discussed. And they can be part of this specialized committee. In 2002, after this boom, I'm sorry, in 2022, after COVID, and because of and there was an increase in manufacturing in Colombia in the areas of diagnosis and treatment of COVID and comorbidities associated to it, research, clinical research with medical devices was more relevant. And after that experience, we created the group of clinical research and support from this specialized committee. As you can see, this is made up of several professionals that complement each other. Some are external, some are um, workers from the health authority. So what's a regulation? There's a lot, but it's disaggregated, and this is because there are several actors throughout the process, and these processes are not working together. So the regulation authority needs to look at these different aspects to try to understand and to include all of these aspects and face all of the possible challenges presented by the industry. One of the critical aspects, 4725, which is from 2025, which is the framework of Colombia of medical devices. But after that, and complementary to that, we have several other norms that standardize certain aspects associated to research and development, and some other support research and development in our country. We know that our Latin American countries are mostly importers of technology that gets to our countries, developed countries. And we saw during the morning presentation when we were hearing about the MDSAP programs, where we have these five big producers globally of producers of medical devices. And this is a challenge for these entities because we need to know how to work with this. And here I want two relevant aspects I want to present, which is the ethic and bioethics and scientific integrity. Because when we talk about this, sometimes we focus only on the technical aspects, but intersectionally we have certain elements that, and in those we have ethics and risk management. That's important because we need to focus on that and base our work on that. In 2021, the COMPES report was released in Colombia. These reports are the ones that open up a legislative process, and these are processes that include economic resources to promote it. In this case, that led to Law 2162, where the Ministry of Science, Technology, and Innovation was transformed. It used to be called Colciencia in Colombia. Today, we're working with the Ministry of Health, working on the regulation of the medical devices from the point of view of the inspection of the studies that we do. So we have three aspects on the inspection, and the goal is to have an authorization of the protocol of this research on how it should be done. And to do so, it's necessary to comply with certain requirements. There are also international standards, and the one that looks most like it is guidelines of IRMDF, and it's related to ISO, an international regulation, voluntary regulation, that is also similar to Helsinki agreement, that has similar regulations from the ethical point of view. Once the study is authorized, it should be developed in establishment, in this case, an institution, a health care providers, clinic, like we saw during Dr. Rodriguez's presentation, where they did the testings of their equipment. And it's necessary to have a step-by-step work, controlled and standardized. What Dr. Caprile just showed with data, access to information of the patient, it is important to have clarity on that risk management, the access to data of the study as well as the patient. And why? It's because it's necessary to have that consent first, and based on that consent, what am I doing with this information? And we need to know that this that was approved in that file, and we need to know how is that applied and used. And then we have the third aspect, which is health control, and this health control is the one that we don't really like as much, but it's basically that once this is authorized, how do you maintain this? And if it is maintained, or if it isn't, and it is deviated from the process that has already been standardized, it is necessary to apply this control to get them back on track. So how do we do the surveillance? We do it based on risk management model, and in our institute, this is a model that was developed 7 years ago, and it was standardized and validated in time. And it includes three specific criterion, and this is how we call it, but this is based on risk management. So we have severity, occurrence, and affectation. It includes several variables depending on the type of product, in this case medical devices, and clinical research. And we use variables that are intersectional, and some are specific depending on the approach. And it allows to collect evidence of what the market is offering, and compare it with the research, with risk management, with commercialization, and the novel technologies, and how is that being developed in the country, and what's the behavior. So it finally says the research where they or this project where this research is doing, or there's a group, and we can compare, and with that we can prioritize some studies, some research, and not others. So as of today, we have 24 clinical studies that we are doing follow-ups, and especially, and the specialty that we are working mostly in Colombia is the cardiovascular um, area. And when we say cardiovascular, we mean that bringing products that other countries are also trying to develop, so that we can prove that there is efficiency. And it's the first time that these studies are being tested in humans. This means that since this technology is new and they are innovating on this technology in Europe, United States, and at this moment in Colombia, we are receiving this de novo technology. So when we look at that and the regulatory aspects, it means that this is they need to prepare, because these are new technologies. How are we going to assess all of this before manufacturing? How are we assessing this information? And so then all of this is associated to research centers, ethical committees, and they can guarantee all of these aspects of these investigation. So how do we look at it? Because we are just a part of it from the regulatory uh, point of view for clinical research. But during this scientific conference, we've seen not only clinical research, we have basic and applied research, and one supports the other, and this evolves. So how do we look at this interrelationship from the academia, how they start doing development, but how can we scale up those developments from the health level, so that they comply with the goal that they were thought of in the beginning? And it is very challenging. In Colombia, in 2020, the government back then, after and during the pandemic, said, okay, so what happens with the different aspects from the social economical point of views? And in the health sector, they said it could be very broad in terms of research, but we decided to work on that because they basically screened us, and we realized that there was a lot missing from the research aspect of it, and we decided to prioritize health, and for those to be the same as the priorities that we have for research. So to understand where is research going and where are those interests of public health, what's the epidemiological focus of a country, where is it going to? So are those areas apart, and how do we get them together so that we began walking the same path to increase the quality and the amount? You can say, okay, yeah, let's produce, let's keep on doing. Universities are like that, keep on doing that research and different products. So it doesn't have to do with the quantity, but with the quality of the research. And in 2022 and 23, there was a regulation impact analysis on clinical research with reactive diagnosis, like in vitro and medical devices. And there were several key aspects taken from that. It required harmonized requirements and to have the conditions to have a clinical research, to establish indicators, because what's not measured cannot be improved, to articulate intra and intersectorial the different sectors, to manage the work so that the participants, that is the patients, have a key role in the research, and to get all professionals of the research involved. And this is a map at the macro, meso, and micro. These are different entities with different responsibilities and activities throughout the research process. Post-market level, assessment, clinical assessment. That's also within many of the areas that we just saw. Lab tests, simulation tests, models, simulation models, anatomy simulation models, or animal models. And the industry proposes these challenges, and as health authorities, what can we do? We usually associate regulation as Hulk, the Incredible Man. When Hulk is Nick Banner, he has the jacket and everything's perfect. He looks very fine. But once we have this transformation and innovation grows, it's when we're left short and we have too many challenges ahead because we have a lot of information and different applications. And it's important to include that as health authority because you need to assess that information and tell the researchers, hey, stop. Our approach is not risk. Risk can be found in any application in the health sector because with just one patient dying and one patient having complications, that's enough. So how many cases of morbidity we have? No, just one is enough. There are different applications for 3D printing, not at the mechanical level only, but also at the prosthetics. And we're seeing that we have and we're seeing this in advanced therapies as well. And we need to authorize those research, and so we need to have that information before doing so. So it's basically a translation of our research and not literal translation from language to the other, but how to take that to practice. And so that it's not just bordered research, but applied research. Collaboration between different entities, those promoting entities, regulatory entities on the other end, and the development of that critical mass, those who are in the clinics and the academics. Collaboration networks where we can have uh, basically a collaboration between disciplines and share what each party is doing to get all actors involved. That's necessary, and also guide these resources. And this is what Corfo was showing during the presentation today. And for Colombia is the Ministry of Innovation and Technology. And with that, development entrepreneurship gets stronger because the idea is there, but we are missing the economic support for it to develop. And once we start developing this idea, we have to continue working with the health authorities because just like the doctor showed us, uh, in a very diplomatic way, Dr. Rodriguez said that it took a long time for them to show the FDA that the risk was less compared with the benefit of the therapy that they were developing for those patients. And that's the approach as health authority. We need to standardize through a regulation requirements the risks.
We would like to thank Mucoil for his great presentation. We would like to invite Dr. Gabriele Claudio Maria Graciela to please come on stage so we can carry out a small round of questions, and then we have three very interesting presentations of those who sent in their works and were selected for oral presentations. So this is going to be very brief. So we have Dr. Gabriele, who showed us her presentation having to do with artificial intelligence and radiotherapy. Maria Graciela from ISP showed some of the information related to the design and development and manufacturing of medical devices. Claudia shared with us regarding the alternatives of finance by Corfo, and Dr. Mucoil shared his experience with the sanitary from Colombia with the new technologies, the different experience that they have from Colombia. So these are the people that we have on stage. Please feel free to raise your hand so that we can have 10 minutes to be able to ask questions and answers, and then later we will have the other speakers who will come present their papers, their posters. Is there any question in particular? We have Corfo, for example, if you want to know more about the programs that Claudia told us about, or is everything very clear? Please let us know your name and if you represent an institution. Good morning. Yes, I am very interested regarding the following question. In what sense? I am a doctor from the private from Peru. I'm a doctor from Peru, and my question is there is a possibility of the grants include countries internationally, even in Europe, South America, and in this case in particular with Peru? Is there a possibility of any kind of cooperation or support or grant? Yes, we work with national ecosystem in terms of beneficiaries, but we do have a work line that is specific with some who are, which is Startup Chile, that also works with entrepreneurs or people who are working that are not from Chile. This is a very small line, a very small stream. Therefore, you would have to see the possibility to be able to enter into that category. And in collaboration with technological entities, internationally speaking, even though we do have agreement with different countries, today there are some programs like Technological Consortium in the line that we have that could come as co-executors or allies of some projects having to do with consortiums. That is the figure that we have. Now, in terms of the individual projects, the subsidies or the grants are mostly national. In those consortiums, I suppose that you have to be registered for that collaboration in an international way. For those, is there any protocol requisite where we can apply for more information of the consortium and the technological programs? That model, I recommend that you can see on the webpage of Corfo. You have the full portfolio of projects that we have today that are actually working. There you also have the different direct contacts for these consortiums that are working as of right now. But you can look and see what there is there and contact them in a direct way. Let's take advantage before we go to the next speakers. I would like to ask the INVIMA colleague, is there any recommendation for the innovator national manufacturers, considering the experience that you have had in Colombia? Here, I will repeat the words that are very pertinent from the Dr. Rodriguez. Perseverance, persistence. That is, don't lose your focus, don't lose your push, your impulse. And empathy, in the sense that beyond empathy is to generate a relationship of understanding with this health entity. So it doesn't look like that wall to development and innovation in the country. And the entity has to have certain requisites so there can be a standard, and that focus is for the to make sure that we limit the risk in case of a patient especially. So when you see this technology, and as I always say, there is a great difference with medication. Medication is administered is metabolized, synthesized in the body, and then eliminated. However, a medical device that is implanted, for example, it is forever. So it is when I am starting off to research regarding a device or manufacture a product like that, where our countries are low producer of technology, but they are learning, and we can see how the material is there. We need that push to continue in that direction. As final conclusion, developers, industry are in the hand with the medical, with the health system. Is not easy, but it has to be done. You have to open that space to have a previous dialogue or understanding. Thank you for your excellent presentations, and thank you all for all of those of you who are still here interested in this extensive yet interesting day that we have been able to have today. So to continue now, we have Dr. Janet Cidia is going to give an acknowledgement in, in appreciation of your participation today. So continuing with the program that we have today, we are going to present the following three speakers who sent their work in the frame of regulation, a pillar in innovation and development, and they were selected to be able to carry out an oral presentation. The first presentation is for the physical therapy, Roberto Vera Uribe, professor associated to the physical therapy of the University of Chile. The title of his work is Evaluation of Different Available Air Cameras in Primary Care in Chile for the Administration of Salbutamol in Children and Adults. Roberto, the floor is yours. Thank you very much, especially to the organization for accepting our work and allowing us to have the results. This is a study that was carried out in vitro in the laboratory, and we are wanting to resolve a problem since, as you know, one of the main problems that we have in, if you can help me passing the, I can't seem to get it to work. I'll keep going. As we know, the inhaled work is not useful if you do not have the correct aerosol chamber. And these chambers have advantages when you use them with the patients. And the patient problem that we have in Chile is that there is no regulation that says that they have to have certain certification, size, volume, or property that is anti-static, for example, and that impacts the amount of medication that the patient will receive. And considering the possible patients who are susceptible to this therapy, only in asthma, we have around 5 to 15% of the population who has asthma. A great percentage of them use these devices. So we know that it has an impact in public health in a significant way. And that is why the objective was to standardize the use with aerosol chambers for salbutamol, and we went to different areas and purchased the ones that were available, and we also communicated with the Ministry of Health to know the ones that had been purchased during 2024 in the public health system, and those were the 6 aerosol chambers that we used. Each device in this model, in vitro model, we used a simulator of breathing with different. We had one for children, one for adults. We selected a pattern for a standard for children or for adults, and a frequency for 13 for pediatric and 15 for adults. We placed this device that I can show you here. The yellow is observed. And yellow, you can see the mask that was used. We can see here the pediatric. You have a camera that an air chamber that was placed, and the salbutamol was the same when all of the different aerosol chambers. And then we have an Anderson cascade that allowed us to determine, depending on the size of the air weight, what area this could be deposited in. And we carried out this measurement with 30 repetitions, 30 evaluations per chamber using the different points. So we said how much was left in the chamber, how much arrived to the face according to this filtration, and we collected the samples and then analyzed them. Here we have a table. In general, they measured the deposits of the doses through the filters and in the chamber, in the airways, and also the total mass recovered. This was carried out in the adult and pediatric chambers, and what was interesting was the results due to time. What is interesting is that the different ones, for example, arrow chamber that is here, you can see in the first column the total deposit that was obtained and what is the equivalence. You can see in the circles there, these are the other 3 chambers that are compared with the arrow chamber that had the best performance. You would need to increase 2.3, 2.8, and 1.5 times the dose that is given to the patient to be able to have the same final dose, which is carried out with the best result, which is arrow chamber. And in children, we saw something similar, but here it's even more concerning because if you can see in this all briefs, which is given by the public health system, you need 9 times more dose. So I don't know if we go forward with the patients because this is not ethical, but what we could ask and is give this results to the authorities so they can consider this when they assess and have technical evaluation of the devices they use. Another important point is that when we carry out a consultation to the over 300 professionals of primary health care, they told us that they have problems with the valves of the air chamber. So we measured the resistance when they inhale and exhale. And as you can see, there is significant differences in the different flow of what we measured from 5 liters to 60 liters. As we know, depending on the patient, the kind of flow that is going to move. And they have different resistance to the opening of these valves. There are some that are too hard, especially more rigid, and many professionals what they end up doing, and even the patients, they remove the filters, which ends up being a problem, technically speaking, with the amount of deposit that they end up receiving, whether it be the resistance, whether it be in inhaling or in the exhaling. Another important point, you can see the air chamber, and many of them are very rigid. They don't adapt to the face well. So as you can see here in the graph, we have one that escapes significantly with the leaking. Since it does not adapt well to the face of the patient inhales and takes most of the air from outside the chamber instead of from inside the chamber. So there is a great leaking, and therefore it ends up being very inefficient when comparing it with other air chambers that do have norms that are comply with the European norms or for the FDA, for example, with different flows, whether it be in the resistance or the amount of leak In inhale or exhale, it was very significantly different. And here you can see an exhale resistant comparison. Why is it important to see this in an air chamber? Because when you apply the puff in a patient that does not cooperate, then the valve has to close adequately so that in the internal volume of the chamber there is no exhaled air that is warm and humid. Therefore, then that in the next inhalation or the next puff is going to reduce the medication because the molecules are affected and it reduces the amount of medication that reaches the lung, which is what we want to happen. So in this graph, we can see I would not recommend the first one, which does not close at all and it loses everything. And the same for adults and pediatric. There are some that have exhalation leakage that is very significant during exhalation. And in conclusion, with this study, what we want to show is to compare without knowing what was really happening, because there are copies of the original ones that have certain certifications that you can see, and they're identical. So I even recommended them to my patients. I would say, and there's no study. There was no study. But there are studies in the ones with a certification because they are required. But those who do not fulfill the certification that are very similar copies, they only require that they have a certificate of the customs, and that would be pretty much it. And from saying that they are being manufactured in a well way, but they do not fulfill the use for therapy with the patient, which is a great challenge. And I think that is why we were very interested in being able to show the results of our studies today. And what we also would like is to be able to give evidence for authorities who are in charge of purchasing as well as for professionals. We know that you have all the weak points of an in vitro study, and we have tried to be very rigorous and very precise so that this could be very valid. We strengthened the in vitro so we could have strong conclusions. Thank you very much for your attention and the opportunity to present. Thank you very much, Roberto, for your results. We are going to be in communications with you soon. The next presentation is for the Carol Racorza from the Department of National Agency of Medical Devices of the Public Health Institute in Chile. The title is Evaluation of a Program of from 2004. Mammography. So it is the program of evaluation of mammography quality, the review and balance of the equipment and images of mammographies in Chile during the year 2024. Thank you for being here. My name is Carol, as I said previously, and I'm going to present what we have carried out in the context of our quality evaluation program, which is PECM, the review and balance of equipment and images in the mammographies in Chile during the year 2024. This was carried out together with Catalina Ramirez and Gabriela. So, as general context, we know, and they commented a little bit about the cancer in Chile is one of the main causes of death in the country, and within this, breast cancer is one of the highest, and it has mortality of approximately 11 of every 100,000 women. In this context, the mammography is the main test for screening, and we need a good quality of diagnosis to be able to optimize early detection and optimize the treatment. In this case, it is very important to make emphasis in the quality of the image. That is very important for this kind of exam, because no matter the expertise of the person who is doing that, and no matter what their experience might be, their vision, If the quality of the image is not adequate, then they are not going to be able to carry out an adequate diagnosis, and therefore we are not going to be able to have an early diagnosis and be able to improve the treatment for the person. That is why, particularly in this kind of testing, the quality of the image is very important. As we know also that cancer is in an increase of cases, there has been an increase in the amount of mammography equipment available throughout the country. Therefore, also taking into the National Cancer Program, where the law has within its objective, as it says here, to establish a framework for planning of public policy destined to establish cause and prevent the incidence of cancer, as well as establish the adequate treatment and recovery of the person. If we add this to the technical, what we have from 2021, which is the guideline of the quality of mammography, with the main instructions to define and establish the correct procedures to be able to guarantee an adequate test, and also in support with what the decree says of to the ISP has the role of making sure that they have quality of the efficacy and safety of the equipment, in this case of the mammography. And to be able to do so, that from 2023 we have developed in ISP the program of quality evaluation in mammography that up to last year was a pilot program carried out mostly in the metropolitan region, but as of last year we already started to open up to have it a national program, taking it to the different areas of the country, in different regions even. So with all of this, the objective would be to contribute to the protection of radiological protection of the patient, ensuring quality through images, through the evaluation of the mammography machines for public and private services, as well as in support and implementation of the technical area in number 2214, which I mentioned before. And also, we want to ensure that the equipment can fulfill all of the technical infrastructure and diagnostic quality defined by the at the level of the country, and also with international standards, like for example OIEA and in European and North American regulations, and also presenting relevant results and findings during 2024.
How do we do this? In 2024, we visited 8 services of imaging in the metropolitan region and one in Antofagasta region. In total, 11 teams evaluated. We did applied several quality control, safety control, mechanic control, dosimetry, and quality of the images, following the steps we mentioned earlier. We have 11 teams that were assessed in total. 4 of them are services of APS, and 7 of these teams are health service centers. We covered 6 communes in total. So how do we do it? We need to use different materials, and I'm going to go from left to right. On this side, we have a solid state detector called Radical that allows us to identify different parameters such as productive layer, dosage, the rate of the dosage. We use also the contract of Cerimam that has a random distribution of discs of different thickness of gold, and we use a camera parallel and different acrylic plates that are polymethyl to simulate different thickness of the breast. With it, we can assess the symmetry controls such as HBLA, and with a medium granulometry with automatic control to exposure as well, which is how do we look at the characteristics of the beam for a specific thickness and how the characteristics change according to the thickness of the breast or the acrylic that is assessed. And we also assess the quality of the image, as we can see in the first image, and the performance of the detector by measuring the answering response and the noise. The results can be observed as such, with control of safety and of the services in general. We know that 82% complies at 100% all of the measurements that are assessed, and only 80% complies with an 80% of them, of these measurements. On the other hand, 73% complies with the current regulation. And what it's most interesting and I'm more concerned about is that 82% of the teams assessed have a good system of automatic exposure, which means that it recognizes and it compensates all characteristics according to what it will treat. And in addition to that, all of this also have quality, image quality that is suitable with a detectability above 80%. And it's detected within the recommendation and the established tolerance. However, what I am concerned about is that 27% of the equipment have a dosage less that's below the measurements that we have assessed. So it means these equipments are giving more dosage than what we expect, and there is an amount of equipment that has insufficient conditions to comply with what they are offering. Some of the finding is that we have found a lot of refraction equipment with combination of different parts, spare parts, and there's also a lack of maintenance and calibration from the technical services and the dosimetric control or quality controls from these services. And there are also problems in the extraction of the images that are not processed, which are the ones that we use for this evaluation. In addition to that, there is a lack of establishment of a reference dates in order to do all of the other controls. So to conclude, we've seen that there's a need of doing a regulation to everything related to mammographies, and there's also a need to establish the initial reference date so that we can have a reference and assess the work in time. It is also necessary to establish quality control protocols and implement quality guarantee programs, which is similar to what we are doing. And it's also important to improve the quality of the images of the equipment um, to have a good quality of diagnosis. And this also is lacking a maintenance and calibration of the equipment of the technical service to have a better state of the equipment. And to conclude, because of the results that we obtained from 2024, about approximately 1 every 4 equipment has failures in the detector, in the quality of the image, or in the dosage given to the patient. Thank you, Carol, for your presentation. And now we have a third presentation presented by Joana Benitez, representing the inspection group of the Public Health Institute of Chile. The title of this presentation is called Impact of the Limitations in the Validation of Medical Devices in the Health Registry of Chile. Giovanna, you have the floor. Good afternoon. Thank you for having me. Today I'm going to present about the surveillance of the medical devices that don't require to be part of the medical register of the health registry. Why is it important? What are the spaces for improvement? And what are the challenges we have ahead? To give you some context about the history, I want to remind you that this regulation of medical devices is something that has been applied since last century. It's Decree 825 from the year 1998. This is a decree that establishes that Chile was one of the first countries at the Latin America level in regulating these medical devices, but unfortunately it wasn't possible to implement it because and which is why now we need to do some changes at the legislative level. Another important law was Law 20,724 from 2014, or the law of drugs net 1, where it was established that medical devices had to comply with the requirements of the customs law 18,164. And thanks to this law, in 2018, we were able to implement the requirements of the certificate of custom estimates for medical devices that didn't require to be part of the medical health registry. This allowed us, and in addition to uh, being a surveillance when it was first received, and before that we you didn't do that, and this allowed us to collect very valuable and very necessary information. So what's the importance of the surveillance of this entry point? Well, it is to detect any issue that the products may have when entering the country before these products are commercialized. And like so, after obtaining the findings, it's necessary to coordinate with the actors that are inspectors and have the power to inspect so that they can apply health measures. This is very important because our country, it is estimated that about 95% of the medical devices that are commercialized are imported and only 5% are made in Chile. What else can we gather from this? Other information? Well, from 2018 onwards, with the assessment of the CBA analysis of those databases, we were able to identify what are those medical devices that are imported in our country, who is importing them, who are they, who are the manufacturers, the legal manufacturer, where are they coming, is this coming from, what are the address, the final address of the storage and the warehouses, and what are the medical devices that are entering the country. This information is really important because these are today, this is today a tool that will help us whenever we face regulatory changes, specifically when the law is approved, a law that is sitting in Congress since January this year. With what we currently have, we wanted to do an assessment of the spaces for improvement that our current system has, our system of paperwork and import. It has helped a lot, and we've made a lot of progress with it, and we know that there's a long way to go, and we observed several spaces for improvement. The methodology that we applied was, and the first thing that we did was do a review of the bulletin of surveillance and certification of CDA of the medical devices that don't need health registry or be part of the health registry. This goes from 2018 to 2023. And in addition to that, we did an assessment of the current platform called Hicona 3.0. It's a platform that all of you use to proceed with a CDA of medical devices that don't require a medical to be part of the medical registry or a medical registry. And the most important thing that we found in this platform is that the fields don't have a previous validation and that the information is filled by the user, filled out by the user. And what's the issue with that? in order for us to do a surveillance of the entry point, we need to validate all of the databases. And it's important to consider that nowadays we receive 150 imports of medical devices, and we validate all of the risk classes of the users, the legal manufacturers entered by the users. What have we found in these validations? Well, we have found risk validation, which is approximately 23% of the products, and we see that this can be categorized in the classification of risks, and it can be part of it because there are inconsistencies in the description of the products. The other thing that we found is that we have we found products entered in this platform and that aren't medical devices but are products that require health registry, and also medical devices that require the health register and that are entering through this platform. Now, as I mentioned earlier, all of these findings that we made, we channel them and communicate that to the competent authorities that have the inspecting capacities and that they can take the necessary measures with these importers. And this is something very powerful because despite the fact that we have a space for improvement in our platform, the surveillance of the imports in our area is something that we do. And it is important that we could, because we could be much more efficient and we could have more improvements in our platform. What I want to show you now is the number of requests of imports of CDA that are processed without a health registry. And it is the one that has the most requests in this institution. We have an annual average above 50,000, and during the pandemic, as we can imagine, this increased, and it was above 63,000 imports a year. The current problem is was already described, and because of the fields that are filled by the user and that the information isn't standardized in the platform, the work of validation, it's work that we do thoroughly, but we're able to comply with the goals. And the most important thing, other than looking at the current issues that we have, is also looking at the future challenges and what is it that the future holds. What are the challenges? And the first of all of the challenges is the modification to the regulatory framework. We are very happy that the law of modernization of the systems, the national systems of health that entered the Congress in January 31st, and that includes powers for the Public Health Institute, as well as regulation for medical devices. We hope that that will be approved, and we are going to be able to apply that in a short period of time. That's the first thing. The second thing is that we definitely need to improve the information systems. And if we do so, we will have a much more efficient surveillance. And in addition to that, once we modify the regulatory framework, we are definitely going to we're going to need to be more efficient because we are going to have a lot more tasks. And once we have these information systems ready, we will be able to apply tools to the databases and apply risk matrix for surveillance on the entry point. And lastly, and I won't add any further on this because we already discussed this in the morning, and it's something that will be included in all processes. We'll be using AI for the task of surveillance on the entry point. Thank you very much. With these three wonderful presentations that provide results and evidence that allows us to justify the update to the regulatory framework, something that is necessary for Chile in terms of medical devices. With this, we conclude
organizada por el ICP, trata principalmente de dar un espacio a todos los investigadores que hacen.
You can find lunch on the side of this tent. Thank you for your interest and attending this conference.
Once again, the Public Health Institute would like to welcome and thank you for your presence in the 17th version of Scientific Conference. This year, we are talking about ISP, promoting science and regulation for public health. The main, this block now that you are participating in is called Research in Public Health. And our first conference at this time is called Verification of NGS Panel for the Detection of Tumor Biomarkers to support the use of personalized cancer therapies in prevalent and high mortality cancers in Chile. This first conference is going to be carried out by Jorge Fernandez, who is the head of subdepartment of Genome and Molecular Genetics of the ISP. I am going to give a brief introduction of Dr. Fernandez because I don't want you to not know everything he has done. So give me a second, doctor, please. Dr. Fernandez, you let me know if we're saying it correctly. If not, you can correct me. He is licensed in biology of the University of Talca and doctor in microbiology of the University of Chile. Member of the surveillance of genome of COVID-19 in Latin America and also diseases transmitted in food in Latin America and the Caribbean. He has developed projects of research in the area of infectious agent, cancer biomarkers, tumors, genetics of vectors. He counts with 106 publications, scientific publications, 88 of them in international magazines or journals, and 18 in national journals. And I know you're waiting for him, and I leave with you now, Dr. Fernandez. Good afternoon. I am going to talk about a project that we started in 2023 and it was executed in 2024 in the Ministry of Science with funds that they gave us. I am going to start backwards. This is the team of co-researchers. We have the Genomic Laboratory of Cancer. They have been a great support for us. SECAN, also Biobanco of Tissues of the University of Chile, and those who also work in the new human genetics and molecular and genomic genes and genetics. And we have Ivan, Fernanda, leading this team, Barbara and Eduardo. Cancer around the world, we estimate with data from 2022 that around 20 million people have had cancer and 10 million approximately passed away. The more prevalent cancer is lung, breast cancer, colorectal, and prostate. In Chile, in 2022, around 60,000 cases, 31,000 passed away. And the most prevalent is colorectal, breast, stomach, and prostate. In the higher part of the image, you can see the regular cells with one that is sick that starts multiplying and it has a population, and then they have co-mutations that happen in the same cell or in others, and you have a second or third population that starts adding. So when you have a disease with cancer, you have a population of cells with mutations. Not all of these mutations give space to cancer, and not all the mutations are useful for therapy either. Current medicine and how things are carried out in Latin America, around in the world, and here in Chile as well, is that we have an average patient, we have a standard treatment, and you can have some results that are better in some, not beneficious in others, and others that have secondary effects. But with precision medicine, taking in consideration the genetic events in the genome, as well as the encounter and the lifestyle and the data of the population, you can have a personalized treatment. So these treatments can improve the result depending on the patient and the treatment that can be carried out, and also reducing secondary effects. Another genetic scheme that is more genetic, you can visualize the markers of prognosis, the markers that are predictive of drug sensitivity and resistance, and adverse events as well. And they're different for every person, and it depends on the mutations they may have. Here you can see the different kind of cancer with the different kind of treatment. Associated to this, you have the different therapies, and these therapies depend on the mutation that is present, and for that, currently we use sequencing, massive sequencing. I'm going to go back. The objective of this project, I wanted to do different, but we have to compare two methodologies. A methodology that is validated by doctor in the University of Chile, and it's a commercial methodology from Illumina company, which is a panel of 50 genes from Dr. Marcelina. The first thing we did was take samples from colorectal cancer. These samples were extracted with their nucleic acid, and we had the DNA concentration. And to be able to have an idea of this methodology, the SEC tumor has to do with hybridation within the markers, or there are things that are captured. There's a selection. However, this methodology is used in the daily. Oh, you have the probe. So we have these 25 genes. This is a global panel that is selected with genes and mutations that have to do with the Chilean population in Latin America. The other ones are more general. From the point of view of the informatics, of the analysis, as well as the laboratory and clinical part, the tumor sec has its own. Basically, you don't have to pay to have the analysis, but in the other panel, you have to pay to be able to know what the mutations that are present. For example, here in the software of TumorSec and the CLC, you can see the variants and you can know what the mutations are. All of this analysis is carried out with things that we have here in the Institute. And to be able to compare this methodology, we selected 15 genes that are in both panels. One has 25, the other one has a different amount, but we selected 15. And the markers of colorectal cancer are the ones in red. I'm not going to go into more information, but those are the ones that are involved. The first thing that we had to do to be able to standardize the methodology was to use a positive control with known mutations and the frequency of them. And here is a regression that shows that our system allowed to be able to detect all of the mutations and even the frequencies under 5%. That is also very relevant. Many times the level that they cut at is above 5%, but when we have a higher, more depth in genetics, you can take into consideration that mutation. From the 67 samples that we worked on, 30%, 20 people, did not pass the quality standards. That is also very relevant in this because they have to have a certain it has to be very precise. If not, the results are not correct. We had 47 samples, and from those, and the variants were clinically relevant. We only had 43 that were clinically relevant. That means that they were not repeated. That means we had patients that had the same ones, but those were unique. And the concordance in between both methods was around 94%. I'm going to refer the table at the bottom to be able to show the top part, to be able to explain the top. With the tumor-seq system, we found 83 variants. Only with the other, we found 22 that were not found in the other method. In AmpliSec, 65 variants and 4 of them that were not in tumor-seq. So this means that 61 variants were detected by both systems. However, there is a method that detects more. In the graph at the top, you can see 43% are the ones that are shared. 22 is from one methodology, and the other 2, which were 4, but they're 2 because they were already repeated in one patient, and they were in AmpliSec. These are graphs that show, if you look on the left, that graph has the genes, and on the right you have the patients, and in green the mutations and the change in amino acids and the mutations of the different lesions or other kind, and we have the AmpliSec and the tumor-seq panel. And there you can see that there is a higher amount in the tumor-seq panel. The great question is, how can we differentiate? What are the characteristics? What is the positive and the negative in each one of the panels? Because each of them have certain characteristics that differentiate them. One of the most important things is capturing through hybridation. Like I said before, you place a probe and you cover most of the gene, but you cannot do that with AmpliSec, which are hotspot areas. So you can have regions that are not recognized. The other thing is that when you have a low number of DNA, then the capture of that DNA enriches the sample. So those are things that are important in the technical part of these methodologies. Within the 15 genes, tumor-seq covers 333 regions and 333 in tumor-seq and 210 in AmpliSec. Cost depends, you have the cost there, but there is something that has to do with the number of samples. It is different to run 1 sample, 2 samples, than when you have 60 samples. AmpliSec are for 60 samples, but we've never had 60 samples. So this is a methodology that has high cost, but that is being carried out in Chile in some of the labs. Sometimes you have a preparation time. One of them says 2 days, the other one says 6 hours. But however, in the update in the methodology, that is changing in tumor-seq. There is also something that is also very important that you have to take into consideration when you work with the concentration of DNA. Here he says 10 nanograms or 200 nanograms, that's a large amount. But when you use 10 nanograms, the feasibility of having information that is non-relevant is very high. So that is also very important when you work with a sample, that the sample has to be the adequate one. And the sample and the cut that is carried out has to contain part of the tumor, because if it's not going to give the results you need. This is all relevant when you analyze the samples. The important thing of this work that was presented in Congress, and now they just approved the publication in this journal, scientific journal, so we are very happy because this is one of the areas that we started off not very long ago, and it is a part of the beginning of the genetic human section that is not under me like it was before, but there is somebody else, and somebody else needs to lead this area now. And the conclusions. Well, there is a number of variants that were detected by both methods, and the difference that there could be the cases where there was a higher number of variants with tumor-seq can be due to the fact that the level of cut is under 1, 2, or 3% of the frequency. And this has to do with the depth of the analysis. So if you have low depth, you are not going to be able to take into consideration the variants that have 1 or 2%. You're going to have to be much deeper so they can be recognized as so. So that is also very important in the genetic analysis. As I always remember when I was out of the country, if you put in trash, you're going to get trash out as well. So you have to keep that in mind. One of the characteristics of the methodology made in Chile is that it has the informatic system included. It has all the different variants and it has it all included. So it is a system that is robust and complete that is constantly being fed with the information from the other countries as well in the database. And finally, as last conclusion, we are very happy because we can implement this system within the laboratory. We have people, we have the technical expertise, we have everything we need, and we have a good association with the university, and we are thinking about new projects with new people. And based on this same project, that in 2024 we won another project through the Ministry of Science. So now we are in the studies of identifying cancers, samples and cancers in breast and lung. Thank you.
Very interesting presentation and very clear. We thank you for that, and we want to give you a gift. Marcela Giron will give you this award. She's the head of Department of Surveillance and Research of ISP. Congratulations. Thank you. The next presentation is called Evaluation of the Neutralizing Immune Response and Genetic Characterization of Isolated Variants from SARS-CoV-2 and simian pox from vaccinated and or infected individuals in Chile. This is presented by Dr. Eugenio Ramirez, head of the sections of oncogenic virus from the Public Health Institute. Dr. Ramirez is a biochemist of the Department of Chemical Sciences and Pharmaceutical Sciences of the University of Chile. Since 2004, he has been the head of the subdepartment of oncogenic virus and viral diseases from the Public Health Institute. He's a virologist, and his lines of research have been focused on molecular biology, pathogenics, and in different models of viral infections such as herpes, paramyxo, toga, bunia, retrovirus, papilloma, and coronavirus. He's currently doing research on immune response, genetic variability, and pathogenics of HTLV1, 2, VPH, and SARS-CoV-2. Dr. Eugenio Ramirez, you have the floor. Good afternoon. Thank you for the invitation. Everything they read is from someone else. Yeah, that's a lot of information on that paper. And I will try to talk about viruses, and because of the time that I have, I only talk about COVID and not simian pox. Simian pox will be discussed maybe some other time. And I want to do something, and I want you to do it as well. I always like to do this, but I want you to imagine things. As of today, I have never seen a virus, but I have dedicated my life to those. And it's like we work with ghosts or myths that you have to study every day. And during the pandemic, nobody worked with SARS-CoV-2, and we were forced to work with this model and use old models to see how it worked. Just one glass is enough, thank you. And we had to change some things that seemed uh, to respond our previous knowledge, but it wasn't enough. And when you look back to the past now, it seems like things that children would do. And I think it's great teaching because when you say that you know all of it, it's basically that you know nothing. We always have to validate ourselves and obtain new knowledge and work with new people because it helps you get younger. Otherwise, you'll stay behind. Like Dr. Fernandez said, I haven't given anything yet, but I think I would have to for the next time. Well, you see here three presentation of SARS-CoV-2. The one to the left is a microelectronic photograph where you see the structure of the virus. In the middle, there is a diagram, very beautiful, where you see the 3D structure, and you can see the red areas, which is from spike protein. And I'm saying this because great part of the research as of today has focused on that. It's a trimer, those trimers, and the protein spike, that yellow part on the lipidic protein, it's key because that's the first contact that the virus has with the white area as the target, which in this case is the cellular receiver. It is important because vaccines as of today have been developed against spike. And if you look at the outer part of it, and that's what you need. I need you to start imagine. If this were orange, you would only see the outside of the shell, not concerned about what's inside. What are those seeds and what's in it? And in 2020, with the participation of Maria Teresa Valenzuela and Dr. Fernandez, and desperate facing COVID on May 2020, the first thing that we did was isolating the virus. That's the first thing that we did. Nobody in Latin America did that. We did it here at the Institute. And with that, we did the first isolation and then a neutralization. Why neutralization? Because we needed to measure if those individuals that were infected were developing a protective response because there were no vaccines. The way that we had to do it was blocking the virus to avoid entering the cell. In simple terms, that's neutralizing it. And 40% of the Chilean population were at home, and some were around here running around trying to isolate the virus and doing neutralization. Dr. Fernandez was one of the people who were working with this, like the Avengers. And the first thing we noticed is that at the 15 days, the people had a very little response to the virus, and afterwards we would see it at 100%. Everybody was happy, but when then we started seeing that antibodies would decrease, something that we didn't expect. And then on the image of the right, we were able to see that, before the first vaccine was launched, we saw that there were several strains around the country, and we found that depending on the strain, the same person at the same time had a different capacity of blocking it. So the spike, those blue ones are those blue dots. But if you look to the other ones below, then we're too close to zero. Those were the new strains, which is people without vaccines, depending on the virus that they had a capacity of blocking it or not. That caused a lot of tribulations, and that is why we propose a treatment, and we're able to work on the neutralizing response that people had on time after time passing. And I have to acknowledge that about 60 people of this institute offered voluntarily their work and we had to take samples of them. They received the vaccination, and some still ask me, how are we doing? And yes, relatively fine. And what we would study was the neutralization. But what these images show is that neutralization doesn't only depend of the virus spike with the cellular receptor, but also other components. And that makes it a little bit more interesting and more fun and more complicated. And what we can show here is that this group of 60 people were observed with several samples. We did it for a period of time of 60 days. And if you look at to the right, you can see that we followed some for 201 days, and the one that we followed the least was 36 days. Some were bored after the study, which is natural response. And in between, everybody was receiving the vaccine. The first ones were all Sinovac, and then some received the Pfizer vaccine. Then another dose of Sinovac, and then another one of Pfizer. And the third dose included three vaccines, Pfizer, Sinovac, and AstraZeneca. And I don't know if you remember, but each one of us had different kinds of vaccines applied. And when we measure the neutralizing response in time, we are capable of observing the first part up to the third dose. Follow-up is very heterogeneous, as well as the antibody titles. Right after the third dose, you see that there's a stabilization, and this it's maintained in time high. And if we compare this to the right, the response of the antigen of the original vaccine, of the Wuhan virus, and then if you compare that to the first Omicron BA.1, you can see the response of the neutralization. And those were the people who already have the first bivalent vaccines. And although we were very happy about this, we were very cautious about that. And to the left, you have the comparison of Wuhan and Omicron. Both were in the first bivalent vaccine. And then we ask ourselves, what happens with those variants that are not included in the vaccine? And that's what you can see to the right. It varies a lot depending on the strain that was used to do the neutralization. However, our concern went beyond that, and we wanted to study the immune response. And what happens if we start studying the virus now? Because viruses aren't as dumb as you think. Sometimes I believe they are smarter than we are. They have a very little genetic material, and have been here before us, and they will still be here after we leave. And so we said, let's characterize this virus genetically, and that's what Dr. Fernandez does. And we had a lot of discussions during our conversations and of P3, and not everything is based on the genetics. I want to know what changes in the genetics are translated into changing the behavior and in the transferal, because we allegedly have a very similar genetics with each other, and sometimes two humans that have the same genes act differently, and same thing happens with viruses. And when we looked at the virus, the one to the upper left, it has a very different it's a very different image, and then the other images show the damages that this virus caused. These small spheres that we call syncytia, and then others that, I'm sorry, no, those small spheres are the lytic variants. Those infect and break. That's what they do. And the other one generates syncytia, from small to large. And the behavior of this strain is different. The behavior of this strain is different, and therefore, the next year, 2023, we started proving what happens with this within the cells when it enters. What do they do when they're inside, and what do they do when they leave? That's the infective cycle. That's how we call it. And in the lab, we can isolate virus, we can count them, and we can similar quantities of the virus on cells. And that's what we did with different strains. And you can see the arrows here of the four targets that we assessed. First, the entrance of the virus. The strain, the Omicron, Delta, they're absolutely different in the capacity they have to associate to the cell. Some do a little bit more, some less. But what's interesting is that inside a cell, we see them entering at different timings. And to the right, we have two images. The one in between, it's 5 hours after the virus entered, without a previous treatment. And to the left, to the other end, we see remdesivir. So the viral replication has been blocked. So what we want for those first 5 hours, I don't know how to go back now. Can anybody help me? So what we did was infecting infecting the cells and wait for 5 hours. With that amount of time, the virus does not replicate, and the only thing we measured was the entrance of the virus. And then we observed that there was no correlation between the capacity of joining the cell with the capacity of entering the cell. There were delta viruses, or BQ.1.1, that's Omicron. Those would enter very rapidly in those during those 5 hours. Then the blue one, which was within the bivalent vaccine. So there was no correlation between the entrance and when it was joined. And there isn't a exact correlation either on the capacity of growing inside the cell. So if we measured it after 5 hours up to 72 hours, the curves, the growth curves of viral protein, as you can see, the one in orange, is the one that grows 150 times more than the others. And the blue one, it's like in the middle. That's Wuhan variant. So what does it mean? Variants stick to each other, and they do so at different rates and strengths. Once they are entering, they do it differently. Some are slow, but then it seems that the slower ones are the ones that have the faster reproduction. That's progene. And in virology, this has a name. And if we add the dynamic factor of time, the viruses change and they accommodate to the growth. And that leads us to study in this new project what is happening with today's strains compared with what we had in 2020, 2021. And our main conclusions are that all of the studies that have to do with spike, with neutralization, have a way of 30% reduction. And spike is on a very narrow area of the genome of the virus. But what's interesting now is we're not interested in knowing what's ahead of the body of the virus. We want to know the entire body of that genome. Why? Well, because we already know, and it has already been described, that there are mutations that reorder the cell. And this means that different viruses infect a person, and that creates a third virus. That's there's a progeny viral production, and nobody knows what's the impact. And we had the first recombinant, which was XBB, and now we have several. And what's the significance about that? Is that these recombinant are not in spike anymore. And we could say it might be spike, but now we're seeing changes in other genomes through the polymerase of the virus, through the internal proteins of the virus. And when you are studying the life cycle, that finally has an impact. The virus can enter very slowly. It might be hard for it to enter, but once it's inside, it acts. And that is expressed in living organisms and more dissemination in the population as well, which is why we are facing now viruses that don't seem to be as lethal, but are spread very fast, a lot faster. And the most important thing about this in the 2 seconds I have left is to thank the people who have participated in this study. Their contribution has been very key in the administration, management of resources, in their knowledge, and we hope to have more people participating, especially in this project that wants to study how this virus change and how they will change in the future, because they will do it. Thank you. And I didn't use the water. Thank you very much for your presentation. I think we all used our imagination and we were able to understand what you wanted to share with us. We invite Marcela Giron now, who will give you a gift for your participation.
Our next lecture is Antimicrobial Resistance, Antimicrobial Consumption and Resistance Profile Study in Clinical and Environmental Strains Isolated in Healthcare Centers in the Metropolitan Region. This presentation is by Dr. Esteban Paredes. There he is. Dr. Paredes is biochemist of the University of Concepcion and master's in science and microbiology in the same university. After that, he has the doctor of microbiology, molecular microbiology in the University in England in Nottingham, and he is specialized in molecular testing for bacteria, parasites, virus, and virulence associated. He carries out the surveillance of enterotoxins, bacteria, and resistance to antimicrobial resistance in the environment. Lately, he has started studies of genome sequencing of bacteria through Oxford nanopore technology in water and soil. And he is also teacher of microbiology, histology, and biochemistry. Currently, he is the one in charge of molecular microbiology and the microbiology of food and environment in the department, in the National and Reference Department in the ISP. I will leave with you then Dr. Esteban Paredes. Thank you very much for this presentation. First of all, I would like to thank the invitation to be able to show this project. This project, as they mentioned previously, Dr. Fernandez and Dr. Ramirez, is with funds of the Ministry of Science in the 2023 agreement. And I have a small alteration for the title you showed because it has two components. One of them has to be with the consumption of antimicrobials, and the other one has to do with what corresponds to the antimicrobial resistance. And that part of the consumption, we were showing that a little bit yesterday in a poster. So this part is more to the resistance. The first thing I want to show you has to do with the news that has come out publicly. We are even becoming famous in Spain due to this aspergillus fungus in hospitals, which has to do with certain news that catches our attention that happen within a hospital. The aspergillus is nothing in comparison to other news that have also come up in the news. For example, in the Clínica Santa Maria, there's a person that was pregnant, ended up with an intra-hospital infection or death that has happened, for example, in 2018, a little bit before the pandemic, regarding infections within the hospital, which is what we wanted to focus on during this study. This famous infection in the public health, before they used to call it intrahospital infection. And why is this so important? Because it provokes more morbidity and mortality in patients, the increase of the time that they have to be hospitalized, and more expense and deterioration of the life. They also estimate that 5 to 15% of the patients that are hospitalized could acquire some kind of IAAS, what represents economic load for the system, the health system. What is also clear is that a percentage of IAAS are also inevitable if they can be carried out with good prevention and infection control. So regarding these intra-hospital infections that are very related to the list that have come up in the OMS, in the WHO, are the pathogens that we have to keep in sight, the things that we have to be paying attention to because they're provoking large problems, especially associated to the resistance, antimicrobial resistance. And within this, we can find in the critical ones, we have the actinobacterial and enterobacterial, which is also many pathogens that are those that are resistant to carbapenem resistant. That is the focus that we used for this study, to focus in the enterobacterial, which are resistant to carbapenem. And this also reinforces the idea that, for example, the Klebsiella pneumoniae, which was already on the list, and even in the update of 2024 in the WHO study, that this is the fourth, if I'm not fifth. We had it in 2017, but it went up to first place in the year 2024, just recently, and as priority for its study. So what are these carbapenemics? To give you a little bit more, it is an antimicrobial that becomes in the beta-lactamic, and this is a group of antibiotics that particularly has the characteristic of affecting the membrane of the peptic layer in the negative as well as gram positive and gram negative. These antibiotics have the capability then of modifying and altering and destroying the union, and this way acting on the bacteria and being a bactericide of these. Therefore, this bacteria, as you can see, as Dr. Ramirez said, that they're not dumb. They are not, bacteria are not dumb either, and they create mechanisms to avoid the action of the carbapenem. And there are many things that have been written, and we wanted to focus only on one mechanism, which is the search of enzymes that can destroy and alter the carbapenem, which is the carbamenic acid. And in these, we have many kinds of ways, and we have these that are more particular, which are the KPC and BC, and the OXA 48 especially, and they are isolated from the hospital. So they take the antibiotic and they alterate them, and that way we have a bacteria that is resistant to carbamenic. And the institute also has a registry regarding the presence of this carbamenic and the resistance of these that is manifested through the resistance list that is in our page, in our web page. So therefore, the study was to show and to find these bacteria that are present in the different clinical samples, and we also went as inspectors to the hospitals, to different centers, to be able to take samples of the different surfaces at the time that we went to take the sample. We went to two different hospitals in particular, and we put HO1 and HO2 so that there is no conflict of interest. Or we went to Hospital 1 and Hospital 2, two of the largest ones that are here in Santiago. So what did we do? We had samples to where they washed their hands, showers, certain stations where they clean the glasses, and also from the residual waters. So we had the wash basin, cup washer, shower, nursing station, and residual water. So they had been those samples that had been isolated from the patient. So what did we do? We focused on the environmental part, which is what we saw in our section. We wanted to have an isolation of it in a selective way and an identification through the different ways, and have a detection through these plates where we can consider a panel of antibiotics. And all of these antibiotics, and we consider to include cephalosporina and many other panels of at least 17 kinds of antibiotics that were different. And in this way, we made it a little bit faster to be able to identify. After this, we also carried out a genetic analysis, and in this case, when we did this, we had the strain, we had the DNA extraction, and then after that, we had identification of resistance genes, and at the same time, we had a special selection to be able to identify, to be able to sequence the genomes of these bacteria and carry out the analysis and the informatics. What is the result of these monsters that don't care about antibiotics, literally? Well, in the clinical part, at least, there were 30 strains from hospital 1 and almost the same amount from the hospital number 2, in which mostly we isolated Klebsiella and pneumonia from 1, and in the B, we also see Klebsiella pneumonia as the main isolated pathogen from the patients. The resistance of these, we did it this way. We started to show the high levels of resistance to all of the different antibiotics of all the different groups, showing that only there was a certain susceptibility with colestina, and as you can see on the screen, and being the antibiotics which were susceptible to 100%. In the case of those no fermented, where they were also pseudomonas, we also presented very high levels of resistance in the samples of the clinical type. In the environmental, those, at least we isolated almost 300 strains from each one of the hospitals. where we carried out the sampling. And here we had something that was very interesting in the enterobacterias, in which the main finding was Citrobacter humanae, frundi, Enterobacter as the main ones that you can see. And when the non-fermented, we could see the Pseudomonas aeruginosa was the main one that was isolated. In hospital 2, something that was very similar, having to do with what we found, where we also found Citrobacter frundi was the main finding within all of the surfaces that were analyzed, and the same with Pseudomonas aeruginosa. And then the resistance profiles, they were much lower than in the clinical samples in the resistance percentages, and much more in agreement. They were much more concordant, and we did a separation between enterobacterials with the no fermented, and we also had comparison of the profiles. In no case was it with a species in particular, where it also shows that they had a good action of antibiotic like amikacina, apart from the classic one like amoxicillin and ácido clavulanico, and others which also had the third generation ones that also showed high levels of resistance. At the same time, in the clinical strains as well as the environmental, we could also see we carried out a PCR for identification of the genes of the beta-lactamasta, and we also could see that we were capable of, at least in a large portion, to identify the genes of beta-lactamasa that was present, as well as in the clinical samples as well as samples that were environmental. Of all of the set of strains, we selected 63 strains, 43 environmental, and 20 that were clinical, with a total of 63, and we had a complete sequencing of these in the clinical ones and the environmental ones. And in this case, what the first one that was identified is that they corresponded to the species that you can see there, being Pseudomonas, the one that was the most isolated, as well as the Enterobacter ormaceae, Klebsiella pneumoniae, and Citrobacter fruini that you could see in the clinical as well as the environmental. The analysis also showed and were able to identify the sequence types that the 63 were a part of, and they could identify that they could the sequence types, especially Klebsiella, Citrobacter, and Enterobacter, in which there is a scheme to be able to classify them within the sequence type. When we carried out an analysis that was more complete of the complete genome of the environmental strains as well as clinical ones, we could see that Klebsiella pneumoniae, also being the one that had the most resistance, had identified within the genome. This means that the majority of the Klebsiella pneumoniae means that they move between in a range between 5 and 20, that it could also be present in the clinical as well as in the environmental strains. And when we take the total of them, only understanding the resistance, we could see that the clinical as well as the environmental strains were capable of having a certain amount, in average, of resistance genes. But when you place an emphasis on the carbapenimasas that were present in the genomes of both types of bacteria, you could identify even that the environmental ones could have more percentage of identification of these carbapenimasa within the genomes of them. This is a better way of showing the huge variety of resistant genes that appeared, where you can see that in this kind of a scheme, where the biggest square is the glycosis, so these are the most present in the 63 samples. And then you also had the genes in the cephalosporins, and also you could see the genes in resistance against the carbapenemic. Then we carried out an analysis that was very interesting, which is where we align all of the different resistance genes that are present in each one of the different strains. Of the 63 strains, we take all of the resistance genes and we compare them based on this. What is the intention of this? To see if there were bacteria that had the same load of resistance, and in this way we found it to be very interesting, because we could identify that there was, based on the resistance genes, there were groups or clusters that had exactly the same amount of gene resistance, which was already showing us that the environmental strains, whether it be from the bathroom or the wash basins, could have the complete match with the strain that was a clinical one. And they also sequenced the same, and you could identify the same sequence types. And this, even so, was more determined, could determine more when you had the cloning of the strain, which caused a match in all the complete genomes, not just with the resistant genes, but also with the complete sequencing. And in this way, you could also identify three important clusters in which you could identify what the strains were and the clonal strains between the clone itself that exists in the environment with the one that existed in the clinical sample. Well, which were these? So now we could identify where the critical place was within the hospital. So this Klebsiella pneumoniae was found in the position 44, which means a wash basin in the hospital, for washing cups. And this was done with two different strains that were isolated in the same hospital. This hospital HO1, and they were two critical ones that we were capable of identifying from the where they washed the cups. And then we also had a lot of attention that there was an isolate that was clinical that was also a clone from this one that was also in the hospital too. This means that this could be a strain that not only is living in the hospital one, but also living in hospital two. And in hospital two exclusive, we also found a Pseudomonas aeruginosa, which is the sequence type 309, which is also the sequence type 654. Both of them were isolated also in the hospital, where from the wash basins where they wash the hands. Maybe I shouldn't give so much information, but the other one that also was a procedure room where they washed their hands that were there, you could isolate these pseudomonas. These were carried out, and they had a match with three isolates, with the biomedical laboratory, which were three patients who had a multi-resistant bacteria. These are the main things that we found as part of the study. And to be able to end, I want to show you two studies that were carried out. One of them that was talking about the importance of how important the sinks are in the hospitals, and that the impact, even on the way, the shape of the sink, and many of the publications talk about splashing, that the water splashes and that it be micro or nano drops of water that could end up staying around, and the bacteria could still stay in the environment due to the nano drop. And we don't have to blame the personnel because they can wash their hands very well, but if it's in the environment, it could be that it can be one of the ways in which this is still remaining in the hospital. And there's also a publication, this is August of 2024, where they also are talking about removing the sink and having different mechanisms to avoid, because this is where they observe these kind of issues the most. And when we went, it's the ISP is going to sample the hospital, and we said they're going to clean everything, even just in case, and maybe the most probable is that they did. And even so, we were capable of identifying these monsters of multi-resistant bacteria. But we also have to think about this, because this paper that came out published in this year in May 12th caught my attention, because it is from an area that is ultra mega clean, where they use the best, which is the NASA, to be able to send it to space, that is sterile even to the extreme, and they were also capable of identifying bacteria from these places. So what does this mean? For me, the point is more than eradicating these bacteria, because when I have talked or when we show this results to the hospitals, they say, what do we do? What is the solution to this? We have to burn the hospital down practically. Yes, basically, yes. The answer is yes. So I just wanted to show you that, and the conclusion are the results that I already showed you. And I think that it is important when we have the multi-resistant to understand the environment, the role of the environment. This also has to do with the surveillance of the genomes, to be able to know the environment that they are in, and to try to see if this is something, or it would have been ideal. We can propose a project for this year to do a new sampling, to be able to see what happens over time, because maybe there are changes. Maybe there is another kind of strategy that can mitigate, because I don't think we will be able to eradicate, but at least mitigate. So that would be it.
Dr. Paredes, very clear presentation and to know to prevent, right? I'm not sure I'm going to be able to sleep tonight after the images we just saw. We invite Maria Marcela Giron, who will be giving him an award, a gift for his participation. Now we will hear the presentation called Detection of Arboviruses in Mosquitoes of Public Health Importance to Strengthen Entomovirological Surveillance in Chile. This presentation will be presented by Carolina Reyes, the head of the section of entomology and genetics of vectors of the Department of Genomics and Molecular Genetics. Carolina is veterinarian from the University of Chile with a master's degree in science, specialized in entomology. Since 2008, she has been working as a professional at the reference lab of medical entomology of the Public Health Institute of Chile, where she received a specialization on morphology of triatomines equilicidos with a focus on morphology of immature status of culicidos. These past years, she has worked with the line of work of vector genetics and entomological surveillance and has collaborated in projects and consultancy and entomological consultancy to the different regions throughout the country in research of entomology and vector genetics, as well as coordination and management of scientific coordinations, trainings, and specialization course in the same area. Carolina has participated in several publications of journals, as well as in the creation of a book. Carolina, you have the floor. Thank you very much. Good afternoon, everybody. I'd like to begin by stating that our new section of entomology and vector genetics has as one of its main roles, functions, the support to the Ministry of Health in the area of vigilance, surveillance, and control of vector mosquitoes, and which has high significance in our country. And part of that line of work is that we did together with the infectious agents, or the genetic of infectious agents, and we supported this project from the Ministry of Science, and I'll tell you a little bit more about it. I would like to begin with this image that represents, I believe, with no necessary words, the importance of mosquitoes as vectors of diseases that are significant for public health, and these are the viruses that they transmit in the world, and the most dangerous insects that we know of, these very small insects that have double the death that we cause as humans. And based on that is that we know and we have heard on several occasions the viruses that these mosquitoes transmit, and Aedes aegypti, it's a type of mosquito that we've heard several times for Zika, yellow fever virus, etc., among others. And on the table to the right, we can see how this vector not only transmit these four viruses that we know, but others, and not just that type of mosquito, but also other kinds and other species. And that's the reason why our lines of work are focused on identifying and understanding that this knowledge that we have uh, globally is something that we are working on nationally. This is happening, people are dying. These are the distribution of dengue globally as of March 2024 and February 2025. And we have a big area with a lot of cases. And most of the areas is the Americas, and we are there in the Americas with a lighter color, but surrounded by all of these other countries that, as of 2024, as it's seen in the graph below, which is a PAHO graph, and as of 2024, we had almost 5 million cases, and it was almost 5 million on 2023. So we basically tripled the amount of dengue on 2024. We are basically surrounded by a time bomb as a country due to these diseases that are transmitted by this mosquito. But as a reference lab, which is why I included this figure there, all mosquitoes, all of these mosquitoes that we know have a biological habit, and this habit is consuming blood from animals or people. And it is because of that habit that they transmit these diseases. And not only Aedes aegypti, which is the main disease vector, but there are other mosquitoes that are the most common mosquitoes that in other countries also transmit diseases. We decided that in this project that we didn't want to left out these other viruses and other mosquitoes that transmit other types of viruses, not only the Aedes aegypti. And last year in Argentina, there was an exponential increase in the equine encephalitis of the West. And because of this, the goal was basically ask the following question: are there enough samples of the mosquitoes of Easter Island? We know this is a key place for Chile, and then in the north area of Chile. And putting this in context of the red area, because we are surrounded of countries where the virus is transmitted. So what we wanted to do first was characterizing this virus, vector virus, of vector mosquitoes in Rapa Nui and in the north of Chile, and we wanted to check which mosquitoes we were talking about, check what virus these mosquitoes had, and implement the technique of RT-PCR in real time in our institute. And if we had any positive samples, we wanted to characterize these arboviruses. Our the places where we collected samples were Arica, Parinacota, Tarapacá, and Valparaíso. In this case, Valparaíso was just Eastern Island. So what did we do? We traveled to these places because the samples that are sent from the Ministry of Health are dead in alcohol, and if they had the virus, we're not going to be able to detect it with those types of samples. So we had to implement this from scratch, from the beginning, from the moment we took the sample, so that we afterwards transport the sample properly so that we could detect the virus here at the institute. We went and took samples to Arica and Parinacota, to the city specifically, and in the Azapa Valley, and then in Tarapacá, specifically in the city of Iquique, and in Alto Hospicio, which is located more to the mountains of the city, and then in Easter Island, in the Angaroa area, where most of the population that lives there. We wanted to collect samples of Aedes aegypti and also Culex fasciatus. That is a vector that transmits diseases, and it's another gender. The way that we collected it was, as you can see in the image up there, with an aspirator, like a vacuum, a manual one, and with a net. We were the bait. human baits, basically. And with traps that we use here at the Institute that has a bait that could attract the mosquitoes. It's called bee trap. We collected adults, and what we did was going to the local labs of the ministerial regional secretariat that was supporting us, and we had to identify the gender, the species, through the microscope of the taxonomy keys, but we needed to be careful of not uh, uh, having the mosquitoes alive. So we had to take care of that. Our friend, the refrigerator with low temperature diminishes the metabolism, makes them slower, and we can put them in a Petri plate and then refrigerate them again to put them in a conservation medium afterwards. So in the case that there was a virus, these mosquito wouldn't die and we could detect it in the lab. That was a critical aspect of our research because what we wanted to do first was transportation in liquid nitrogen. However, the transportation of this tank of liquid nitrogen and was impossible. Was impossible to transfer via air, and we had to do something that is used in other countries, which is RNA later solution as a medium of conservation. So we had to put the mosquitoes alive, and they would die uh, drowned on that medium, and that way we would conserve the DNA. We adapted a protocol that we learned in the Evandro Chagas Institute in Belém, Brazil, in 2023. We used this protocol to extract the RNA. So we had the mosquito immersed in the liquid, we washed it, we put it in liquid nitrogen, then we crushed the mosquito, and then we did the RNA in the cellular culture, and then we did the extraction of RNA through RT-PCR in real time. We knew if it was positive, we would go to cellular culture or to analysis of the entire genome. And the results per region are as follows: the pools, the species, the amounts, and for the pools we needed to have the females because the females are the ones that eat blood and transmit diseases, not the males. So from all those samples collected, we had to divide and separate the females. Those are the ones that we included in the study. We studied 225 females with 65% of pools, and of course in Arica, most of the species were Culex quinquefasciatus, and with larvae, because we didn't have more, and we decided to include these larvae anyway in the study. And in Tarapacá only Culex quinquefasciatus, and Valparaíso Rapanu Aedes aegypti. We analyzed dengue, Zika, chikungunya, yellow fever, alpha virus, that includes a lot of encephalitis, and the West equine encephalitis virus, which was the outbreak that occurred in Argentina. And all of those were negative, fortunately. Results: virus, non-positive, and with this standardized technique, the controls were all positive, okay, and therefore the negatives were actually negatives. So what are the discussion and conclusions of this project? That we were able to implement the extraction technique of total nucleic acids. We were able to detect the viral RNA of the mosquitoes because the controls were positive, and the detection through RT-PCR in real time. This is installed in our lab as a new technique that it is available, and we are doing a few adjustments to it. But these are the techniques that we learned at the Institute Leandro Chagas, and after a visit we received from Mexico, we complemented it, and the absence of arbovirus in mosquitoes of the study, that's a conclusion, and the fact that those are all negative. That's a positive result for surveillance, the surveillance that we have in the country. This is a positive result because it also shows that there are no autochthon viruses seen in people, and because in the study we were able to adapt the transportation strategies, which was also a critical aspect, and also the storage of the samples so that we could conserve the RNA in the case that we have a positive result in the future of arbovirus, and in the case that we need to use this technique as routine techniques applied by the ISP and the Ministry of Health whenever we want to analyze mosquitoes. And something else that was interest of the project was the optimization and the quality of nucleic acids. So this allows us from now on to implement other trials. So with this first part that has already been standardized, we can start working on other projects about pesticide resistance, also some from to understand what are mosquitoes eating. So this wasn't just a contribution on the viral search, but also it was an opportunity for us to continue with other projects and to strengthen, the title of the project states, to strengthen the surveillance that we do in our work. Thank you. Thank you to the section of infectious, section of genetic agents and also vectors. Thank you to those teams. Thank you, Carolina, for your presentation. It was very easy to understand, very significant to know the improvements you've made on sampling. The head of the Department of Surveillance and Research is going to give you an award for your participation.
Let's continue then. The next conference is whole body vibration measurements of a sample of Chilean workers from a gender and migration perspective. This presentation is going to be brought to us by Valentina Astudillo. Valentina is a civil engineer in sound and acoustics from 2014. She has been involved in an active way in the implementation of acoustic solutions, participating in the elaboration of projects from the creation of technical and financial proposals, as well as design, installation, and measurement after the installation. From 2017, she has been specialized in the area of occupational hygiene, especially in the evaluation of sound and vibration in work environments. She has experience in the carrying out of measurements on the field, elaboration of technical reports, and the design of proposal of control oriented to the reduction of professional problems or injuries due to the exposition to loud noises. She also is training other so that they can correctly evaluate the agents on the field. Currently, Valentina is an analyst professional in the sound and vibration section in the Department of Occupational Health in the ISP. With no further ado, I will leave Valentina so she can bring her presentation. Hello, everybody. Good afternoon to each of you. I must thank first my internal team in the my colleagues who have supported me in this project, as well as thanking those who participated in the Ministry of Science that helped us with the statistics of the project. I want to highlight that this project was carried out with the funds of the Ministry of Science, and also we counted with the approval of the Ethical Scientific Committee. So that you know a little bit, the introduction, the vibrations, an occupational vibration is in different labor that is carried out in different work conditions. Regarding the full body, it has evidence that it produces an increase in physical illnesses, whether it be neurological or having to do with the bone or the muscles, and depending on the intensity and the duration that these might have, and the different occupations, and so in the different works. So, in spite that Chile has this recognized since the 70s up till today, there are still very low surveillance and action and policy and inspection of these agents, even though last year there was a new protocol that allows these people to be a little bit more visible, and we are hoping to continue with this growth regarding surveillance and prevention of the consequences and the risks. Regarding this project as well, in the case of Chile, we have guidelines or norms from 1999 which approves the guidelines in the workplaces, in which it indicates that the Public Health Institute has the authority as reference. So, considering all of this, this project is a pioneer to be able to know the reality of occupational consequences of vibrations, and in this case would be in transportation, especially in trains. That is why the objective of this project was to quantify the exposition of the physical agent and also validate a questionnaire that could be used to assess the knowledge of the risk associated to the pain that the workers had. That is why also that this study is an observational study, transversal and descriptive. Mainly, it is focused on measuring in a quantitative way the workers in transportation. And to do so with the financing, we were able to obtain a team that analyzes vibration, which is what you can see, which is a plate with its calibrator that is especially for the whole body, where this equipment allows us to measure the 3 axes in a simultaneous way, which allows us to see results that can be trustworthy and representative and reliable, and we don't have to have them separate, which can have some differences and can have a could affect the project. This project has 2 parts. The first part, which is the quantitative measurement of vibrations in the full body of the workers, and one second part that has to do with a survey. And in the first part, thanks also to 2 companies of the trains of the state that has the transportation of passengers in the metropolitan area, and as well as those that have cargo. They opened the doors and they allowed us to visit them and carry out the measurements with them, as well as giving them information regarding their workers, whether it be those who were the machine workers or the helpers, who allowed us to carry out evaluations and have a certain amount of samples. The samples that were calculated through a study and an analysis where we had the support from the Ministry of Science, where we considered the train tracks and whether it was passenger or cargo and the routes, and we had a minimum size of sample that was of 8 samples. Of these 8 samples, minimum samples, we consider that we needed a little bit more because, as I said, this is completely new for us and we are evaluating and we want to generate alignments with this. So therefore, we need to have more sampling to see any extraordinary condition that could happen. And that is why we had 41 measurements in the passenger train. 41 in total, 30 in passenger train and 11 in cargo trains. And logistics also allowed the transportation of cargo to be also a little bit more difficult to evaluate, since Maybe it is one a day, and usually it's at night, and they are much longer because the speed of the train is very different than the speed than when you have passengers. Also, you have to consider that for this sampling that you can see here, where what I am showing you is a passenger train in the Alameda Nos route, you can consider the conditions of cargo and the weight of the passengers and also the train tracks, since they also consider the track itself has to have a different state than what the cargo trains have, and they also have different composition and different technology. As you can observe on this table, we had 16 samples in the passenger Alameda Nos, where the three first accelerations that you can see here show the results that we had in these 16 samples. After this, in the next three columns, you can observe the limits in the times of exposition according to the norm that we have today, the guideline, and we can also observe that the limits were not exceeded. We have the following route that is Alameda Rancagua that also carries passengers, where there are 14 samples, where we can also see that there are three accelerations that is equivalent to what we see on the field and the limits according to the times. And there was also, they did not go over the limit, as I said, and it was within the norm. We also can see the samples of the cargo trains, in which we can see at first we have the three first accelerations of the cargo trains on the field, and we can also see the limits, the allowed limits according to the exposition times, and they were not exceeded either. This is for cargo trains that go from Alameda Barrancas, which is in San Antonio, and then also from Alameda Yayay. The second part of the survey had to do with mainly taking everything that had to do with the survey for a study to be able to have a pilot campaign. This had 5 different stages, in which the 5th stage had a final version. We had carried out many surveys with the team of the Ministry of Science that had to do with those who channel them as questions of communications and to make sure that they were well understood. And also we had the specific part of those people who were specialists in vibration, where we could see if they could understand the part of the occupational questions and also the different illnesses that could be related to everything having to do with the muscle and skeleton or neurological. After these 5 versions of surveys, the last survey, which was the last one, we carried out a process of a pilot that was carried out even here with the mobilization people of the Institute, where we had 7 samples trying also to reconsider migration and gender, which were also included in these surveys. With this pilot, we can validate the survey, and it makes this survey be completely we can rely on this. It is trustworthy, and it allows us to continue analyzing. Regarding the analysis of statistics, all of the results were taken from the software of the calibration equipment that we showed you before, that it comes with a software. We got the results and we put it on a tab, and in this table it is organized in the different routes, whether it be passenger or cargo, and also due to the technology of the train. Not all trains are the same. Some of them have technology that are much more up to date, and some of them only have one driver, and some are older where they have a machine driver and also a helper or an assistant, because of the what they can and cannot see. The compositions change, and that's why it's very interesting to be able to broaden the sample and not leave it only in those 8 minimum that were required. And we organize the tables, so we see them and we compare that with the analysis and with the survey and with the validation of the content and the pilot. And then also we broadened this survey to the drivers of the trains, where they also carried out a survey, and there were some we had to adapt some to be able to continue with the validation within the discussion of the project, which gave us the measurements carried out in the evaluated routes allowed us to see that the routes are within the norm, at least nationally speaking. And that it is also very important that these companies can continue with monitoring in a constant way the conditions that the workers have regarding occupational vibrations for the use of certain of these trains. Why? Because the compositions change. We also have to always consider, ideally, everything that has to do with the kind of the tires or the kind of tracks and the type of train and the composition of the train. However, part of the development of the method recommends as well to continue with the pilot survey. Because those who do not know the story, you know that this was long data. We had to cut it down, and therefore we finished it and concluded it. We were able to carry out measurements and have conclusions from these measurements. However, we are still lacking to have certain things like, for example, validating the survey with a sample that is much broader, where we could have higher samples. And I say this because these 7 samples were only men, for example. So to be able to see gender equity and migration was not evaluated within the same study or survey. So our conclusion, the measurements, as you could see before, within the tables of the cargo or passenger do not go over the vibration limits from the guideline, the national regulation at this time, as well as for the exposition time that were declared in each of the workers. And also, we recommend that these monitors, that they can continue monitoring and it be constant so that they can continue with the analysis, whether it be for this industry or for other or different kinds of tracks, and where it can be also from the different transportation vehicles as well, because that is the objective. Vibrations are everywhere and they're hurting the people. And therefore, we also, due to the conclusion, part of the what we say is that the validation says that it is a tool that is clear for this, and the idea is that this can be used in the future as well. By those who have to drive, we have to take care of the health of these workers, and we have to validate this survey. How can we do that? Obtaining more samples. That is what we are lacking to do as a section and as occupational health, having to do with vibration. And I have said everything I had to say. Thank you very much. Valentina, please don't leave. This is all very interesting. Your project, thank you for highlighting things that are not known for many workers. And now we will give you the acknowledgement who will be the person in charge, the head of occupational health, Alexi Ponce. We will now continue with the program today, and we are going to present now the work of the selected oral presentations of the posters. The first one will be carried out by Pamela Perez. The title of her work is called Innovation in the Treatment of Cystic Fibrosis, helping with the efficacy of ciprofloxacin against biofilms with microcapsules of gelatin. So I leave with you, Pamela Perez. Hello, everybody, and thank you very much for being here today. As I am Pamela Perez, a student from the University of Valparaiso and also part of the Laboratory of Pharmaceutical Tech and Microbial as well. And I'm going to talk to you about the last that we have been working on in our lab, which is based on the formulation of these microcapsules of cyclofluoxacin that are covered. First of all, let's talk about cystic fibrosis. Cystic fibrosis is a pathology that is genetic. that has the way you handle it is very complex, high cost, and it has a lifelong span that is very low. How does this affect us as a country here? We had a screening between 2015 and 2017 where we could see that there's an incidence between 1 and 1 in 800 and 1 in 10,000 of the newborns that were affected by this pathology. This is characterized by the hypersecretion of mucus, which is very thick and hard to eliminate, and therefore it generates an environment that is ideal for microorganisms. Therefore, these people have an infection that is very frequent. And it is important to mention that this is the resolution of these infections are a marker of morbidity and mortality, which is very important. And the infections that are most important are those that are the microorganism Pseudomonas aeruginosa, which is in many places, as they mentioned previously. And this is a pathogen that has a large amount of virulence and can form biofilms. And as biofilms, as you can see on the bottom, is a community of microorganisms that are protected by a matrix, extracellular matrix, and this works as a physical barrier and chemical barrier for the immune system or messenger cells and also therapies, for example, antibiotics. Therefore, it promotes the antimicrobial resistance, which is a global problem that is very important at this time. So, what are the current therapies? Right now, they have oral therapy and inhaling therapies. In oral, we have low bioavailability, and since we are talking about cystic fibrosis, let's talk about bioavailability in the lung. And how do they try to counter this? It would be with the increase of a dose, but increasing the dose, we have as a consequence systemic toxicity. On the other hand, having to do with inhalation therapy, we have platforms like nebulization, and this doesn't have that problem to reach the lung. However, it does have administration times that are prolonged and high frequency. This means that the patient has lower adherence, therefore they promote therapeutic failure. And lastly, we have what are inhalation of dry powder. So they're versatile, they don't have doses problem, however, there are not very many on the market, and the excipients are not all approved by the FDA. So, taking into consideration these disadvantages and advantages of the products that are on the market, our laboratory proposed a platform of inhalation of dry powder. So we have to also take into consideration the challenges that this presents due to the making sure that the actives reach the place where they have to go. For example, with the ciprofloxacin to be able to reach where they have to go. And as a laboratory, we formulated microspheres of this product. And this way it had good results on the biofilms. It had an action time of up to 6 hours. However, we had a problem. The problem was that we could not modulate the liberation of this active. Therefore, when we optimized this formula, we had a follow-up of problems and solutions.
The first step was micronizing the active, and this would increase the surface, and we couldn't make sure that the active would get to the site. And in order to solve this, we proposed to cover it with a lipidic layer, but this acted as a gel that wouldn't leave the active leave. And that's when we added the porogens. And we had the microcapsules that were breathable of ciprofloxacin with this lipidic layer that is discontinued in the forming of pores. The first step was the production. Regarding the production, the technique that we opted for was drying through atomization. This drying through atomization has an advantage that allows us to encapsulate, dry, and amorphize with one step using strip of and we use jello. And this is one of the a few excipients that are used by FDF for the airways. And we use palmitic acid to have work on this rigid wall, and we use polymer surfactant of PPC to allow the exit of the active, and we use it in what we had in our lab in different formulations, such as D-manosa, and sodium chlorine. And there you can see our microcapsules. So the formulations that were tested during these trials were ciprofloxacin, amorphized ciprofloxacin, microcapsules without porogenes of ciprofloxacin, and ciprofloxacin with different pores. And to release the profile of ciprofloxacin of the microcapsules, and by this I mean the speed in which it is released from the capsules, we use these front cells, and that allowed us to simulate the environment, the same environment as a lung environment. So what we did was to compare the remnant amount of what you see in the donator compartment with the ciprofloxacin that went through the dissolution mean. For the evaluation of aerodynamic properties that could be affected by this layer, we use the aerolyzer RS01 coupled by the new generation cascade. And when we concluded this trial, we took the plates and we analyzed the absorbance of it. With that, we can obtain the average aerodynamic. And lastly, and most important, was to evaluate the properties of these biofilms of pseudomonas. And we did this through adjusted suspensions that were incubated in plates of 12 recipients for the treatment of for this treatment with microcapsules, with porogenes and without porogenes, and what would be compared as the ciprofloxacin, free ciprofloxacin. 125 micrograms per ml that had a time of treatment of 6 hours and of 4 hours, and that were suspended, and then they were taken to drip seeding. The idea was to reduce the preformed biofilm. Therefore, our results were the following. Regarding the liberation profile, we see this beautiful graph that shows the percentage of ciprofloxacin release with the time. The sampling times were of 6 hours. We see that ciprofloxacin that was not treated had a maximum dissolution up to 3 hours of 3.4%, which was improved when it was amorphized, reaching 24.9%. But we can see a substantial improvement when it was encapsulated and when we used the porogenes, and it almost reached 40%. Regarding the aerodynamic properties, which was one of the things that we were most concerned about, you can see on the screen that this represents the percentage of ciprofloxacin that was deposited throughout the cascade graph. And you can see in the graph this port of induction that represents the the pharynx area, or pharynx area. And the ones in blue are the ones that are deposited throughout the lower respiratory cycle. What we can understand from this graph is that there isn't a big difference between those that were tested with the D-manosa microcapsules with the encapsulated ciprofloxacin, and if compared with the respiratory fraction, our microcapsules with porogenes have a higher percentage of respiratory fraction if compared to other products in the market that reach 20 up to 40% of respirable fraction. Therefore, we can guarantee of the arrival of this component to the site of action. And regarding the efficacy to the area, which was one of the things that we were concerned about, you can see the following. We have this graph that talks about the viable cells of the biofilm in this logarithmic scale versus the times of treatment that we said were 6 to 24 hours. In this case, you can see that microcapsules with porogenes managed to have a higher inhibitory effect compared with the reduction of the pre-existing biofilm. And at 24 hours, it completely eradicates the biofilm, something that the other formulations didn't achieve. And we can say that this formulation that isn't covered with a porogen and it's just a microcapsule and with a non-processed ciprofloxacin continues to have these remnant units 24 hours later. Therefore, we can say the microcapsules with porogens improve solubility? Yes. It has a significant improvement compared with other formulations. It also improves the efficacy of biofilms and it manages to eradicate all of the biofilms after 24 hours, the Pseudomonas biofilms, and the aerodynamic properties aren't substantially affected by this layer of porogens. We actually can have a respirable area that's better than others that exist in the market. Thank you very much. Thank you, Pamela, for your presentation. You did wonderfully. The second presentation will be presented by Javier Bravo. The title of his work is called Improved Production of a Novel Anti-SST2 Therapeutic for Ulcerative Colitis. Javier, you have the floor. Good afternoon. My name is Javier Bravo. I am a candidate to doctor's degree from the Catholic University of Valparaiso, and I work in a lab led by Dr. Claudia Altamirano to produce recombinant proteins. Today, what I want to share with you is how we can apply different strategy of processes to improve the production of these recombinant proteins. Some context. Monoclonal antibodies are within a market that's called the biotherapy market that includes biopharmaceuticals that are recombinant proteins or nucleic acids, recombinant nucleic acids, but it also includes cellular therapy. Specifically, in this market, we have monoclonal antibodies that represent the highest sales. You can see here it represents about 80% sales in 2021. And we are not choosing any biological system to produce these complex molecules. But there is a system of continuous mammal cells, which are the ovarian cells of Chinese hamsters, that have been used because these comply with certain characteristics, and so that these have become the standard for the production of these monoclonal antibodies. And some of the characteristics of them is that these cells have been able to adapt to culture mediums free of in suspension and free of way of saline solutions. And when I talk about this, we're talking about different that includes bioreactors, and this has an orbital vegetation and have an engine to have this mechanic work. And from a structural point of view, monoclonal antibodies have two heavy chains, two light chains. And what I want to highlight here is that the antibodies present different structural characteristics that give them critical attributes of quality. And one of the most important one is glycosylation pattern, the glycosylation, I'm sorry, glycosylation, which has specifically the 1297. And why is this important? Well, because we have observed that these glycosylations, because these antibodies are glycoproteins, right? So this glycosylation impacts the therapeutical activity of several antibodies. And in some cases, this can also influence the average life of these antibodies on the blood flow. And so when we produce these antibodies for the industry or the academia, we have to consider these quality attributes. And the study model that we are using, some of the researchers like me in our group, is the innovative or this novel anti-SS2T2, and this is led by Dr. Tutorov and Marcela from the University of Chile, and this has a therapeutic potential to treat ulcerative colitis. And one of the effects that we've seen from this antibody is that it allows to control and reduce the inflammation in samples of patients, and this is all for in vitro applications, okay? And in some samples of patients that have ulcerative colitis and Crohn's disease. And these heal the tissues, and this confers this SST2 therapeutical potential that we decided to study so that we can improve its productivity. So what we're asking ourselves in the School of Biochemical Engineering is how can we develop strategies, rational designs that allows us to optimize processes so that we can improve the glycosylation pattern. And we did this for SST2, but it can also be applied to other kinds of monoclonal antibodies to improve its therapeutic activity. And this is exactly the topic of my doctor's thesis. We have developed this along with the University of Manchester for the United Kingdom. And the first goal of my thesis that I did was to do the regeneration of a cellular line that express in a stable manner this anti-SST2, which you can see in the figure down below, where we transvected different lineages through electroporation and then linearization of these antibodies that produce SST2 were affected by the standard, and we apply this. Then we did the cloning because we don't want to have a pool where the expression of the antibody is sequential. We don't want to clone. So we finally created this cell banking, and we used this cell so that we could continue with the next objectives. So in objective number 2 of my thesis, what we did was to do a selection of different supplements that from now on we are going to call precursors of the glycosylation, and this will help our culture medium and impact this glycosylation that we are applying to this recombinant protein. And we study the effect of these supplements through a experimental design of the type of box banking to look at the responses on these methodologies of response surfaces. These are some of the results of this analysis, and we can see that specific supplements, in this case, described as supplement 1 and supplement 2, this impacts the viability that the cells can have on the cultures in time. And as an example, a culture of mammal cells can last up to 8 to 10 days in general, and from day 6 on, the viability starts to decrease. We can see that there are determined specific concentrations of these supplements that manage to have high viabilities, above 100% of viability on day 7 of culture, which represents a significant improvement at the process level of these monoclonal antibodies. And a second result that I want to share with you is that in addition to these concentrations of these supplements, these impact the productive capacity that these cells have. You can see here that we can go from certain conditions where we have about 60 to 65 milligrams per liter of anti-SST2, a condition in which we are getting close to 90 milligrams per liter of antibody. And for the projection in the academia, it's a really good result. And what I am showing you here is an experiment of validation. And so based on this response surface, we get this optimum point in some of this optimal supplementation. In some cases, commitment, we cannot have all of the response variables. So here we are seeing the behavior of this experiment of this response surface. You can see on the left the graph of growth, this continuous line. You have to remember that these are suspension cultures, so we see millions of cells per milliliters, and these correspond to the viability in time. You can see the blue line, which is the one that has a supplement, grows faster than the control line, which is the black one. And so these supplements make the cells grow faster. And on the upper right side, where it says IVCC, that is the accumulated integral of viable cells, accumulated cells. This is the area under the growth curve. This means the blue curve that has the supplement has a bigger area under the curve, which is the black curve. This means that these supplements have a higher level of live cells in time per volume, unit volume. And what I'll share with you later, that's a very important and significant result. From the productive point of view, these supplements also increase the productive capacity. And as you can see here, for the supplements, we checked that it can reach several milliliters per milligrams per liter. And I'm going to stop on the graph that's on the middle where it says QP. This is the specific productivity. PCD means picograms of recombinant protein per cell per day. And so therefore this is the contribution that each cell does in the bioreactor. And why are we producing more of this? And not just because we have more cells, but because each cell uses the carbon sources more efficiently and therefore produces a higher amount of protein every day. Therefore, what we do in our school is try to explain this behavior through metabolism, and we can see that these cells, when are affected by the supplements, it has a differential behavior. They consume less glucose and produce lactate slowly. And some of these results could be related to a metabolism that's more oxidative, that's generally more related to a more productive, because those oxidative lines create more GDP. Like we say in the School of Biochemical Engineering, this strategy allows us to increase the volumetric productivity of the system. This is the only equation that I have in my presentation. The volumetric productivity is the Q in capital, the capital Q, and that's a volume of production rate. The QP, the capital Q is a volumetric production rate. The other Q is a specific production rate. It's XB is the viable cells concentration. How many cells do I have in my bioreactor? And as I was saying, it improves the non-capital Q, the specific production rate, but we also have an area under the larger curve. So we have more cells in the bioreactor for a longer time, and this means that we can affirm that we have had a successful development of a productive line of anti-SST2, and we have done this stable, and with that we have been increasing the volumetric productivity. So for each unit of volume of our bioreactor is much more efficient. And to conclude, what I want to say, comment, is the work of one of my colleagues, Francisca Torres Garcia, another thesis student. What she asked herself is, how can we develop strategies to improve the protein bioprocessing of these recombinant proteins? And we have a lot of bottlenecks in these secretion lines when the protein goes from the topology to, and basically the decision that Francisca made is study how implementing novel antioxidants can impact these bottlenecks of this secretive line and improving the assembling of the proteins in the reticulum. These are the results, and here you can see the curve to the left, where we can see the growth, and we can see the curve that has growth and has increased 100 to 1,000 micromolar. And we can see that purple line has higher values. So the supplemental antioxidant, it's in its lowest concentration, makes the cells grow faster, and the small one to the right, right below, we can see the QP non-capital, and the concentration is low, and it also increases specific productivity of the cells. And that isn't something that happens when we supplement the with an excess of antioxidants, and we can see it's not it does not behave as it was in the control one. And then we can go and discuss the flow. When we see the graph to the left, you can see that there is a culture of control with no kinds of supplementation, and the cells of the bioreactor change the oxidative state on time from day 0 to day 5, and it moves, and this histogram moves then to the right, and therefore the cells oxidize and accumulate the intracellular in time.
In the middle, you can see once again the control on day 5 in blue, and if on day 5 we supplement with a concentration of low antioxidants, we can then reverse in a partial way that oxidative stress that the cells are having over time and get closer to the state that initially they had. So on the other extreme, if we go too far and we supplement too much antioxidants, we can promote the appearance of reactive species, and this seems more intense oxidative stress. And this is very interesting because you have to be very careful with antioxidants in certain concentrations. Antioxidants have their behavior that is desirable to prevent the emergency. However, in other conditions, they do become a substance that can be pro-oxidative. Therefore, we have to be careful and have to take this into consideration if we want to have a production. We have to find the point of best condition where we can capture in a better way the behavior of the antioxidant that we desire. And to conclude and to close this presentation, I just wanted to say that, as you already saw in the results, we developed in a successful way in the university. There is a producing line of anti-SS2 T2, and we use the CHO cell line, and it allows us to have a better comprehension of how the different phenomena in the environment that these cells grow in are giving impacts that are intracellular in different compartments, and at the same time how these intracellular can impact the productive phenotype of these CHO cells, and how this can allow us to produce in an efficient way biotherapeutics. And to close, I would like to thank the different sources of financing that include NIDO and also the United Kingdom, and also I would like to thank you for your attention. Thank you very much, and if anybody has any question, I will also be available to answer afterwards. Thank you, Javier, for your innovative presentation. We hope you have great success in your doctoral thesis. The third presentation corresponds to Hamilton West. The title of his work is Influenza A virus at the human-animal interface. So now, here is Hamilton West. Hello, everybody. I'm going to present briefly the results of many years of investigation and research from the veterinarian from the university. And we have the University of Chile, where we have been seeing the influenza A virus in human-animal interface. Influenza A virus have reservoirs in birds, especially those who are in water bodies, and also we have seen some viruses that can cause infections in other species as well, and some of them, through mutation and adaptations, can have been able to be in other species, and they continue circulating them. For example, in the pigs, for example, some stational one, and also marine mammals. And also they are recognized as a threat, zoonotic and pandemic. We already have had many pandemic having to do with influenza virus, the last one being H1N1 of 2019. Globally, there is always a very high interest of what happens with the influenza viruses, especially since some subtypes emerge that can have a severe disease, that have high pathogenicity, and not only with subtitles, you can within the context of these species that are the reservoirs of the influenza virus, that are the birds who are connected to water bodies, they have migration. So with these movements that go around the world, they could also be moving different kinds of viruses and generate a connectivity globally. These viruses that you are seeing in the wild birds are especially they are severe, and sometimes it can be something that is not severe. Others, they can also have mortality in the birds, and in some cases when the virus H5 and H7 have contact with domestic birds, they can have some kind of mutations that make them go even to another where you can have high pathogen infections, and this is probably the circulation of the influenza in the last few decades. However, from 2005, there is a subtype in Asia, which has been maintained over the years, which is GSGD, and it is having high pathogenicity, and it can generate infection in some individuals and some species of wild birds without causing disease or mortality. So this continues circulating globally in high pathogen, and we have not had events of this virus until 2014, 2015, where we had the first bird flu, but it's mainly in North America. And a few years ago, we just had the first epidemic of bird flu in Central America and South America. And we have the presence of this virus, and I can imagine that most of you have seen what has come out in the news many times, where you can have it in wild birds as well as marine animals, and even some humans here in Chile, and some and one in Ecuador, the avian flu. So what did we know about this avian flu before? Unfortunately, we didn't know very much. The Central America and the Caribbean and South America are known as the areas that have less attention having to do with research of the influenza virus. There is not very much information before this. However, within our region, Chile also has some particularities, where it had been the only country that had a high pathogen in 2002. One of the cases, and it had other events as well. Low pathogen affecting the industry of birds in different years, but associated to low pathogen virus. So with this framework and this theory, with our group, we started our research around a decade ago. We have been in an interface where we find that it's very relevant that the society and the production of animals in the backyard, that maybe you don't know it by that name, but when there is any rural path, when you go out to the more urban part, and you can find that on the streets, you can see some chicken or some animals that are walking around, and all of those animals have an owner. And many times they're associated to production systems of people who are more vulnerable, who are associated to more vulnerability. So they also to be able to sell in product of higher needs. So in this production, especially the birds, the chicken, and pigs are the species that are most predominant, because they require low investment to be able to purchase them and also to maintain them. And they also generate food and the possibility of sale in case they may need to. And in this interface, there is a close contact with these wild animals and domestic animals, including animals, the birds, the pigs, and people. So this is very interesting to see what happens with the influenza virus. And here, summarizing many years of work, we started carrying out research, especially in the central part of Chile and the subpopulations, and we find that there is a higher infection than what we thought. 20-25% prevalence and a positivity of PCR real time around 80%, with higher positive in the central part of this area of studies. We can characterize how this type of productive systems work and isolate some virus, for example, the swine flu. And it is very interesting because there's also the virus from the pandemic and also human virus, and it also is transmitted in models, so they are risk for people. And you could transmission in ferrets, so it's risk for people. So we could see what is happening in these reservoir populations. So we went to the place where they have concentration of wild birds in Chile, and after a lot of work and a lot of samples that we were able to analyze, we could identify the greatest amount of diversity in the subtypes that are in Chile. We were able to identify that these viruses are circulating throughout the whole year, with peaks of prevalence in the months of spring and fall. Another important thing is that we can describe that we are in the presence of South American lineage, and it has been evolving for many decades, and these have been detected, and they are very different from North America and Asia, for example. And the circulation of these viruses are also associated to environmental issues, for example, vegetation. And we also have the income of stations from those who come from the northern hemisphere that have a different lineage, and they have different recombination in our territory. We have been able to study how this interface is moving between the wild birds and the domestic birds who are present in the different production systems in the backyard, and we can identify that there are also other species of birds that can have have be a part of the transmission as well. And many times they have not been taken into consideration in this context of the study of influenza virus. And they are the ones who can be doing a contact that is very important between the concentration of wild birds as well as those who are maintained in the producers. With all of this information, we have also studied what happens in this interface, and we have started with the study of some of the human cohorts. But before that, we also started to characterize all of the different things that people do with these animals in their backyards, because the interaction that they have is very important. Many times with the same handling that they have every day, and go from when they give them food, and many times in these homes where they have these backyard birds, there is a contact between people who are elderly or small children with the birds who are on the in their property. And we have been carrying out this activity with these animals that have with the birds. We can see risk factors that can be combined with the different, so we can see the probability of the transmission of these pathogens and identify that there are some conditions in particular in some scenarios that could allow the transmission of these influenza virus that are in the animals towards the people. I just wanted to show you some of the things that happen in these properties. Sometimes you have the consumption of the property that is carried out in the same production site, and these are different things that they have to do, from the sacrifice of the animals all the way to all of the where they have the possibility of high exposition to the possible pathogens, not only the influenza virus that are in these populations, but sometimes even what is the animal residues can be given, they can pass on to other animals on the property as well. And this is something very interesting that we have to take into consideration and study. And also important that many times these people, as they have a lower educational level, they don't have access to information, and they do not consider this as some kind of a risk factor or take any measures to be able to prevent any kind of infection with a pathogen that might be present in this species. With all of this in mind, it is very important to be able to continue carrying out this kind of study, because we have different information about the different subtypes that can be circulating in the species that are in these production sites. And I said even pets that are on these properties could have influenza virus, especially with the human influenza virus. But when these have co-infection, in these scenarios of co-infection, for example, in the same house where we can have the virus of different origins that are circulating in these different species, you have the possibility of a transmission of them, and we can see some events of recombination, and it can make subtypes that can come up that can cause infection in people. And if we think about this in the context of the risk that we have at this time of the income of new virus of bird influenza, of avian influenza, we need to be more cautious and pay more attention. Other elements that have happened in the last few years in our region are very relevant. For example, these infections that have happened in marine mammals, and also people that make us be more alert to all of these situations. I cannot go give you the final results that we have been able to identify in humans yet, because they are still in the process of sequencing at this time. But what we hope to see, if we can identify some virus from animal origin that is infecting people, we are trying to identify these viruses that can circulate in people who handle animals, and also generating serum banks with what we find to see if there is the presence of antibodies for them. This is the current scenario. More than concerning, maybe we should be more alert on what could happen, since we know that there is important circulation of the avian influenza in our region that maybe has not been taken into consideration. It's not so visible, but with the income of the new virus from the northern hemisphere year after year, where we have more probability that new avian influenza can come into our area, and we can also have a contact with the different production sites, and it can have different effects and different ways of presentation in the different animals, like has what was happened, for example, in influenza that is causing infection in bovines in the North America, with a manifestation that seems inapparent, that doesn't visible. However, it is still a risk for the people who are exposed or are in contact with these animals. This was briefly what I wanted to present. These are many years of work with different groups of investigation, of research, especially the University of Chile and the Catholic University. And this is the excellent, this is different, here we mentioned part of the, who has been a part of this throughout the years. Thank you. Thank you very much, Hamilton, for sharing your research with us, very connected to the concept of One Health. This is very interesting. So now we will carry out a small break, but before that, we would like to invite you to visit the different posters and that are at the beginning of this tent, and then share a coffee here at the side of the tent. We will see you in a few more minutes.
We must continue with our program.
Así que los invitamos a
So
sentarse.
I invite you all to take a seat.
Muy bien. Volvamos, por favor, a la
Everybody,
carta
please
principal.
take a seat.
Tenemos que dar continuidad
We
al
must
programa.
continue with our program.
Nos falta aún una
We have
conferencia.
one more
Nos
lecture.
falta la premiación. Todos estamos
We
muy
have the awarding ceremony
expectantes para ver
as
quién
well.
o
We have high
quiénes son los
expectations on
ganadores y
who will
ganadoras.
be the winner.
Sí, voy a apagarles la luz y le voy a sacar el alimento.
I'm going to turn off the
Vamos al
light
café,
and
vamos a
remove
las galletitas.
the food, the
Vamos a
coffee, the
empezar.
cookies. We're going to get started now.
Como les comentaba,
And as I was saying
dar
earlier,
continuidad a
we
nuestro
want to
programa. Nos
continue with
falta
today's
ya
program,
parte
and
final.
we are just have
Pero antes
one
de
presentation
comenzar,
left.
quiero darle
But
la
before we
bienvenida
begin,
y
I
también
want to
darle el
welcome
agradecimiento
and I would like to
a
thank
Elizabeth Vera
Elizabeth
Mancilla.
Vera
Ella es
Mancilla.
presidenta
She
del
is the
Consejo de la
president
Sociedad
of the
Civil
Council of the
COSOC,
Civil
quien nos está
Society, COSOC,
acompañando
that
hoy al término del
and she is
programa.
joining
Muchas
us
gracias, Elizabeth, por tu compañía.
today to conclude the ceremony. Thank you so much, Elizabeth, to be with us today.
Continuando con el programa, corresponde
And
la charla
let's continue with the
denominada
program. We
Estudio de
have the
percepción
lecture
sobre la
called
utilización de la información
Perception Study on the
contenida
Use of
en
the
el contexto del
Information contained in
medicamento,
the Drug
así como las
Package
preferencias
leaflet,
para la
as well
información
as
en
the
físico o en
preferences for
formato
information
digital
in physical
por
or
parte de los
digital
personal
format by
médico,
medical personnel,
pacientes o usuarios
patients, or
finales.
end users.
Esta presentación
This
está a cargo del
presentation
doctor
is in
Jorge
charge of Dr.
Fuentealba,
Jorge
decano de la
Fuentealba,
Facultad de
Dean of
Ciencias
the Department
Biológicas
of
de la
Biological
Universidad de
Science
Concepción.
of the University of Concepcion.
El doctor Fuentealba
Dr.
es químico
Fuentealba is
farmacéutico
a
de la Universidad de
pharmaceutical
Chile,
chemist of the
máster
University of
en
Chile
estudios
with a master's degree in
clínicos.
clinical studies.
Tengo un... Deme un segundo.
Give me
Los voy
just
a dejar
one second.
expectantes.
I'm
Sí, es muy
giving you a
importante la
lot of
conferencia, por
expectation. The
eso.
conference is very, very important. Just give me one second.
Parece que nos toca rellenar.
I think I'm going to have to buy some time here
¿Quién falta?
because someone's missing.
Entonces vamos a esperar
All
un
right,
poquito.
let's wait for a
Por favor,
minute.
paciencia.
Please
No se
be
devuelvan al
patient.
café,
Don't go back to the coffee
porque me va a
area
costar mucho traerlos de nuevo.
because it'll be very, very hard
El
for me
único
to get you
permiso
back.
podría ser que vayan,
I
tomen
allow
una
you to
galleta, un
go grab
café y
one
vuelvan.
cookie,
Pero vuelvan, no hay
coffee,
más,
and come
¿cierto?
back right
Esperemos
away.
un poquitito,
Okay,
por
so
favor.
please, let's wait for a few
Podría
minutes.
preparar alguna dinámica,
Maybe I could
pero
have
no.
prepared
Ahora sí
something,
voy a hacer la
some jokes or
presentación.
something. Maybe we can have the presentation or something like that.
Me dicen acá que no confían en mi rutina de chistes que tenía preparada.
They do not trust on my jokes, and so we're going to play a few greetings. I guess you've seen this, but after this we will do an assessment to check if you learned these videos by heart, and these are some greetings from people who are helping us with this conference.
así. Me iba a acercar a cada uno de ustedes para pedir algún tips de relleno. Se salvaron. Se salvaron.
We are now going to continue with the program. First of all, I would like to apologize to Dr. Jorge Fuentehabla for the wait and things that happen when we are live. And today he is going to present study of perceptions regarding the use of information contained in medication package insert by as well as the preference for if they prefer to be in physical by the patients or the doctors and the final end users. I'm going to repeat once again, this presentation is going to be carried out by Dr. Jorge Fuentealba, who's Dean of the Faculty of Biological Sciences in the University of Concepcion. Dr. Fuentealba is chemical pharmaceutical in the University of Chile and Master in Clinical Studies of the Autonomous University of Madrid, Doctor in Pharmacology in the Autonomous University of Madrid. He is a professor of the University of Concepcion and was president of the Society of Pharmacology in Chile since 1917 until the year 2019. He was Chilean representative for International Union of Pharmacology from the year 2017 until the year 2021. He is an external advisor of the Committee of Incorporation of New Medications in the Hospital Guillermo Grant de la Benavente from the year 2020 till date. Till today, and he was also coordinator of the consultative committee accreditation committee during 2019 and 2020. Author of 67 publications, 2 patents, 7 chapters of different books, main researcher of projects Fondesit for close to 15 years, scientific director of the Advanced Research Center of Biomedicine from 2009 until today. He has led 15 postgraduate theses and more than 30 pregraduate. He has been acknowledged with Henry Nestle Award 2011 and 2012 in the version of science. Without any more ado, I will leave now with you, Dr. Fuente Alba.
Good afternoon. I'd like to thank for the invitation of participating in this conference. And the second thing I want to do is to congratulate you, because ever since I arrived to this Congress today, I've seen that the level of the presentations have been at the highest level. So congratulations to those who presented and also to the organizers. All right, so this is the title of the presentation that we are doing today with my colleague Humberto Fierro. And this is a study that had the goal of doing an evaluation of the perception of the use of drug package leaflet. And this had the participation of the Ethical Committee of the University of Concepcion. It was sponsored by the ISP, and also had the sponsorship and support of the Chamber of the Pharmaceutical Industry. The goal of this study was to assess the usefulness and efficacy of the information included in the information leaflet included in the package, the medication packages, and the information and to know what's the perception from the users as well as those who are the distributors of this medication. How was it written, whether it was in a physical format, or maybe if it was possible to consult that information digitally. It included 4 questions, this study, and it was related to the patterns of behavior, desirability, preferences, and literacy in health, and it had a initial stage that included a validation of the survey. And to do so, we invited 50 users and also 50 healthcare professionals. With that, we validated the survey, then we executed the study, and lastly, we assessed the results. This protocol included 300 users and 300 professionals, and we were able to recruit or invite 399 users and 238 healthcare professionals. Here you can see some examples of the questions we made. When you need information of medication, do you look at the information within the package from the users? And you can see in the one in black, 52% of them said that they did looked at it, and of healthcare professionals, 61%. When the first question was positive, then they were asked if it's necessary for you to ask somebody else to help you understand the information contained in the package, and most people said that they didn't need help and they can look at it on their own. And why were the reason of why they were looking at it? For those who were patients or require medications, the main frequency were adverse effects and the use while taking other medications. And the reason of why healthcare professionals said in a 77% looked at it because of adverse effects, and others looked at administration. Then we asked them how they would qualify the usefulness of the information included in that leaflet, and whether if it is practical or. And most of the questions are that the information can be very useful, 44%, or somehow useful, 54%. Other questions, such as if you're not looking at the leaflet that's included in your medication package, why is that? 45% of users said that they could not find the information they were looking for, while 32% said that the information was hard to understand. On the other hand, healthcare professionals, the frequency of response was that 47% said that it wasn't convenient because it wasn't easy to find this information. And in second place, among other reasons, and some that said that had no access to the leaflet. And how understandable this information is, and 46% said that it was mildly convincing, and 21% said that it wasn't understandable. And why was that reason? And the main one that you see in question number 6, 61%, 34%, 34% said that the information is not contained in the leaflet. We also asked them, what other sources of information are you consulting? And 43% said that they asked this information to their doctors, and 49% also asked in the pharmacy, and 53% looks the information on the internet. And what other sources healthcare professionals were consulting? 91% said that they search information on the internet, and 22% in the pharmaceutical lab to check the origin of the medication. What other information is value from those sources of information, and what users value is the rapid, fast access to the information, 74%, while healthcare professionals value the convenient and fast access to the information with 74%, and the information that they needed was included in this leaflet with a 58%. Why do they use other sources? 60% of users says that information that they are looking for is about the disease, and adverse effects, 77%, while healthcare professionals, as you can see there, says adverse effects and interaction with other medication, 77 to 55% respectively. Do you have access to the internet? Users, 97%. Professionals, 100%. How often can users take a look at it on a daily basis, and they can do so with sources of the internet. And healthcare professionals, 98% is permanently searching for that information on internet sources. Then they were asked on a scale of 1 to 3 if the access to the information of the leaflet about the medication would be easier if there was an increase in the access to information, understanding of the information, trust on the information, and convenience. And you can see that in the user group in gray and healthcare professionals in red has a higher valuation in all aspects. Access, understanding of the information they are given, the trust of the information, as well as the convenience. And when they are asked if they would rather obtain the information on a digital format instead of a printed format, and 34% of the users, patients, said that yes, they would like to have it on a digital format, and 38% says both. Would you prefer to get the information of the medication on a digital format instead of the printed format? And 73% of the people who were surveyed said yes, and 24% said that they prefer to have both. This is a summary of what I have already mentioned about the preference of the prospect versus the digital format, and in general terms, professionals, users, and the totals, and that represent 51% of the preferences preferring digital format. I don't want to entertain you with the summary of what I just showed you. So I want to share with you the main conclusions of the study that say that there is a clear preference about the interest of migrating to digital systems due to two factors. One, the rapid, how fast it can be, and then how trustworthy it is, the information required. The other thing is that it is necessary to have an effort between or in the institutions so that we can provide these work together for healthcare professionals, and so that we can offer this to the users. This means academia, the regulatory entity, and the companies have to work together to give us information to the users, because what we finally will achieve with this is equity to the access of the information of the medication. And Santiago has the possibility, uh, Santiago has a lot of pharmacies and a lot of medical consultations, but whenever you leave the capital, or those macro cities, the level of rurality increases rapidly. So the access it's more difficult to have. And thank you so much for your attention. Thank you for inviting me to present these results. I am very happy to be here. Thank you, Dr. Fuentealba, for your presentation. This is a topic that is very important for us at the Public Health Institute. Thank you so much for the conclusions. We'll see how we can continue to progress. We invite now Marcela Giron, the head of surveillance and research of the ISP, who will give you an acknowledgement for your participation. And we are concluding our conference. This took a long time, and we had to do an assessment of the presentations that you watched throughout these days, and we also assessed several posters that were presented during these 3 days. To do the awarding ceremony, we invite Ms. Catalina Ferezio, our director, and we would also like to invite Inepsi Diaz, who's the head of the Department of National Agency of Medical Devices. Please come forward. Let's begin with the awarding ceremony of the posters, and we're going to begin with the third place. The third place is for Cristobal Quintul. Is Cristobal Quintul here? He's not here. And no, we're not choosing another third place because he's not here. You're a representative of Cristobal? Okay, yes, please come forward. The title is The Analysis of Legislative Gaps and Proposals of Recommendations for New Synthetic Drugs and Possible Use of Abuse Drugs in Chile. Benjamín Bustamante is representing Cristobal. Congratulations. The second place is for José Conejeros. Is José here? Or someone representing José from his team? All right. The title is Space, Temporary Trends and Social Determining of Tuberculosis in Chile, 2011-2021. 21 implicances for the prevention and control. He's not in the room, but let's give him a big round of applause. Maybe he's watching us through streaming. We are going to give him his gift of acknowledgement. And first place, hopefully he's here, Felipe Hernandez. Is anybody from Felipe Hernandez's team? He is probably watching us from the streaming. Epidemiological surveillance of mpox and detection in Chile. And Alejandra Acevedo is representing Felipe Hernandez. We have concluded the awarding ceremony of the posters. Let's now continue with the awarding of the oral presentations. The third place is for Rayana Santos Araujo. Is Rayana or someone from her team here? She's running, coming here. Her title is Application of Remote Sensing for the Prediction of Infectious Diseases in Body Waters in Chile. In water bodies in Chile. Rayana. The second place is for Nicole Brunel. Is Nicole here? I think she is Nicole. It's a follow-up to women infected with BPH, persistent BPH from 2014 to 2024. Big round of applause for Nicole. In the first place is for Maria de Los Angeles Spencer. Is she here? No? Anybody from her team? Dr. Catalina Ferrecchio. This is a self-awarded award. Development of a diagnosis algorithm for liver disease in primary care. Big round of applause to Maria de Los Angeles, represented by Dr. Catarina Ferrecchio. It's from her team, so that's all right. I don't know if you notice, and we're linking this to the inauguration because at ISP, for us, it was very important to acknowledge the women who have led scientific research. And if you paid attention, all winners are women. So first, we want to give these women a round of applause because they are leading in research, but we also want to give a physical acknowledgement to the winners. So please come to the stage to receive this award. Rayana, Nicole, and Dr. Caterina. It's the first time that this happens. Big round of applause for all the winners, and to those who didn't win, congratulations for the presentation. So innovative, very clear presentation. Thank you for the participation of the posters, the oral presentations as well. And now I want to introduce Dr. Janet C. Diaz, who will also provide a few words, and then Dr. Catarina Ferrecchio will conclude and close the day.
First of all, I would like to ask Dr. Eugenio Ramirez to come onto the stage. He's going to speak in representation of the scientific committee of this conference in reference to the quality of the works and the papers we have received for this great activity. Dr. Eugenio. Thank you very much. The truth is that we are highly surprised. First of all, due to the high quality and quantity of the works that we have received, the papers we received. We thought it would be an intra ISP, and it ended up being a scientific round, and 75% are external to the Public Health Institute, which is something that has never happened before. And all of the selection of posters and the oral posters was very complicated because, as I said before, the quality of the work was very good. And I hope that many of these papers these work turned into papers. I support the legislation in health and that they support primary care because you represent very important progress in our country. I would like to thank each of you who are present here for the time you dedicated to this and all of the authors and all the researchers. I think this is a detail. If you had any kind of acknowledgement or award, That is just something small at this point. When you observe that each of the works that were that were turned in, were very high quality. I am very proud. I have not been here that many years, but I am very, it's a joke. I have seen many young people who know a lot and have an amazing potential for this country. This morning I was reading what was happening in the United States, the leak of many young researchers. I hope that never happens in our country. And what is happening here is the opposite. I believe that success of this days, of this conference, is due to you, to those who are present, those who came to listen, and especially those who took the time to present your research. Thank you very much. We would like to thank you for the participation of each of you and the organizing committee for these conference. And as you know, this happens every 2 years. So we are waiting for you in May of 2027, and we hope we can have even more than what we have been able to carry out today. I'm going to give you the numbers. Our director is going to give you the numbers. That is her surprise. But I would like to thank each of you for the participation, the quality of your work, and I would like to invite you to send these works of the journal of the ISP that she is here representing there in the back. You can see who, you can give them to her. I invite you to publish in our magazine, in our journal. You have amazing work, and there is a large amount of them. Therefore, thank you very much. And I will leave you now with the director of the Public Health Institute, Doctora Ferrecho, and who is going to give the closing words for this great conference. Thank you. Well, we have numbers. I'm going to give these numbers now. Record numbers, it seems 600 people who have circulated during the 3 days that we have had this encounter. We have had visitors from outside of Chile, from the different countries in the region, in such an important moment for the world. This is very important to have the collaboration. So, as you said, while in other moments people would leave, at other occasions we have people who are still coming in, and we are very happy. And I agree. I was in almost everything, and I was partially online watching streaming, which was amazing. I find that technology, and I congratulate the company who supported us, because everything was impeccable. And the level of the presentations, I completely agree. It is very high quality, and I am very happy. And I would also like to present the new president of COSOC, who is here with us, Elizabeth Mera. Please stand up so we can recognize you. She was chosen. We called to a consultation. There was 60 people who applied, 60 organizations, and she was the one chosen. It was very competitive. And we are going to have a very good group, a support group for our social part. And I would like to thank you for your patience. I know we're a little bit late. That's why we have less audience, but everybody who is here, you are very loyal. Thank you very much for being here with us, and have a great rest of the day. Thank you. We would like to thank the words from the director. We have had them. We are very happy with the amount of people who have been in this event with us, and with this news, we would like to finalize the 17th version of Scientific Conference of the Public Health Institute of Chile. We appreciate your participation, and we will wait for you. We hope that you can be with us in the year 2027.